We would like to welcome you to this session. These talks will be for 2 minutes, and the first talk is entitled Investigating the Role of the Ciliary-Associated Protein AHI1 in Cortical Development using Animal Models and Human Brain Organoids. Ms. Costanzo is going to present.
Good morning, everyone. My name is Panagiotta Di Costanzo, and I'm a master student in Professor Kirousis' lab, where we investigate cortical development. During corticogenesis, neural progenitors proliferate to increase their number and then differentiate into neurons that they will migrate to the cortical plate. Alterations in these processes can lead to malformations of cortical development. Malformations of cortical development, and studies link these malformations with the primary cilium, which is a small organelle that is believed to play an important role during development. Specifically, mutations in the ciliary-associated gene HI1 have been identified in the malformation polymicrogyri. So, in the lab, we have manipulated HI1 expression in vivo, and we have shown that there is a change in the progenitor's position to more basal locations and presence of ectopic neurons. We have also observed changes in the numbers, orientation, and length of the primary cilium, as long as disruption of the apical belt at the site of manipulation, which indicates cellular delamination. We have also shown that HI1 exhibit different expression patterns between species. So we have already started our analysis in a human-specific cerebral organoid model, and our first data indicate that there is a change in the neuronal output. To conclude, we propose that primary cilium is a master regulator of cortical development with species-specific differences that we would like to further investigate. If you have any questions, please feel free to come and find me after the session. I'll be very happy to discuss about our project more. Thank you.
Thank you very much for keeping the time. The next talk is by Dr. Kokinopoulos. Is he here? Not yet. Probably the third talk is by Mr. Kokorakis. The talk is entitled Mirk-Dirk 1-big kinase controls the generation of the columnar organization of spinal motor neurons via sonic Hedgehog pathway.
Maybe we can go and move to the fourth until they're ready, so there was a mix-up. Is the presenter of the next one here? Evaluation of induction of fetal hemoglobin? No? Okay, I will call Christina Pascu, who is definitely here because I know her. So, So she's going to present her work on cell-intrinsic pathological characteristics in A53T alpha-synuclein iPS-derived astrocytes from Parkinson's disease patients.
Good morning, everyone. As you probably already know, Parkinson's disease is one of the most common neurodegenerative disorders that still remain incurable. Alpha-synuclein protein has been related to familial PD, and the mutation causing the amino acid substitution AFT3T has been linked with early onset and severe PD pathology. Our question is whether AFT3T mutation causes intrinsic deficits in PD astrocytes. In order to address our question, we used our previously established model of induced pluripotent stem cells to generate midbrain astrocytes from healthy and PD donors. Both healthy and A53T induced astrocytes express typical lineage markers such as vimentin and CD44 and aquaporin, and exhibit functional characteristics such as the upregulation of interleukin-6 upon cytokine stimulation. A53T astrocytes had higher levels of expression of alpha-synuclein and increased size of phosphorylated form at serine 1 to 9 of alpha-synuclein aggregates. Moreover, the most prominent phenotypes of A53T astrocytes include cytoplasmic vacuolization, aberrant calcium transients, and the dysregulation of genes associated with the unfolded protein response, such as BIP and ATF4, both at basal condition and after subsequent-triggered ER stress. Also, A53T astrocytes had higher levels of increased protein aggregates, increased in size, and also after phagocytosis assay, we observed that the A53T astrocytes possibly have impaired clearance capacity. Finally, the proteomic analysis of PD astrocytes versus the control revealed that in affected pathways there are the regulation of autophagy and the regulation of endocytosis. Our observations suggest that AFT3 mutation can cause intrinsic dysregulation in astrocytes that are related with the proteostasis and the clearance mechanism that may have a possible critical contribution in the PD pathology. Thank you.
Maybe we can move on with Dr. Kokorakis.
Good morning, everyone. Our study concerns the elucidation of the functional role of DYRK1B kinase in CNS development. DYRK1B kinase is normally expressed in developing neural tube and is a known inducer of cell cycle exit. And in chick embryonic spinal cord, through gain and loss of function studies, as well as phenotype rescue studies, we saw that DYRK1B induces cell cycle exit and neuronal differentiation in a cell-autonomous manner, while in a non-cell autonomous manner induces in a ventral side of spinal cord apoptosis in a motor neuron domain, and the loss of motor neuron and interneuron progenitor and the postmitotic populations. And this intense phenotype connects DYRK1B and Son-Hedgecock, since we have found that DYRK1B is a transcriptional suppressor of Son-Hedgecock and its effector GLI3. And this crosstalk between DYRK1B and Son-Hedgecock is reflected on populations of motor neurons, specifically in LMC, medial motor column, at a later developmental stage in cheek spinal cord. And all were co-expressed with Son-Hedgehog. And all this data suggests that there can be acts as a potent transcriptional regulator of Son-Hedgehog pathway through which it regulates the survival and columnar organization of spinal motor columns in the developing cheek spinal cord. Thank you.
We move to the next talk, evaluation of induction of fetal hemoglobin synthesis by genome editing of cis and trans-acting components of the beta globin locus, by Professor Lederer.
So it's really work by Vaniota Papasava, who couldn't come today, an excellent first MSc student, then PhD student, then postdoc, and now she's resident hematologist with us. The poor woman has been with me for a while. So this came as a sideline of editing for HBB IVS 110 for therapeutic use. And it turned out that when we designed a CPF1 Cas12 molecule for the mutated locus, that we also edited very effectively a delta globin, as is shown here, because there's only very few mismatches. And it turned out that this introduced a 7.4 kb deletion, which resembles either delta beta thalassemia mutations or high persistence of fetal hemoglobin mutations. Now, the first one would just leave a patient ill if you perform the editing. The second one might be therapeutic. So we wanted to investigate which of the two is the case. And doing this, she looked at the editing efficiency, and most relevant here is this deletion event. And for this, she developed a digital PCR assay that shows a high efficiency of 25% of fusion production that is specifically detected with this. The remainder of the events are about 60 to 50% of single edits on each of the two loci that are cut in beta and delta globin. And this is here compared to the editing of two different repressors of gamma globin, the editing of which might also be therapeutic. So we can compare how this edit in terms of induction of gamma globin would compare to these other two therapeutically relevant loci. This is in UDEP2 cells, erythroid cell line that very much resembles CD34 cells and CD34 cells. Now, moving on to hemoglobins and globins, we find actually that this Cas12a CPF1, relatively speaking, highly induces HbF compared to the other two modifiers, so that would indicate that it might be therapeutic. And when we then look at globins as well, we see that gamma fold increase is much higher here for this CPF1 editor than for the other editors that we used. And across experiment, this is a highly, significantly different compared to control. So this might be therapeutic. Now we have to analyze if this also in absolute terms is increased, and for this we have to look at clonal analyses of different editing events. Now for HbF induction, similarly we find that 40% of cells are HbF positive compared to other editing events for the other modifiers, which again shows that Cas12a is superior, at least in relative terms, to the other edits. Well, that's my department. Here's Mariota once again, and thank you for your attention.
Thank you.
Next speaker, Ms. Margariti. She's going to talk about in vivo microglial BIN1 deletion following LPS-induced neuroinflammation, regulates microglial proliferation and inflammatory responses.
So, good morning, everyone. To begin with, we know that microglial activation is a crucial player in the pathogenesis of Alzheimer's disease, but the contribution in the AD progression has been controversial. BIN1 is an adapter protein that regulates membrane dynamics as well as apoptosis and endocytosis, and there are SNPs in the locus harboring the gene that show the second strongest association with AD after apolipoprotein E. However, the role in microglial activation of BIN1 remains to be clarified. So, our purpose is to investigate the effect of microglia-specific BIN1 deletion in the mouse brain under homeostatic and inflammatory conditions. To this end, we developed a double transgenic mouse here, and we inducibly knock out microglial BIN1 after tamoxifen injections, and we cause neuroinflammation after an LPS injection. We performed a single nucleus RNA sequencing, and now I'm going to show you some of our results for the microglial subpopulations. As you can see here, we have two subpopulations that are absent in the homeostatic conditions, and their numbers increase in the BIN1 knockout model. We characterize them, and the first is characterized by the upregulation of pro-inflammatory genes, such as the STAT1, and the second is characterized as a prone-to-proliferation microglial subpopulation, as it's been marked by the KI67. So to conclude, we identified two different microglial subpopulations enhanced after the LPS treatment and further increased in the BIN1 knockout model, and we can verify that BIN1 regulates microglial proliferation and pro-inflammatory response in the adult mouse brain after the induction of a neuroinflammation. Thank you very much.
Thank you.
So we move now to the last talk, efficient epigenetically mediated reactivation of gamma globin expression in an immortalized human erythroid progenitor cell line.
Good morning, my name is Papadopoulos Fotis, and I'm gonna present our work regarding efficient epigenetically mediated reactivation of gamma globin expression. Over the last few years, development of genome editing tools has enabled the targeted introduction of genomic mutations for the reactivation of gamma globin as a therapeutic approach for beta hemoglobinopathies. Despite the promising results that have already been emerged, there are still limitations due to the generation of nucleus-mediated double-strand breaks. The development of epigenome editors offers an alternative and less invasive approach for targeted gene regulation. In our study, firstly, we established a HUTEP2 cell line that stably expresses the epigenome editor CRAB with Firstly, we established a HUTEP2 cell line that stably expresses the epigenome editor DCAS9 with CRAB suppressor. Then, we designed guide RNAs to target cis-regulatory elements of three master regulators of gamma globin expression, namely BCL11A, LRF, and recently identified GNF410. In order to introduce specific epigenetic alterations, we target specifically BCL11A's erythroid enhancer, along with LRF and GNF410 promoters. After transduction with lentiguide viral vectors, cells were cultured in expansion medium for 6 days, and after that in erythroid differentiation medium for another 8 days. As we see, transduction with lentiguide viral vectors and epigenetic suppression were well tolerated and didn't affect cell proliferation, neither during expansion nor maturation. Although we did observe a small reduction in HbF in cell numbers while targeting LRF promoter with LRF guide 1. During expansion, we didn't notice any significant differences in HbF levels between our groups. However, in later stages of maturation, we did observe significant increasement of HbF positive cells within transduced populations in all of our groups, and especially when targeting GNF410 promoter. Overall, epigenetic-mediated inactivation of three master regulators of gamma globin expression seems to be effective, leading to an to a significant HbF induction. Following these experiments, we want also to investigate if the same applies in primary cells that comes from healthy or beta thalassemia donors. Thanks for your attention.
Thank you.
Thank you.
Yeah, so we can move to the next speaker, Ms. Thanu. She's going to talk about the chemical injury by chemotherapeutic agent, RSC, and how it induces myelin dysfunction and ectopic neuroblasts recruitment in the brain.
Sorry, I'm short. This is a larger talk, so don't expect to be in 2 minutes. So my name is Irini Thanu. I'm a PhD candidate in the Hellenic Pasteur Institute. Okay, recently studies have given insight into the mechanisms of the so-called phenomenon of chemobrain. Among these mechanisms are increased inflammation, reduced neurogenesis, and of course, problems with myelination. In our model of chemical lesion, we performed 2 intraventricular infusions of the chemotherapeutic mitotoxic agent RSA, commonly used in the chemotherapeutic protocols. After the RSA infusion, in different time points, we study the integrity of the ependymal zone and SVZ integrity. What we observed is that even from day 4 after the infusion, we had a tremendous disruption of the ependyma integrity, as well as ependymal cells and neural stem cells, as you can see in all these pictures. As expected, we had extensive inflammation with astrogliosis and microgliosis, along with a heavy wave of phagocytosis with a very high expression of CD68, especially located in the lateral walls of the brain. Another indication is known also from the literature. It's a glial scar is replacing the loss of ependyma in the SVZ. Further, what we notice is, especially at the early stages of after the infusion, we noticed cell apoptosis and infiltration of peripheral cells, especially T cells, in the brain parenchyma, in the striatum, and also an accumulation in the choroid plexus, suggesting that there is a loosening of the blood-CSF barrier. So moreover, we noticed doing different measurements of the G ratio, which is the ratio of the axon diameter to the diameter of the fiber, that we have demyelinating effects going on in our model, collapsed axons, as well as a decrease in the oligodendrocyte lineage and a decrease in the level, the protein levels of myelin protein CMPAs. So, oops, this didn't. Okay, you will not see the whole picture. Sorry. Beside the SVZ neurogenesis disruption, to our surprise, we noticed that there is a huge accumulation of ectopic double cortin cells in the striatum. With 3D reconstruction and confocal imaging experiments that we'll show you right now, we evaluate that these ectopic cells are vastly located inside myelin structure, something that usually it's not the case in other models of injury. Having an insight of what is going on with these ectopic cells, we noticed that their maturation state is heavily dependent on their location inside and outside the myelin structures. So what we can see here is that the cells that are located inside the myelin bundles are mostly, they are organized in chains and having a migratory phenotype, and they are expressing immature markers. And in contrast to the cells that they are located in the brain parenchyma, the gray matter, that they acquire a mature phenotype, neuronal phenotype, and expressing mature neuronal markers are CTP2, which is a medium spiny neuron marker. Okay, now we were questioning what is going on with the other neurogenic niche of the brain, the hippocampus. So we checked also the different time points, but now I'm showing only the 6 weeks time point, where we observed that there is a large increase in the numbers of the double cortin positive neuroblasts. A result that it's not expected in a model that we have a problem and an inhibition of the proliferation with the RSC. What we also observed is also in the case of the hippocampal neurogenesis, we have in a different pattern an abnormal migration outside the limits, the usual limits of the migratory roots of double cortin inside the niche, so deeper in the granule cell layer. So we suspect, along with other data, that the problem is in the maturation and integration of these newly born neurons instead of an increase in neurogenesis per se. For more, we can check Mika's poster with all the rest of the data. Okay, so the arising question in our model is what drives ectopic newborn neurons to follow, especially the route of the white matter tracks. Chemotaxis or durotaxis, two different but also combined patterns. Our primary results show that both we have an mRNA increase of factors that are implicated in both chemotactic and durotactic migration. So, and very recently, we came across a publication in a different context, in a glioblastoma cancer model, where we see a very similar image of distribution of ectopic neurons, in this case glioblastoma cells, inside the myelin bundles. What they say is that plexin B is the key factor that drives this migratory route. So our goal is to check and to focus also in this molecule to see if there is a common pattern between these. Okay, and with this, thank you very much to all my colleagues and all of you that you came to hear all the morning talks. Thank you very much.
Thank you very much. Questions?
Sorry.
Okay, so we can proceed to the next talk.
The next talk is entitled Estimation of the HLA homozygous cord blood units for the production of induced pluripotent stem cells for transplantation and regenerative medicine purposes of Greeks.
Sorry for the long title. Good morning. And thank you for giving me the chance to present you the data analysis on the estimation of the cord blood units required to cover the HLA matching requirements of the Greek population. These are actually preliminary results from a very effective collaboration between the 3 cord blood banks in Hellenic, Academy of Athens, and Crete. So the need for organ and tissue transplantation is evident worldwide, and besides the the ethical issues that may arise, there are some very major important factors that may pose some very important barriers in this procedure. Some of the most important one is the immunological reactions, and is between the host and the recipient that can cause either host versus graft or graft versus host disease. And some of the major factors also of this organ rejection may be the histocompatibility antigens, which may be coded by more than 40 loci. And the most important ones are the major histocompatibility complex, which is composed of mainly of HLA antigens. The evolution of typing methodologies and sequencing technologies have revealed an amazing 3,700% increase in the number of variants between 1998 and the last month. And following the data analysis of the IMGT database, the latest data, showed that the main loci of class 1 HLA genes, which are ABC, and class 2 genes, which are DR, DQ, and TP, they estimated that there are about 90,500 variants that are coded as coding. So you can realize the vast diversity of HLA, which can pose us a major barrier in finding a compatible donor between a donor and recipient during the transplantation process. So for this reason, alternative methodologies had to be discovered. And as we heard the last 2 days, very promising results have been reported for the stem cell therapy. Besides the adult and embryonic stem cells, a third category is the induced pluripotent stem cells, which are basically somatic cells that have been re-engineered in the lab to look like pluripotent embryonic. I won't go through a lot about the background of induced pluripotent stem cells because Professor Papadakis will elaborate on the matter later on. But just very briefly, in 2006-2007, Dr. Yamanaka and Takahashi managed to overexpress 4 pluripotency-related transcription factors and successfully reversed the somatic human and mouse somatic cells in the pluripotent status. That gave Dr. Yamanaka the Nobel of Medicine in 2012. And indeed, the induced pluripotent stem cells, they look like embryonic stem cells, in that they're similar morphology. They have a similar differentiation rate and a similar stem cell expression profile. And the most important of all is they have a minimum to non-compatibility issues. So the last years, there have been a tremendous research effort to use these induced pluripotent stem cells in clinical trials. And we had a very successful clinical trial in Japan when they used induced pluripotent stem cells from retinal pigment epithelial cells in age-related macular degeneration. But however, the process of developing induced pluripotent stem cells from autologous pluripotent stem cells is very time-consuming and very expensive. So for this reason, the allogenic model is preferred. However, the allogenic model, you have to select a 100% HLA matched donor in order to avoid the IPS rejection. So, however, as we've seen, the diversity of HLA is so vast, so it's very difficult to find an HLA matched donor, 100% HLA matched. So for this reason, an alternative idea has been proposed to develop iPS cell lines from donors homozygous for the common HLA haplotypes. So with this process, you just need a very small number of iPS cell lines that can cover the HLA match requirement of the target population. And an example of this is a Japanese study where they showed that you just need 50 iPS cell lines to cover about 91% of the Japanese HLA matching requirements. As a resource of these iPS cell lines can be the banked cord blood. Indeed, the cord blood contains stem cells from young donors, which have a lot of advantages, which are listed here. So because of the time, I won't go through it. And However, the HLA frequency, allele frequency, is affected by the population structure and demographics. However, the frequency of the top common alleles is not really differentiated between different regions of the same nation. So this is true for most populations. However, if certain and specific rules of loss of Mendelian inheritance are violated for that specific population, like genetic isolation due to common ancestor or geographic isolation, then this rule may not apply. So in order to overcome these obstacles, two very important questions had to be answered. First of all, what is the estimated number of cord blood units that can cover the HLA matching requirements of the target population? And the second question is how easy it is to find these HLA homozygous donors. And these questions are very relevant in populations with specific population characteristics like the Greek population, and especially the population from the Greek islands. As we've showed in a recent study that we did, the comparative analysis between the Cretan population and 20,000 samples from the German bank of DKMS, we've showed a high allelic and haplotypic diversity between the Cretan population and those samples, and especially the diversity was evident when we compared the Cretan population with the Greek immigrants of DKMS, and where we showed that just 21% of the haplotypes were common between the two cohorts. And as you can see on the plotted here of the top 10 haplotypes, there's significant difference even in these common haplotypes. Based on these results, we decided to expand our analysis and formed a very effective collaboration with the 3 cord blood banks and the Center of Bone Marrow Donors in Patras with Professor Spiridonidis and Dr. Kosteas in the Cyprus with the Karaiskaki Foundation. And we decided to map the immunogenetic profile of Greeks and identify the regions of high diversity. And a side project of this collaboration is we used the cord blood units of the 3 cord blood banks. And what we did is we tried to answer those 2 questions I mentioned before. And we used a combined cohort of 2,500 samples that were typed at high resolution for A, B, C, and DR, which are known to affect...
Sorry, you have half a minute to...
Okay, so it's... and a sub-cohort of 900 patients for the other 4 loci. And we try to find the samples that they're homozygous for these loci. And what we did is we used an iteration algorithm, and we calculated the total coverage of the homozygous samples from this population. And very, very fast, we showed that we had 1,600 haplotypes, but only 5 of them were with a frequency of more than 1%. And for the 4 loci, we had 900 haplotypes with haplotype frequency of more than 1%. And plotting this, the top 10 haplotypes, we showed that the very characteristic diversity of the 3 cord blood banks, which is mainly caused by the Cretan population. And we showed that among the 1,600 haplotypes, only 5 cord blood units were homozygous for those haplotypes, and we selected only 1 because only 1 was present in more than 2 samples. And for this haplotype, there was a coverage of 6.9% in the combined cohort, and there was a diversity when we stratified the cohort by the Cretan blood bank, by the cord blood bank origin. So the final estimation was that the total coverage, we need about 100 haplolines for the demand of the HLA matching requirements of the Greek population, which satisfy about 69% of that population. However, if you consider that only 15 haplolines were satisfied the 91% of the Japanese population, you can see the diversity of the Greek population. So in order to overcome these obstacles, because of the diversity of the Greek population, we need a very effective collaboration between the centers. And maybe the haplotypes may not be common, maybe they can also be rare, because what is rare to some may be common to others. And what we're trying to do is we expand our analysis using 4,000 cord blood units from Cyprus, and also collaborating with a very big cost program in order to include some potential IPS cell lines, Greek IPS cell lines, in the World Registry of Human-Induced Pluripotent Stem Cells, which are now, as you can see here, this is 0. So sorry for being over a bit. So if you have any questions, thank you very much.
Thank you.
We have to move forward to Ms. Platania. She's here. Yes. Yeah. She's going to talk about the biofabrication of 3D bioprinted vascular-like structures functionalized with platelet-rich plasma for endothelial tissue regeneration.
Okay, good morning. My name is Varvara Platania, and I'm currently a PhD candidate at the Department of Material Science and Technology of the University of Crete. And today I'm going to present you under the supervision of Professor Maria Hadjinikolaidou. And today I'm going to present you our latest work entitled Biofabrication of 3D Bioprinted Vascular-like Structures functionalized with platelet-rich plasma for endothelial tissue regeneration. One of the main challenges in tissue engineering remains the vascularization of large grafts. With a great demand of organ and tissue transplantation, there is 3D bioprinting is offering us a groundbreaking technique to 3D bioprint personalized functional structures for tissue engineering applications. In our study, cell-loaded bioinks were used to 3D bioprint tubular structures, mimicking the features of blood vessels. More specific, the average height of our construct was 1 centimeter, with an inner diameter of 3 millimeters and a wall thickness of approximately 1 millimeter, similar to small arteries and veins. To this end, 3 key components, gel and gum, platelet-rich plasma, and laponite, were combined to have a synergistic effect. Gel and gum is a well-known biocompatible polysaccharide, which possesses also angiogenic properties. The addition of laponite in our bioinks, aiming to further affect the endothelial cell migration, increase the printability effect, and also increase the pore size of the cross-link network. The functionalization with platelet-rich plasma, from now on PRP, was to further enhance the endothelialization capacity of our construct, because it's a well-known rich source of autologous growth factors and proteins. In this slide, we can see the overview of our work. Human Wharton gel mesenchymal stem cells were isolated, cultured, and then incorporated to our bioinks to 3D bioprint vessel-like structures. These final constructs were evaluated both in vitro and in vivo as prominent candidates for tissue engineering applications. All compositions which were used in this study were extensively physicochemical characterized for their swelling properties, their degradation profiles, and also the incorporation of each and every one component to the final structure. Both bioinks maintained high shape fidelity compared to the respective CAD models, as we can see in the pictures below, after 14 days in culture. Also, both vessel types present high elastic modulus values, over 10 megapascal, which is matching the characteristics of the native femoral and internal mammary arteries. In vitro biocompatibility assessed through a live dead stain, and as we can see from the graph on the right, both vessel types are highly biocompatible with over 95% viability rates. Also, for the PRP vessels, we have enhanced the proliferation of the Gordon gel mesenchymal stem cells, something which is lead to denser cellular networks. The evaluation of vessel extracellular matrix formation was assessed through histochemical stain of the produced glycosaminoglycans and collagen, which is two structural components of the vascular ECM. Both stains are positive, and for the PRP functionalized vessels, we have accumulation points of both glycosaminoglycans and collagen on after 28 days in culture. The endothelial differentiation capacity of the final constructs assessed through immune staining of the two endothelial markers, von Willebrand factor and PECAM1. And our initial hypothesis that functionalization with PRP will enhance the endothelial differentiation, differentiation is confirmed. Additionally, vascular endothelial growth factor secretion levels in vitro were assessed through ELISA, and we have a twofold increase for PRP vessels compared to the control ones without PRP. The in vivo evaluation experiments were conducted in collaboration with Professor Vidaki and Dr. Alpadaki, and histological analysis present no signs of foreign body reaction or immune response toward both vessel types. In conclusion, in our study, we successfully managed to bioengineer 3D tubes mimicking the feature of blood vessels. Both bearings maintain high shape fidelity compared to the CAD models, high cell viability, and enhanced elasticity. Also, further functionalization of the vessels with PRP increase viability, promote extracellular matrix formation, and enhance the endothelialization, demonstrating promising constructs for endothelial tissue engineering applications. Some ongoing investigation is being carried out to further investigate the endothelial potential of these constructs, both in vitro and in vivo. Thank you very much for your attention.
In the time for a question? If not, then we can proceed to the next speaker.
So the next talk is entitled Effect of Stem Cell-Derived Microparticles on Viability, Apoptosis and Differentiation Program of Hemopoietic Cells.
Καλημέρα και από μένα. Αρχικά, το ομφαλιοπλακουντικό αίμα αποτελεί μια εναλλακτική πηγή αιμοποιητικών στελεχείων κυτάρων που χρησιμοποιούνται στη μεταμόσχευση και παρουσιάζει πληθώρα πλεονεκτημάτων, όπως είναι η διαδικασία συλλογής και επεξεργασίας, είναι εύκολη και μπορεί να είναι γρήγορα διαθέσιμο προς χρήση, ενώ δεν απαιτείται αυστηρή συμβατότητα των HLA. Επιπλέον, ο κίνδυνος για τη μετάδοση μολυσματικών ασθενειών είναι μικρός, όπως και το κόστος συγκριτικά με τη συλλογή μυελού των οστών. Σε όλα τα βιολογικά υγρά, όπως και στο ομφαλιοπλακουντικό αίμα, ανιχνεύονται διάφορα εξωκυταρικά κυστίδια, όπως είναι τα εξωσώματα, τα αποπτωτικά σωμάτια και τα μικροσωμάτια. Συγκεκριμένα, τα μικροσωμάτια αποτελούν εξωκυταρικά γυστίδια μεγέθους 100 έως 1000 νανομέτρων, που απελευθερώνονται από την πλασματική μεμβράνη ενεργοποιημένων ή αποπτωτικών κυτάρων και μπορούν να ανιχνευθούν υπό φυσιολογικές αλλά και παθολογικές συνθήκες. Μεταφέρουν στο εσωτερικό τους νουκλεϊκά οξέα, λιπίδια και πρωτεΐνες και με αυτόν τον τρόπο μπορούν να συνεισφέρουν στη διακυταρική επικοινωνία και να επηρεάζουν φυσιολογικές διαδικασίες των κυτάρων, όπως είναι η αιμοποίηση και η απόπτωση. Όσον αφορά την αιμοποίηση, μικροσωμάτια τα οποία προέρχονται από αιμοποιητικά στελεχεία κύτταρα έχουν ανιχνευθεί στο ομφαλιοπλακουντικό αίμα και φαίνεται να μεταφέρουν microRNAs. Επιπλέον, μικροσωμάτια από εμβριονικά στελεχεία κύτταρα φαίνεται να οδηγούν σε αναπρογραμματισμό των αιμοποιητικών προγονικών κυττάρων και μεταφέρουν mRNA για διάφορους μεταγραφικούς παράγοντες, ενώ μικροσωμάτια από αιμοπετάλια φαίνεται να ενισχύουν την εμφύτευση των αιμοποιητικών στελεχείων και προγονικών κυττάρων. Ενώ επίσης μικροσωμάτια από ερυθροκύτταρα φαίνεται να είναι αυξημένα σε ασθενείς που έχουν αναπτύξει οξέα νόσο ομοσχεύματος έναντι του ξενιστή. Όσον αφορά την απόπτωση, αρκετές έρευνες δείχνουν ότι οι μικροσωμάτια από διάφορους τύπους κυττάρων μπορούν να υπάγουν την απόπτωση και να αυξήσουν την ενεργότητα πρωτεϊνών που συμμετέχουν στα σηματοδοτικά μονοπάτια της απόπτωσης, όπως είναι το εξωγενές και το ενδογενές μονοπάτι. Μία από τις πρωτεΐνες αυτές είναι η κασπάση 3. Σκοπός λοιπόν της μελέτης μας ήταν η εκτίμηση της επίδρασης στο CD34 θετικών μικροσωματείων στη βιωσιμότητα των κυττάρων και στην έκφραση αποπτωτικών γονιδίων στην προμυελοκυταρική λευχαιμική σειρά HL60 και στα μονοπύληνα κύτταρα του ομφαλιοπλακουντικού αίματος, όπως επίσης και στο πρόγραμμα διαφοροποίησης των αιμοποιητικών στελεχείων κυττάρων. Αρχικά πραγματοποιήθηκε απομόνωση των μικροσωματείων από ομφαλιοπλακουντικό αίμα και στη συνέχεια προσδιορίστηκε ο αριθμός των CD34 θετικών μικροσωματείων μέσω κυτταρομετρίας ροής. Ακολούθησε ανοσομαγνητικός διαχωρισμός των CD34 θετικών μικροσωματείων και επόαση των κυττάρων HL60 και των μονοπύρινων κυττάρων με την προσθήκη MPs. Έπειτα από επόαση 24 ωρών προσδιορίστηκε η βιωσιμότητα των κυττάρων, η γονιδιακή έκφραση και πρωτεομική ανάλυση αποπτωτικών μορίων και επίσης πραγματοποιήθηκαν οι μυστεριαίες καλλιέργειες και προσδιορίστηκε ο αριθμός των απικιών που αναπτύχθηκαν. Αρχικά παρατηρείται ότι η βιωσιμότητα των κυττάρων, τόσο των κυττάρων HL60 όσο και των μονοπύρινων κυττάρων, μειώνεται υπό την επίδραση των CD34 μικροσωματείων. Όσον αφορά την έκφραση αποπτωτικών γονιδίων στα κύτταρα HL60, η γονιδιακή έκφραση του προαποπτωτικού γονιδίου φας αυξάνεται υπό την επίδραση των μικροσωματείων, το οποίο επίσης επιβεβαιώνεται με την πρωτεομική ανάλυση, όπου ανιχνεύεται η πρωτεΐνη BCL2, η οποία είναι αντιαποπτωτική πρωτεΐνη και ο αναστολέας της BAX, και επίσης αυξάνονται τα επίπεδα της προαποπτωτικής πρωτεΐνης κασπάσης 3. Όσον αφορά τα μονοπύρινα κύτταρα, αυξάνεται η γονιδιακή έκφραση του BCL2, το οποίο επίσης επιβεβαιώνεται με την πρωτεομική ανάλυση. Αυξάνεται επίσης τα επίπεδα της πρωτεΐνης BAX και της κασπάσης 3. Όσον αφορά το πρόγραμμα διαφοροποίησης, παρατηρήθηκε μείωση των προγονικών κυττάρων, κοκκιοκυττάρων, μακροφάγων, η οποία ήταν στατιστικά σημαντική. Συμπερασματικά, λοιπόν, η βιωσιμότητα των κυττάρων HL60 και των μονοπύρινων κυττάρων μειώνεται υπό την επίδραση των CD34 μικροσωματείων, τα οποία φαίνεται να επάγουν το εξωγενές και το ενδογενές μονοπάτι στα κύτταρα HL60, ενώ στα μονοπύρινα κύτταρα φαίνεται να επάγουν κυρίως το ενδογενές μονοπάτι. Η ενεργοποίηση της αποπτωτικής σηματοδότησης στη λευχαιμική σειρά HL60 υποδεικνύει έναν πιθανό αντιλευχαιμικό ρόλο των CD34 μικροσωματείων, τα οποία επίσης φαίνεται να επηρεάζουν και το πρόγραμμα διαφοροποίησης των αιμοποιητικών κυττάρων. Ευχαριστώ πολύ.
Ευχαριστούμε πολύ. Έχουμε χρόνο για ερωτήσεις. Μάγια.
Ευχαριστούμε πάρα πολύ για την παρουσίασή σας. Δεν με ακούτε?
Ναι.
Θα ήθελα να ρωτήσω, έχετε κάποιο πιθανό μηχανισμό που λειτουργούν τα σωματίδια, τα nanoparticles που έχετε τα μικροσωματίδια που έχετε απομονώσει από τα CD34 κύτταρα?
Συγκεκριμένα...
Δηλαδή, για να το κάνω πιο συγκεκριμένο, υπάρχουν κάποια μόρια στο φορτίο των εξωσωματίων αυτών που θεωρείτε ότι επάγουν τους μηχανισμούς αυτούς?
Δεν έχουμε μέχρι στιγμής προσδιορίσει ακριβώς τον τρόπο με τον οποίο γίνεται αυτό. Σε μια προηγούμενη μελέτη, όπου φαίνεται ότι μεταφέρουν κάποια συγκεκριμένα microRNA στο εσωτερικό τους, τα οποία δεν υπάρχει κάποιο μονοπάτι που να φαίνεται συγκεκριμένο ότι μπορεί να επηρεάζει, να υπάγει την απόπτωση.
Εντάξει. Ευχαριστώ πολύ.
Ευχαριστώ. Να ρωτήσω κι εγώ κάτι. Η απομόνωσή τους πώς γίνεται? Ποια μέθοδο, ποια ακολουθείτε?
Υπάρχει το πρωτόκολλο που γίνεται φυγοκέντρηση από το πλάσμα.
Okay. Αυτό με τη φυγοκέντρηση από το πλάσμα.
Ναι,
ωραία. Ευχαριστώ πολύ.
Και εγώ.
Ωραία. Προχωράμε λοιπόν στην επόμενη ομιλία. So our next talk is from, I think, Ganti Nikoudis from Kritis Lab. Yeah? Is he here? Anyone from the team? No? Okay, maybe we can proceed then to the next one. Ms. Dayu. HIF1A restricts cardiomyocyte regeneration. Thank you.
Thank you. So today I'm going to present you some of our in vivo data, which suggest that the transcription factor HIF1 alpha restricts cardiomyocyte regeneration in newborn mammals. Due to the inability of the adult human heart to regenerate cardiomyocytes post-injury, cardiomyopathies remain the leading cause of morbidity and mortality worldwide. Unlike humans, other organisms such as zebrafish and salamanders retain a long life ability to regenerate their myocardium. Newborn mice and pigs can also regenerate their myocardium post-apical resection, LED ligation, or cryo-injury within a few weeks. However, the regenerative window closes only a few days post-birth. In mice, the loss of the regenerative capacity has been linked to a postnatal switch from anaerobic glycolysis to a highly oxidative metabolic environment. Due to the role of hypoxia in this metabolic switch, a lot of researchers have indicated the crucial role of the transcription factor HIF1 alpha, or hypoxia-inducible factor 1 alpha, in cardiomyocyte development and regeneration. However, its exact role still remains controversial. On the one hand, a recent study suggesting that the gradual exposure of adult mice to hypoxia activates HIF1 alpha, and this leads to a metabolic switch toward anaerobic glycolysis and to increase in the cardiomyocyte proliferation. So these researchers suggested that systematically exposing patients to hypoxia could induce their cardiomyocyte proliferation percentage. On the other hand, early experiments by Greg Semenza, who was awarded with a Nobel Prize in Physiology or Medicine in 2019, showed the exact opposite result. These researchers showed that the knockout of HIF1 alpha in mouse embryos leads to an excessive cardiomyocyte proliferation and thus to left ventricular hypertrophy by embryonic day 14.5. So based on this controversy, the aim of this study was to determine the metabolic role of HIF1 alpha in mammalian cardiomyocyte proliferation and regeneration. To this end, one-day-old neonatal biopsy mice were subjected to apical cryo-injury or SAM operation, and then HIF1 alpha or glycolysis was targeted by daily subcutaneous injections for 7 or 21 days. HIF1 alpha was pharmacologically inhibited by the compound YC1 or activated by cobalt chloride. As for the regulation of glycolysis, we used two different dosages of the drug metformin. Metformin is a clinically approved drug for patients suffering from type 2 diabetes, and data have shown that in lower dosages it promotes oxidative phosphorylation, whereas at higher dosages it enhances glycolysis. Immunohistochemical analysis of mouse hearts 7 days post-cryo-injury showed that YC1 successfully inhibited HIF1 alpha, whereas cobalt chloride induced a robust nuclear immunoexpression of HIF1 alpha in almost 100% of the cardiomyocytes. In addition, the lack of positive immunostaining for the hypoxia marker pimonidazole suggested that HIF1 alpha is stabilized in an oxygen-independent manner during cardiac regeneration in the neonatal mouse. Similarly, metformin enhanced the cardiac AMPK activation and led to lower glucose and higher lactate levels in the blood of the mice that received the higher dosage of metformin, but not in the animals which received the lower dosage. Now, in order to verify the role of HIF1-alpha in cardiomyocyte proliferation, we did immunohistochemical staining for sarcomeric alpha-actinine and phosphorylated histone 3 in serine 10, which is a mitotic marker. After that, we measured mitotic cardiomyocytes in the apical, border, and remote zone of the ventricles. Our data showed that inhibition of HIF1-alpha by YC1 significantly increased the percentage of mitotic cardiomyocytes 7 days post-cryoinjury relative to control, whereas the number of mitotic cardiomyocytes was significantly lower when HIF1-alpha was stabilized by cobalt chloride. Notably, the same result was observed in all these 3 regions that were analyzed, and not only in the apical zone at which we did the cryoinjury. Similar results were obtained after the experiments of metformin. More specifically, the lower dosage of metformin significantly increased the number of mitotic cardiomyocytes relative to control, whereas there was a tendency to abrogate these effects when glycolysis was induced with a higher dosage of metformin. These results indicate that inhibition of HIF1-alpha or of the glycolytic rewiring could induce the cardiomyocyte proliferative potential in neonatal mammals. Based on this outcome, we sought to investigate whether this limiting effect of HIF1-alpha on cardiomyocyte proliferation is also accompanied by any changes in the regenerative capacity of cardiomyocytes. What we did was to do a picroseries red histochemical staining in mouse heart cryosections 21 days post-cryoinjury. Our results showed that the stabilization of HIF1-alpha by cobalt chloride significantly delayed the regenerative capacity of cardiomyocytes, as shown by the higher fraction of scar tissue relative to control. On the other hand, when glycolysis was inhibited by the lower dosage of metformin, we observed a significantly accelerated regenerative capacity of cardiomyocytes, whereas again there was a tendency of abrogation of this result when glycolysis was induced by the high dosage of metformin. To sum up, our results show that cardiac regeneration in neonatal mice is mediated by the oxygen-independent stabilization of HIF1-alpha, which limits cardiomyocyte proliferation and regeneration, as well as by the oxygen-independent switch of the metabolism toward aerobic glycolysis, the inhibition of which accelerates cardiomyocyte proliferation and regeneration. Collectively, we believe that our results could potentially lead to the development of new therapeutic approaches for cardiac regeneration in patients suffering from cardiomyopathies through the pharmacological inhibition of these mechanisms. Thank you for your attention.
Thank you. Questions from the audience? Mrs. Dr. Hadjinikolaidou.
Thank you very much for this very interesting talk and the nice results. I wonder if you have considered to investigate also other markers regarding the regeneration potential, because the serious spike is only on the collagen synthesis of the tissue, if I'm not wrong. Right?
Yes.
It's only one marker. I mean,
yes,
other
yes,
markers?
I understand. We could do that. These are our primary results. Firstly, we had focused on cardiomyocyte proliferation, and for this reason, except for these in vivo experiments, we also have a human iPSC model, and we observed the same results. And now we started these experiments with the regeneration, but we're planning to continue with that. Thank you so much.
I have a question, maybe a bit naive. Are there like more specific inhibitors for HIF1A available?
That's a nice question. YC1, we could say that it's not the best inhibitor, but there aren't any better options for now. But that's why we had the in vitro human model of iPS cells, at which we did a complete knockout of HIF1 alpha, and we saw the same effects there. But now our research team has identified a cardiomyocyte-specific target of HIF1 alpha, and our next plan is to target HIF1 alpha through this target in order to have cardiomyocyte-specific inhibition only of HIF1 alpha.
Okay, and what is the rationale behind using these two extreme doses of metformin? The 2.5 and then going up to the 100? Because it's quite a big difference, so you're not like ranging from...
There are previous studies which suggested these two dosages, and we used them because we wanted this biphasic effect of metformin. And the previous data suggest these doses, and we tried different doses as well. Okay. But our data which showed that these two doses successfully enhanced glycolysis or oxidative phosphorylation, respectively.
Thank you.
Just continue from this last question. I would like to ask please remind me, metformin is a diabetic drug?
Yes.
Great. So you probably suggest a new potential use for metformin in cardiomyopathies?
That's right, but there's still much work to be done because we haven't checked any possible side effects. We have only checked that there are no effects on body weight, and we don't have so much data, but that's our plan to see a new role for metformin in the heart.
Very nice, Kirk. Thank you.
Thank you.
So we were waiting for someone to connect, but it doesn't seem to be the case. So I think we're ready to close the session. So thank you all for attending so early this morning. Thank you, Elena, for being here as well.
Thank you. Shall we move to the next session now, or shall we wait 5 minutes?
5 minutes.
Okay.
What the audience?
Πώς θα τα μοιράσουμε. Να σου πω, εσύ έχεις διατηρήσει κάποια σχέση με αυτόν τον κύριο?
Κάτι μια σχέση την έχω.
Κάτι είχα υποψιαστεί, αλλά δεν το έλεγα. I think we should get started. Okay, it's 9:30. So I want to welcome you in this session, which is called More New Tools. No, it says just New Tools and Development 2. So I'm gonna hand over the microphone to my co-chair, Nicoletta Psatha, to welcome you in the session and also introduce you to the first speaker.
Good morning to everyone. I would also like to welcome you to the session. We are going to start this session with Professor Lefkothea Papadopoulou. She's a professor of pharmacology in the Department of Pharmacology, Department of Pharmacy in the Aristotle University of Thessaloniki.
Good morning. Good morning to everybody. First of all, I'd like to thank the organizing committee, Dr. Yanagi, for their kind invitation to participate in this excellent meeting and present part of our work on mRNA therapeutics and the development by our group of a novel PTD-mediated IVT mRNA delivery platform. We all know that the discovery of mRNA goes back to 1961. And it is the molecule that ferries the information from DNA, from the genes, to cytoplasm in order to proceed in the protein, protein synthesis. And mRNA is chemically similar to DNA, except that it is single-stranded. It carries uracil instead of thymine and has ribosome instead of deoxyribosome. And it was the middle of 1980s that mRNA was first synthesized in the lab, and a number of efforts have been started in order to use these molecules as therapeutics. However, there are a number of obstacles that have to be overcome. And in the beginning of this century, three main companies have been founded. CureVac, BioNTech in Germany, and Moderna in USA. And during the pandemic, the vaccines that they developed and the speed they were developed put this technology in the frontiers of modern medicine. However, the obstacles that have to be overcome for mRNA-based technology were the instability of these molecules, the sensitivity to RNAs that will exist everywhere, the strong immunogenicity, the inadequate translation, the negative charge, and thus the necessity for efficient delivery system. However, due to the work by pioneers like Katalin Karikó and Drew Weissman, as well as the number of many research scientists all over the world, a number of structural chemical modifications have been founded in order to have been proposed in order to increase the stability and decrease the sensitivity of these molecules. These modifications concern the optimization of coding sequence, the replacement of UTRs with ones from much more stable RNAs, like the RNAs from murine and human globins, the replacement of cap structure with the ARCAs, the anti-reverse cap analogs in order to increase the efficiency of translation, and poly-A tail of more than 100 adenines. And the main advantages of mRNA technology is that are that there is expression only of the antigen of the interest. There is no requirement for nuclear entry, thus there is no risk for insertion mutagenesis. The process is cell-free. It is much less complex than the production of recombinant proteins. It is flexible and cost-effective, and it is easily scalable, ideally for off-the-shelf and personalized treatment medicine. The basics of this technology are, first of all, the construction of a plasmid as a template for the in vitro transcription, with the sequence having all the necessary modifications in order to be, as I told you, more stable and less immunogenic. Then you proceed in linearize the vector, and you proceed in vitro transcription. The in vitro transcribed mRNA, after that, using an appropriate delivery system, will be intracellular delivered in order to be translated inside the cell in the protein of interest. And the applications are numerous, like protein replacement therapy, CAR immunotherapy, and of course the vaccines, as I already know, we all know, with the vaccines developed for COVID. And concerning the vaccines, it seems that from the 30 vaccines back to Jenner and to Pasteur Hillman with the attenuated and inactivated vaccines, the subunit vaccines and the conjugated vaccines, DNA vaccines, we finally reached the era of mRNA vaccines. And after this explosion, a great number of technologies, biotech companies have been developed in order to, first of all, focusing on the development of prophylactic vaccines, like the vaccines against COVID and now RSV. And however, many of them are working in parallel for the development of prophylactic vaccines, like cancer vaccines, as well as mRNA therapeutics. And in a most recent paper, you can see the areas where mRNA therapeutics can be applied. And as you see, many companies, of course, Moderna, CureVac, and Biotech, but many others, concerning therapeutic areas like infectious diseases, oncology, genetic diseases, metabolic diseases, by development vaccines, antibodies, CAR-T therapy, and protein replacement therapy. So I'm going to tell you about our work in a lab, in lab of pharmacology in Thessaloniki. First of all, we applied this technology in the frame of protein replacement therapy. And concerning the protein replacement therapy, this can be done either by the production of biotechnology recombinant proteins, either by the use of viruses like the adeno-associated viruses or retroviruses, and of course via the modified mRNA. In our group, we first started with the production of biotechnology recombinant proteins. The whole process was quite complex. Thus, we finally decided to focus on modified mRNA. And as our first model, we used a fibroblast from a patient with a mitochondrial disorder due to SCO2 mutations. SCO2 from synthesis of cytochrome oxidase is the fourth complex of the respiratory chain. Here are the 5 complexes of oxidative phosphorylation. And as I told you, SCO2 is a nuclear encoded mitochondrial protein located in the inner mitochondrial membrane, necessary for the correct assembly of cytochrome C oxidase, since it plays a crucial role in the copper pathway. And in order to, copper will be finally provided to COX-2 subunit of cytochrome C oxidase, and thus we can proceed in the electron transfer, and thus the oxygen goes back to water. This gene was identified during my sabbatical in Columbia University in Ericsson Lab, and we identified furthermore 3 mutations as you can see here, in 3 related patients that were suffer from fatal infantile cardioencephalomyopathy and cytochrome C oxidase deficiency. Thus, Andrew Lamiliotou, during her PhD, started in 2016. We had to apply protein replacement therapy via IVT mRNA technology for this as a therapeutic approach of this mitochondrial disorder. I have to say that till now there are no therapies for mitochondrial disorder. There are some therapeutic approaches. Thus, we working on that all these years. And however, for the delivery system, we focused on the PTT technology. As you all know, the most widely used delivery methods for the RNAs are the lipid-based ones, like the LNPs used in the vaccines, lipid nanoparticles. However, there are also other systems, and we focused on PTD technology since we use the technology also for the production of recombinant proteins. And that is the prototype of these protein transduction domains, or cell-penetrating peptides, derived from the TAT protein of HIV. These peptides are either derived from native proteins having this ability to penetrate membranes, or they are synthetic or chimeric peptides. And this 6 amino acid, PFVYLI, is the PTD that we used in this study. And these peptides are less than 30 amino acids in length. There are more than 1,000 now, and they are able to penetrate almost all biological membranes, either via endocytosis or a direct translocation or pore formation, and carry on intracellularly a variety of cargoes, ranging from small molecules like chemotherapeutics to macromolecules like the oligonucleotides and proteins. And the experimental procedure is the one that I already told you. First of all, the construction of the template for the in vitro transcription, then the in vitro transcription, purification of the IVT mRNA, then conjugation with the PTD already said this 6 amino acid PFVYLI via a novel reaction, and then evaluation of the stability and the intracellular delivery. And here is the construct we constructed in our lab in pcDNA3 under T7 promoter for our SCORE2 study, in order to finally have this molecule as IVT mRNA with the coding sequence of SCORE2, of human SCORE2, of full length, by replacement the UTRs with the murine or human beta globin, a replacement of cap structure with the ARCAS, and having a poly-A tail of more than 100 adenines, which was done by using this kit, high scrub kit of from NEB, providing the T7 RNA polymerase, the ARCAS, and the ability for the poly-A tail. Of course, first of all, we have to send the plasmid for sequencing to be sure that it is the sequence that we expected, and to analyze also after linearization for in agarose gel, and then we proceeded in the in vitro transcription. And we also analyzed the produced IVT mRNA by running in 2% agarose gel, or furthermore by cDNA synthesis and PCR to take the corresponding protein product. And of course, after that, we proceeded in the conjugation reaction with this PTD that we selected. As we can see, there are also other groups that use PTDs for the intracellular delivery of IVT mRNAs. However, we used a novel conjugation reaction using puromycin as a linker between the PTD and the IVT mRNA. And to confirm the conjugation reaction, we first applied a band shift assay, as you can see, versus the naked IVT mRNA, the PTD IVT mRNA, as you can see, we observed a retardation in the mobility. And to further confirm this conjugation, we asked from Professor Spiroulias in Patras to perform NMR analysis. And as you can see, there is a differentiation in the magnetic environment of the PTD, the amide protons, when PTD is free in DMSO versus PTD conjugated with the IVT mRNA. You can see a shift in the peaks, you can see double peaks, and also a reduction of the density in the presence of heavy water, meaning that this differentiation in magnetic environment is due to the changes in the chemical environment due to the conjugation. We also evaluated the stability, and we see that in case of the PTD IVT mRNA, the stability was much more statistically significant increased. So we were ready to proceed in the delivery experiment. We used as model, as I told you, the fibroblasts derived from SCO2 COX deficient patient, kindly donated by Professor Eriksson. And we also see the IVT mRNA conjugated with PTD. PTD will give the opportunity for this molecule to be penetrated inside the cells, go to the ribosome, and be translated in the protein of interest, which finally, due to its internal mitochondrial targeting sequence, will be moved and translocated in the mitochondria, in the inner mitochondrial membrane, where will be processed by the mitochondrial processing peptidase, giving the mature protein that will play its crucial role for the correct assembly of the cytochrome C oxidase. And kinetic analysis of uptake and translation of the PTD IVT mRNA in our model in fibroblast, we observed, as you can see, from the first 30 minutes, an increase of almost 7-fold increase in the intracellular levels of SCO2 protein, as we see from flow cytometry analysis, and we also observed the intracellular levels of the IVT by RT-PCR as well by qPCR. In order to figure out which mechanism used by the PTD IVT mRNA to be penetrated inside the cells, we used two inhibitors of endocytosis, chlorpromazine as an inhibitor of clathrin-mediated endocytosis, and genistein as an inhibitor of caveolin-mediated endocytosis. And as you can see, in the presence of chlorpromazine, there was no ability for the PTD IVT mRNA to be inside the cells, and thus we didn't observe an increase in the intracellular levels of SCO2. Thus, using our model delivery platform, clathrin-mediated endocytosis in the uptake mechanism. Then we wanted to see if there is this protein is also functional, and based on the first work I already told you, published in Nature Medicine 1999, we had observed for these 3 patients that COX activity, having mutations SCO2, COX activity was reduced in all these organs and tissues like skeletal mass, heart, and thus we used fibroblasts from a patient from one of our patients, and versus the untreated cells. After transduction of the PTD IVT mRNA, observed by using a histochemical assessment of cytochrome C oxidase activity, dose-dependent increase in the staining, meaning that there is an increase in cytochrome C oxidase activity, thus meaning that our protein was also functional. Androula Miliotou, during her PhD, had also to apply this technology in another model in beta thalassemia attributed to beta globin mutations, as you all know. And again, our construct in PCDNA3 under T7 promoter to take the corresponding IVT mRNA with all the necessary modifications, and we proceeded in the conjugation reaction with the PDD. And with a bad shift assay, observed also the retardation in mobility of the PTD versus the naked IVT mRNA. And concerning the kinetic analysis, we observed again from the first 30 minutes 2 to 4-fold increase in the intracellular levels of beta globin, both in K562 cells as well as in bone marrow from 3 thalassemia patients. This work was published in Molecular Therapy Nucleic Acid, and one of our next group's plan is to assess the efficacy of the PTD IVT mRNA. I hope we will have the opportunity to work with a mouse model of beta thalassemia versus normal mice in order to see whether what will be the delivery and uptake and expression of the IVT mRNA in mouse hemopoietic tissues at the preclinical stage. I hope we will have this opportunity in collaboration with Dr. Yanaki. And as I told you, we first applied this technology in a protein replacement therapy approach, and we also tried to apply this technology in a CAR immunotherapy approach. As you all know, in CAR therapy, we have to reprogram immune cells, T cells, and the last years, natural killer cells, to express a chimeric antigen receptor in order to recognize and kill cancer cells expressing the corresponding target antigen. Here are the CAR sequence for first or second generation with a single chain fragment variable from a monoclonal antibody against the target antigen, the hinge and TARIS membrane domain, and activation co-stimulator domains. And usually what is happening is that they transfect the target cells, T or natural killer cells, by using leti or retroviruses, even electroporations. We wanted to see if we can apply our PTD IVT mRNA technology platform again in this in a CAR technology. Thus, we designed 2 IVT mRNAs for second-generation CARs. As you can see, we have in one of them the CD28 as a co-stimulating domain, or 4-1BB. All the others are the same. This is a single chain fragment variable for RBB in order to finally the transcribed IVT mRNA will be conjugated with this PTD, this 6-amino acid, in order to produce the CAR engineered natural killer 92 cells. We use these cells as a cell line model, and for us it was a model, in order to have these effector cells, in order to finally target HSC3 or MCF7 cells having as an antigen the RBB homodimers or heterodimers. And you can see the naked IVT mRNA and the conjugation, and after the conjugation with PTD, the retardation of the mobility. Then we performed time-dependent intracellular transduction of both CAR, A and B, and we see the intracellular levels of the IVT and the corresponding protein of the CAR in natural killer cells, and thus we decided to transfect cells for 36 hours with our PTD, IVT mRNA of both CARs, and proceeded the co-conjugation, co-incubation treatment with HSC3 or MCF7, and we observed for both CAR A and CAR B, as well as for both HSC3 and MCF7, an increase in the intracellular, statistical increase, significant increase in cytotoxicity. This work was done by Sofia Georgiou with Androula Miliotou and Hara Dedi. And thus we believe that our novel PTD-IVT mRNA delivery approach is a promising platform for developing innovative mRNA therapeutics. And here is our reaction. First of all, we conjugated the PTD with puromycin and further giving this amide bond, and then proceed with the conjugation with IVT mRNA to take the final PTD-IVT mRNA construct. And for this reaction, we asked for a patent, and we granted patent from the Industrial Property Organization of Greece till 2039. We applied also for a European patent, and here is the main group of for this work: Androula Miliotou, Ioannis Vizirianakis, and from Thessaloniki, Associate Professor of Pharmacology and Pharmacogenomics, and Ioannis Papas, Professor of Pharmacology and Toxicology in the Department of Veterinary Sciences. We were all, except Androula, did our PhD under the Professor Asterios Tsiftsoglu guidance, and we are friends also. And I am grateful to them. And with two photos from Thessaloniki with the White Tower and Aristotle University of Thessaloniki. I'd like to thank you very much for your attention.
Thank you very much, Professor Papadopoulou. Is there any question from the audience? Could you please go to the microphone? Thank you.
Thank you for the talk. It's really intriguing. I got a technical question. Why did you use chlorpromazine instead of using dinosaur to check the clathrin-mediated endocytosis? That is one thing. And the second thing is, there are many ways that you can check where your mRNA is targeted or guided, and if it finds the right way.
You mean to find out for the uptake mechanism? Because it is one that used by when they want to figure out if the endocytosis is via clathrin or other.
I know, but all I'm suggesting is that it's an obsolete tool. You can use dinosaur, which also has some side effects, but it's much more specific. And on the other hand, you can easily test by other means if it uses clathrin or macropinocytosis or whatever. The RNA itself can go inside the cell.
To be honest, due to because this PTD, PFDYLI is also already used by others. It's not founded by us, and we knew that it is via. When it's used as PTD via clathrin-mediated endocytosis, but we wanted to figure out that after the conjugation with PTD, it will be the same mechanism take. That's why we first of all focused on these two, and it worked. Thus we stopped here.
Can I also ask a very quick question? So I saw that you proposed to deliver the beta-globin gene in both a thalassaemic mouse model to achieve beta-globin production. Do you expect that this beta-globin expression, is going to be stable, or is there any way to make it stable using your IVT
From
mRNA?
our experience, we used last week IVT mRNA from behind 1 year, and it was stable by keeping it in freeze, -20.
No, I mean in vivo, after delivery of the RNA.
I don't know what would happen in vivo. We expect to have the opportunity to work on that and see if we can do this in vivo and have a result.
Okay, thank you very much.
I don't know. All our work is on tissue culture.
Can I ask one? Thank you. Nice talk, congrats. My question regards this, because we're not familiar with the technology. These peptides, do they have any specific receptor on the cells? How do they bind, or is it pure physicochemical something?
No, there is no specificity. This is maybe one of the disadvantages. They can penetrate almost all
tissues,
biological membranes. In case of in vivo, we have in mind to use in parallel and add a peptide as a homing peptide if we want to focus. However, concerning mitochondrial disorders, all of the cells have the problem, so it would not be a problem. Concerning other diseases, maybe we have to be more specific. First of all, we have to figure out that in vivo will have a result. And as I told you, there are a number of companies that have been founded and tried to go to protein replacement therapy for a number of disorders, but they hope that they will have the product after 5 or 10 years, because none is knowing at the moment.
Now, another question. In the protein expression data you presented from in vivo experiments, in vitro, you had a timeline up to about 4 days. So you actually feed the cultural waste your mRNA, or once you inject? Just once.
Just once.
And it's gonna be.
In case of CAR, we observed the IVT mRNA even till 120 hours, more days, 6 days.
Okay, thanks.
Thank you very much for your nice talk.
Okay.
Yeah. So our next speaker is the well-known, I wouldn't say famous, but he's a well-known scientist in the field, Takis Athanasopoulos, who has a certain mobility program in the European Union area. So at this time, I see that you are Vice President, correct, in gene therapy and vector technology, Regenerative in Germany, and you're gonna talk to us about, go ahead, small molecules and novel vector design to enhance cell and gene therapy applications. Taki, the floor is yours. Go ahead.
Thank you, George. Thank you for the very lovely introduction. And of course, thank you, the organizing scientific committee, to invite us. We have a very good time, I guess, in the venue, in a fantastic restaurant with the speakers. The talk is not affiliated with Regenerative, but I would like to invite you all to check perhaps the Regenerative website, and Stelios in the audience also is a scientific founder and member, I think. Something I did. We're back. And there are 3 interesting technologies from a Regenerative perspective. The novel AAV vectors, a platform for dual AAV mRNA reconstitution, and gene transactivation, I would encourage you to check their website. But as a disclosure, the work presented here is not a property or a claimed IP of Regeneron, and scientific literature confidential work will not be presented. In the field of R&D and cell and gene therapies, there are three modalities. The modality, of course, that we are interested as a part of our network is the cell and gene therapies, but of course, well-established modalities of monoclonal antibody therapies and small molecules are there, and you can see the distribution, preclinical, clinical, and launched products of these technologies, and you can see that small molecule therapy has the greatest proportion. They are hand in hand, though, because some of these modalities, and especially the antisense oligonucleotide therapies, have been established products in the field of cell and gene therapy, like Spinraza, Exondys 51, and Vyondys 53 for Duchenne muscular dystrophy, and we'll talk a little bit about this product. But also in the field of cell and gene therapies, the CAR-Ts, AAV, transgenes, the gene editing are now quite well established. Monoclonal antibodies transformed the pharmaceutical industry, cell and gene therapy will do the same, and we are very optimistic and that this is the case. You can see that year by year there are lots of approvals from FDA, so that's fantastic. And again, you can see here as by a distribution of the top 20 biopharmaceutical companies, the modality by asset. 16 out of 20 of these companies have been introducing the technology of cell and gene therapy, and the great multiplicity of them, the small and molecule therapy. And in the field of cancer and oncology, cell therapy is expected to become the third largest modality. This is almost there actually by 2030. And you can see antibodies, small molecules, and perhaps cell therapy is giving a pivot to catch up. And of course, gene therapy are independent, is a different class, let's say, on their own. And you can see the pipeline of products, field approved, and the vector systems. The purpose of this talk is to merge the two fields. And small molecules and gene therapy are, of course, distinct approaches used in the field of medicine, but they can be also of complementary nature. So we will be exploring in the next couple of minutes the synergies between these two modalities. Small molecules, as a definition, they refer to low molecular weight organic compounds, can interact with specific targets within the body, can be extensively used in drug discovery processes. They have certain advantages. There's a wide range of available compounds with diversity of chemical structures. They have been established in the drug discovery. They can be orally biodistributed. But also some limitations. Targets can be challenging. They can be of limited selectivity. And of course, difficulty in reaching intracellular targets. As we know from the field of cell and gene therapy, there are lots of challenges on their own. So there is a need for high safety and potency vectors, the tissue tropism, the regulatory landscape, the pricing, pre-existing immunity barriers. Animal models of disease can be appropriate or not, and especially as we work, let's say, with smaller animal models. Manufacturing and supply is a bottleneck and a limitation. There are lots of demand in the preclinical and clinical phase, and the manufacturing actually is a limit. So how can we bridge the two technologies, and what are the interactions and complementarities? Small molecules and gene therapy can be used together to enhance the treatment outcomes, can play a vast role in gene therapy by augmenting the effectiveness of the delivered gene. And small molecules can be utilized to modulate different cellular pathways, biological processes that are complementary to the therapeutic effects of gene therapy. And this is we used some ChatGPT is the new modern, I think, way of identifying information. So I asked in the ChatGPT to introduce me the correlation between small molecules and cell therapies, and you can see in the first box, and small molecules and gene therapies, and you can see, I think, a huge interaction and modality. So you can have, let's say, cytokine modulators, co-stimulator molecules, senescence modulators, hominin receptors, apoptosis regulators in the field of cell therapies. And also highlighted in blue in the field of gene therapy, the RNA-based modalities, RNA splicing modifiers, stability enhancers, interference enhancers. So the field is quite rich, is quite prominent, and has a great potential. And if you thought that you haven't worked, let's say, with a small molecule as part of your cell and gene therapy, perhaps you are wrong, because all your polyethylenamine transfection reagents to start with, let's say with the plasmids, are a small molecule interaction. So we published a review article with 250 papers as part of GSK. These were offering a review in the field. We had an invitation from Royal Society of Chemistry to give, let's say, some intake, how we can use the libraries of small molecules in GSK, and how we can see, let's say, some interactions with cell and gene therapy, because back in time, I think 8 years, we had a cell and gene therapy division as a part of GSK. So compared with biopharmaceuticals, small molecules have the advantages of rapid kinetics, oral bioavailability, enhanced penetration, and we will be checking some of these properties. And here are the structures. There are lots of diverse chemical structures, and you can see, for example, a protein kinase inhibitor with a cell and gene therapy application, the ex vivo inhibition of cryopreservation. You can see the bortezomib, that's a proteasome inhibitor, and we used extensively in the field of AAV as an ex vivo AAV vector transduction enhancer. JK1, a BET inhibitor that was very useful in ex vivo inhibition of T-cell differentiation, and we used also as a part of GSK studies. ACT inhibitors, M1s, so a diverse range of small molecules that can be utilized in cell and gene therapy applications. And this is quite extensive, so just some more paradigms with tetracycline and tetracycline modulation. And I will refer to one example of how you can use doxycycline and tetracycline receptor-mediated selection for manufacturing purposes. But you can see the list is huge, lots of chemical structures. In the field of direct application of oligotherapeutics, there are already a list on FDA-approved oligos. And you can see here some oligos from Novartis for eye indications. You can see the Ionis, the G-enzyme, the JAZ. For us, we had, as part of George Dixon's laboratory, that it was at Royal Holloway, and Michalis is also here from Royal Holloway, graduate at the audience. He was an expert in Duchenne muscular dystrophy, and we used collaboration with Sarepta for both exonic indications 51 and 53 of Duchenne in skeletal muscle. The first one for exon 51 was a 30-mer PMO steric block antisense that we will talk a little bit about exon skipping and exon skipping approaches. And the second one for DMD, exon 53, was a 25-mer PMO. Antisense oligos are designed to induce the exon skipping approach in order to restore the reading frame and therefore dystrophin expression. Dystrophin is one of the largest actually proteins. It has 11 kb ORF, 79 exons, and lots of things can go wrong. So you have lots of mutations, and you can try either conventionally with supplementation therapy, and we used in the field of adeno-associated virus direct supplementation of microdystrophins for years, but another a direct advancement in the field was in the field of antisense oligos, and George Dixon actually lab was one of the pioneers of the antisense therapy approach for Duchenne. Targeting now, you can do at different levels, at DNA, RNA, or protein level, and you can check, let's say, the different presence of regulatory elements, the GC content and stability, and this is known. And of course, you can do it in an ex vivo or in vivo situation. You can play with small molecules either in modified autologous setups, ex vivo, in allogeneic and universal autologous cells, but also direct in vivo administration. And I will show you some examples. One of the critical, I think, key interactions with targeting RNA structures with small molecules, and there are quite lots of evidence in the literature. And this relates to the ability to design small molecules capable of interacting and modulating RNA structures. This can open new avenues to target challenging disease pathways, and lots of big pharma, including GSK, AstraZeneca, they are interested actually in this part of the field. And you can see here the different moieties as examples. You can use siRNA or short hairpin RNA to introduce risk-mediated mRNA cleavage and modifications. Antisense oligos, we'll talk a little bit more about the antisense, that they can have an RNase H1 mediated mRNA silencing approach, or through splice repressors, other technology, and of course, last but not least, the microRNA demodulation. So lots of examples of small molecules that are directly applicable in cell and gene therapies, and how to target RNA structures with small molecules. You can see here examples, again, of human RNAs that regulate key biological processes and their functions. What are the types of target and microRNAs? What is the mode of their action? The types of target, and what is the percentage, perhaps, of genome translated in order to analyze this data. And of course, in order to identify these small moieties and small structures, there is a whole cohort of battery of tests and methods to identify chemical matter that binds to RNA. For example, fluorescence-based assay, microRNA, microarray-based screening, DNA-encoded chemical libraries, or methods to design chemical matter that binds to RNA. And Big Pharma is interested in all these technologies. They apply these methods, and they select the most appropriate small molecules for these interactions. And after selection, you can, of course, have some additional stages of the traditional biopharma stages of lead optimization towards candidate selection. And you can utilize this one as a structure-guided lead optimization or as a sequence-based lead optimization, so you can have different variations in the theme. Here you can also see that there are different properties, small molecules that can inhibit microRNA processing, small molecules that can bind different moieties and inhibit the translation at the translation level. You can have splicing modulators that can direct pre-mRNA exclusions, binding of ribose switches, and ribose switches is something that we tried extensively in GSK to modulate cell therapies. Molecular glues that inhibit translations of oncogenic. So I can give you some examples as part of my development in the field, and what are the cell and gene therapy synergies. And this is the building of Royal Holloway. I spent almost 15, 20 years in academic life. It's a beautiful building, and of course, if you visit this nice part of London that is very near to Windsor Castle, and you have seen all these royal weddings. I think you are most welcome to make a visit. It is very beautiful and has a prominent scientific basis. And at Royal Holloway, we developed approaches for different modalities, but the focus of the research group of George Dixon back in the day was on muscular dystrophies. And we published lots of papers that they had synergies of small molecules together, let's say, with a Duchenne gene therapeutic, the abnormal splicing switches of DMD, the microdystrophin enhancements with proteasomal inhibitors, piggyback transposon systems and different systems, and also how to best select adeno-retro viral hybrids. Now, Microberts was one of the pioneers of these models. And just to give you a brief overview of this technology in the lab of George and Linda Popplewell, in a sense, we devised an experiment where animals were injected intravenously with different chemistries of antisense. In this particular case of phosphorodiamidate of morpholino oligomer, PMO, that was conjugated to a cell-penetrating peptide. In the mouse, we're targeting exon 23, and that restores the frame of the mutation. But also, we did a parallel experiment targeting myostatin, exon 2 of myostatin. So we wanted to give these animals not only the restoration of the Duchenne, the DMD frame, but also the strength that comes out of myostatin inhibition. And this was translated later to the studies that we showed and went to the clinic, and they became FDA-approved products. That was the basis. And after, you can have as an example the exon 51 mediated exon skipping and the approach. So you can have, let's say, different exonic combinations. Something can go wrong. You want to restore the frame. You apply an antisense oligo, and you have restoration of the frame, but in a kind of a smaller truncated protein, because one of the exons will be missing out. And here you can have also different chemistries that back in the days we are using, the PNAs, the 2'-methyls, the PMOs. But I think the most recent advancements in the field are coming from the team of Louis Garcia and Aurélie Gouyenval back, I think, 5-10 years ago with the tricyclo chemistries in Duchenne. So all this works, and changing my place of work and also field, I moved to GSK for 6.5 years, and this is how it looks at Stevenage in a nice day. Normally it's rainy, but we conducted research in the T-cell therapies field. And of course, you can have various stages in the T-cell therapies. You can have transfections, viral vector production, the transduction, expansion phenotype regulation, and also the cycle of the T-cell therapies where you take blood from the patient, you lymphodeplete, and you reinfuse. And all this, you can see the different contribution of small molecules in all parts of the process. So you can use, let's say, for example, viral vector enhancers for production. You can use formulation cryopreservation, small molecules. GSK have invented different inhibitors and have a library of small molecules that you can use, let's say, in every part. Or if you want to suppress cytokine release syndrome or versus host disease, they have specific inhibitors that small molecules can add to this effect. But my team, back in the day in GSK, they were more interested in the manufacturing stage. And a brilliant guy that actually now is with AskBio, Konrad Vink, devised one of the pioneering stable cell lines for lentiviral vector production that was based on bacterial artificial chromosomes. And the concept is simple. So all the vector components are going into a bag for lenti production. A single copy of its transcription unit can be cloned into the construct. And then you need to induce these constructs with, because it incorporates some tetracycline sequences. And the induction is utilized by small molecules, like, for example, doxycycline. So you can see the effect of small molecule, let's say, to the cell culture medium. You have also some attractive features, some insulators. We use the HS4 insulator, zeos and selection markers. But to cut the long story short, that was one of the publications that GSK gave a stable cell line for lentiviral vector production. That is very important if you want to do upscaling in the manufacturing process. Similar, we put a patent together with a team for AAV vector production that perhaps can be pioneering in the field. There are one or two, I guess, but that may be transformative if you have an AAV stable cell line that produce the same. But the use of small molecules is there. And of course, the other aspect that we tried while at GSK was ribose switches in order to modulate T cell activation, gene expression or protein activity, and we introduced, let's say, this concept in our T-cell therapies. So I hope that I persuaded you to have a closer look. The field and the synergies between small molecules and cell and gene therapies is exciting, either by enhancing the gene delivery, modulating gene expression, controlling immune responses, enhancing protein stability, function, mitigating off-target effects in the era of CRISPR and gene editing, addressing safety and side effects. And I would like just to conclude with some final slides. And as a summary, we used in the early days caffeine addition in order to upregulate lentivirus. Sodium butyrate could also work. These are the classic, I guess, small molecule contribution to lentiviral production. At GSK, and just before I left, we have identified an undisclosed compound that could work even better. We could work with tetracycline repressors, with doxycycline. PEI is demonstrated for years as an efficient DNA delivery vehicle. Now we have specific small molecules for AAV, also vector production. We tried the proteasomal inhibitor MG132 directly in animals of dystrophin. This effectively rescues the expression levels and plasma membrane localization of dystrophin, but also acts as a further additive because it increases the AAV transduction efficacy in muscle. So 2 birds with 1 stone. And of course, I hope that I made a summary of antisense nucleotides, but we also have used microRNA modulators for safety. Luigi Naldini is pioneering work, of course, in the field. Short herping RNA approaches for different parts of the molecule. And I would like to leave you with this slide with novel vector design and, of course, a small molecule contribution. You can have the different generation of cars we touched here, or TCR, engineered products. You can have different modalities, but the complexity of the structures, sometimes they give rise to remnants, they give rise to non-specific byproducts. And I would like to I think focus your attention in the last cohort of the full-length DMD. Back in the days, we wanted to reconstitute the full 79 exon DMD. So the idea back then, in order to do it for all muscles, was to utilize a triple transplicing AAV vector system. And we tried. It worked in animals in around 1% to 2% levels, but also we observed lots of byproducts because of non-specific cassette binding. And these byproducts could actually be eliminated by clever genetic engineering, introduction of either antisense, microRNA modulators. So the presence of small molecules can effectively add to the safety of the product. I think that's it from me. Conclusions and perspectives with the small molecules. Advancements in the synthetic chemical control systems have provided possibilities for controlling cell and gene therapies. The use of small molecules for CGT manufacturing, I think, is another area that we can focus our attention. But also from a safety, efficacy, potency perspective, I think small molecules are wide contributors to the cell and gene therapy. And I would like to thank you for your attention, and hope that you are intrigued by the synergies. Thank you.
I think that was a great talk. All your personal story through in this field, and the fact that, from what I get, this is the story of how you get better fine-tuning of gene therapy using these little molecules to improve, like superfoods. You know, you have a good build-up, and then you have the superfoods, and you do a triathlon. Okay.
Exactly. So
any questions for this excellent speaker and very good friend? Go ahead.
Thank you.
Thank you, Takis. This is wonderful. I'm getting wild ideas. Well, probably you showed this in your last slide. Maybe I missed it. You just zoomed in too fast. Are there any cases of small molecules that can modulate, like you recruit them on DNA and they can modulate like transcription factors and stuff? Are there cases? What did you show in your last slide?
Let's go back if we can.
It's okay. Like, are you aware of any cases where you showed like small molecules that they can like modulate splicing, whatever. So are you aware of any cases that can modulate like transcription factors? And, like,
yes,
how they
so
bind?
it is known. Normally, you can modulate splicing by different small molecule interactions. Antisense can be used directly, let's say, for the splicing. But also a blockade small molecules, let's say, that can interact with the junction of the molecule. So there are examples actually that you can utilize small molecules to either to enhance or to down mitigate splicing effects.
Wonderful, thank you.
Thank you, Grigoris.
Yeah, one question: this exon skipping story in DMD, has it come out as a product now? Is it there is
yes, George. So there are two products. Now, from a patient's perspective, of course, you get some partial improvement. So they got two products, let's say, FDA approved for exon 51 and 53. The difficulty is that they are very specific for a subpopulation of patients. There are hotspots of mutations, and so they targeted these hotspot mutations in order to increase the percentage of patients. But on the other hand, if you want a universal therapy, I think the way forward is AAV microdystrophin. That apparently, I am aware that in 2023, there will be at least two in parallel FDA approved products in the field of adeno-associated virus microdystrophin that can be good for the patients, hopefully without the side toxic effects of SMA trials. So, because we need vast amounts for DMD. Thank you, George.
We're moving to the last talk for this session. Dr. Georgolopoulos is a senior bioinformatics project manager with Geneva Technologies. He has worked on gene regulation and erythropoiesis in the Altius Institute for Biomedical Sciences and has also taken part in the ENCODE project. And today he will talk to us about genomic applications in gene therapies.
Wonderful, thank you. Well, thank you, everyone. I would like to thank the committee for giving me the opportunity to give you a little vignette on how the field can benefit really from advances in the field of gene regulation. After having a wonderful 2 days packed with fantastic results, I'm not going to show any results. I'm not going to bore you with that. I will just, hopefully, I can convince you how you can take advantage of these, of gene regulation, of our knowledge in gene regulation, to really propel the field of gene therapies. So my expertise in gene regulation bioinformatics, and I've only been working on the periphery of gene therapy, but hopefully we can bring these to the center. So we've talked a lot about gene therapy vectors. So what is how does, whoops, it's not projecting. Oh, there you go. All right, so what are the features of a good gene therapy vector? So there are a few key points, just to mention a few here. One is efficient transgene expression, high yield, safety, and of course cost effectiveness. And let's break this down a bit and see what it really means. So typically when we talk about efficient transgene expression, we're talking about high transgene levels. Safety means that we don't want anything from the vector to leak out into the neighboring genes, or nothing from outside to leak into the vector. Of course, a high yield, the vector of the size is very important here, and of course this has an effect on cost effectiveness. So what does a typical gene therapy vector look like? Well, of course, you have your transgene, which is typically driven by a strong universal promoter. And when we're talking about universal, we mean a constitutive promoter that is active across the genome, across the tissues. And when we're trying to correct a genetic therapy, and when we're trying to correct gene regulation, we first have to know that gene regulation is very precise and highly sophisticated. And when we go with one of these vectors in there to correct gene regulation, it's like we're going with a sledgehammer to fix a watch. Well, it might do the trick, but most likely will break the watch. So how can we improve that? How can we design precision instrument. So this is an example here where you swap out the strong universal promoter by a minimal lineage-specific promoter that ensures that the transgene will only be active in the tissue of interest. Of course, this will be amplified by a lineage-specific enhancer that works only on the specific lineage and the specific cell type, and you can wrap these within insulators, for example, to ensure that nothing leaks out of the transgene or comes into it. Where do we find these elements? Well, a lot of this work has been done, so I'm going to give you a little overview of these methods. So first, you need to map these elements to find where they are. But first, let's talk a little bit about the principles of gene regulation. So gene regulation is a complex switchboard where different combination of switches result in different gene expression programs, that is, phenotypes. You flick a switch wrong, and then you get a disease phenotype. So in its basic form, a gene regulatory circuit, you have the protein which is encoded by a gene, driven by its gene promoter, and this is augmented or modulated by distant cis regulatory element like an enhancer, and of course these two elements are actuated by a transcriptional factor, a TF. You can imagine this as a promoter as being the on-off switch and the distal cis regulatory element as being the volume knob where you turn on and down the volume, and the TF is actually the hand that regulates these two switches. So in terms of gene regulation, we have two major components. We have the cis regulators, which includes enhancers, insulators, promoters, and then we have the trans regulators, which are the elements that acting from a distance like transcription factors. So gene regulation, one major component is a chromatin. So chromatin is not only a structural element, it's a highly dynamic organ that regulates gene expression. And chromatin exists in two primary states. Heterochromatin, where the nucleosomes are packed very tightly together, and then they protect the genes from being transcribed. And then you have the euchromatin, which is nucleosomes are expanded, then they expose the DNA to the transcriptional machinery, and this is mediated by transcription factors going in there, displacing nucleosomes and exposing genes. Of course, this has sort of a side effect. So when the DNA is exposed, this makes it vulnerable to certain enzymes like nucleases or transposases, and we leverage this principle essentially to map the accessible chromatin. So these enzymes will go in there, will start chopping and generating these little shards of DNA. We pile them up on the reference genome, and we have these so-called peaks of accessibility, which is small regions of 200, 300 base pairs. And with this way, we can map all the active cis regulatory elements of various functions. One of these, two of these actually methods to do that is ATAC-seq, pretty popular, and DNase-1-seq. And when you apply these methods genome-wide and across tissues, you get these reference map of accessibility, and you can find peaks that are either constitutive or highly cell-type specific elements. Going back to chromatin, we mentioned about these two primary chromatin states, the heterochromatin and the euchromatin, and we can identify these states as nucleosomes. The histones actually have these little tags, these little flags, and by looking at them, we can tell it to which state the chromatin is actually in at that moment. And pardon me. And a few of these methods are ChIP-seq, cut and run, and cut and toggle. You might have heard any of them. And when we actually will look for multiple of these flags, we can go with very fine grain resolution and find different states of chromatin, like promoters, weak promoters, strong promoters, and same goes for enhancer, genic enhancer, active, weak, and so forth. And we have these unique fingerprints of chromatin states that we can look at. The final element of a gene regulatory circuit is are the insulators. So the insulators are a complex of protein and DNAs that not only facilitate the structure of chromatin, but also mediate gene regulation. Wonderful. Okay. So there are two main functions of insulators. One is the chromatin barrier function, where insulators can protect your genes to be silenced by the surrounding heterochromatin, so they really protect the spread of repressive chromatin. And that, in the context of gene therapy, insulators can protect your transgene where it goes into the genome to protect them from the silencing from the exogenous chromatin. Right. The other function is the enhancer blocker function. So between any enhancer and a promoter, you can interject an insulator and block that enhancer from acting on this promoter. And again, in the context of gene therapy, you can prevent your enhancer in the transgene on acting on nearby genes and preventing like malignancies and tumorigenesis. So one way to find to figure out where insulators are, like I said, they are protein DNA compounds, we'll say, and they are really driven. One of the main TFs is the CTCF factors, as a zinc finger factor, which recognize this sequence. And of course, you can go with any of the typical methods like ChIP-seq, cut and run, or even with DNAs and figure out in the entire genome where CTCF binds and figure out where are the insulators. And having looked at where these elements are, I'm going to give you two examples, two cases from, both of them are from the field of hemoglobins. And this is really because the hemoglobinopathies really rely on the study of the regulation of globins, and the regulation, developmental regulation of globin sweetening has been the textbook paradigm for gene regulation since we discovered them. But of course, gene regulation is not only specific to hemoglobins, it's a universal rule, so why not apply it to any problem of gene regulation in a genetic disease? So the first one is part of our work back in Seattle, and I'm going to show you how we can take advantage of gene regulatory atlases to improve gene therapy vectors. So back in my PhD work, we set to figure out what are the cis and trans regulatory elements that they drive erythropoiesis. So we did that by performing dense accessibility profiling during the course of ex vivo human erythroid development. And with that, we were not only able to identify tissue-specific, meaning erythroid-specific, but also very state-specific elements during the erythroid differentiation. And then, when by performing a multi-omics integrative approach, we integrated gene expression to link genes to their upstream cis regulatory elements. And what is cool about it, that these predictions, when we go and validate them with genetic deletion experiments, like the case of CDH1, we were able to validate both of the predicted enhancers. And of course, we can add an additional level of resolution here. So apart from the temporal aspect, we add some depth to that by integrating single-cell transcriptomics, where we also can take into account the population resolution during erythroid development. Now that we have figured out what are the upstream regulation of genes, we need to figure out now who really drives these cis regulatory elements. So we need to find what are the upstream transcription factors that drive the accessibility of these elements. So typically you would do that with a ChIP-seq experiment, and then you will have to pick out a transcription factor and go after it. But here, by employing bioinformatics approaches, we can save time, we can save money, and do this in a sort of like genome-wide fashion and look at multiple transcription factors. So if we take into account the expression profiles of these transcription factors, and then we look at where these bind by employing machine learning approaches, we can figure out of the many motifs in a single accessible site, which transcription factor is actually driving the accessibility of these elements. So you can think of it as a poor man's ChIP-seq, but in a very high throughput fashion. And then when we link all this information together, we were able to construct a very detailed map of regulatory modules that they act on specific states during erythroid differentiation. And of course, when we piece all these together, we get this high-resolution, temporally resolved atlas of erythroid state-specific gene regulation. So how we can take this into, take advantage of these and how we can put this to work. So as Dr. Psatha mentioned yesterday, and I'm not going to steal her thunder, not all of these elements, these are of course cis regulatory elements, but not all of them are enhancers. Enhancers are just a subclass of cis regulatory elements. To test for that, we employed this massively parallel reported assay, where we clone these elements in front of a clinically relevant gene therapy vector to find which of these are actually enhancers. And of course, we found enhancers, but we were also at the same time able to optimize these gene therapy vectors when we use these elements which are actually erythroid restricted. That, of course, it's a plus for safety of the vector. There's a small elements, like I said, about 200 base pairs short each, which of course had a great impact on the yield and the transducibility of these elements, another plus for the vector design. And of course, as these are really enhancers, or bona fide enhancers, we were able to achieve high transgene expression to correction levels. And then with these, we check all the points that I mentioned initially. By redesigning the vector, by swapping out the universal promoter for a minimal beta-globin promoter driven by an erythroid state-specific enhancer, we were able to do a very fine tuning of the expression of these enhancers, and of course by including the dual C1 insulator, we're also able to ensure the safety of the vector. Well, the second case I'm going to talk about is really a tour de force for genomics and how these findings actually made it into the cleaning into a therapeutic product. And this is the example of the BCL11A, the repressor of fetal hemoglobin. It all started back in 2008 by looking into genome-wide association studies where variation in the levels of BCL11A were found to be associated with levels of fetal hemoglobin. 5 years later, people have employed ChIP-seq and DNase-1 profiling to find that in the intron of the BCL11A, there are 3 erythroid-specific cis regulatory elements which were presumed to be erythroid-specific enhancers. These enhancers were actually validated to be enhancers of the BCL11A by performing a dense saturation mutagenesis by these 2 seminal papers, a group in Boston and our group back in Seattle. They found actually which transcription factor actually binds and actuates these cis regulatory element, which is of course the main driver of erythropoiesis GATA1 that binds to these erythroid-specific enhancer and regulates BCL11A. Following a couple of years later, again, 2 groups, by performing ChIP-seq and cut-and-run experiments, they were able to finally figure out where BCL11A binds on the fetal globin promoter, and of course now they were able to piece together the functional relationship between BCL11A and fetal hemoglobin. And of course, zooming on to today, actually a couple of years back already, people have taken this information and translated to clinical products where can either delete the erythroid-specific enhancer of BCL11A, the repressor of fetal hemoglobin, or actually delete the binding site in the promoter of BCL11A, similar to the work that Dr. Michio showed you yesterday, or take both of these 2 approaches, like the work spearheaded by Dr. Psatha, to delete these 2 elements to reactivate fetal hemoglobin. And this is actually, we're talking about applications that took 10 years from, more than 10 years from the genomics findings to translate it into a clinical product. So we don't have to wait for another 10 years for a new application to emerge. We live now in the in the age of big data. All the experiments have been done, the mappings have been done, the annotation is complete. We're just, as a field, we're sitting right now on top of a gold mine, and we're just waiting for somebody to go down there to start digging. It is a dirty job, but this is really where bioinformatics comes into play. So there is so much untapped potential by the genomics field to be leveraged by gene therapies and therapeutic approaches. Speaking of enhancers, there are a lot of databases out there, but of course the most comprehensive catalog of regulatory elements has to be the ENCODE project, with over 500,000 data sets. There are now annotations for candidate cis regulatory elements that are over 7,000 across hundreds of tissues, normal, disease, primary cells, tissue cell lines, human, mouse, you name it. So the information is there, the mapping is complete, the annotation is there. We just need to take advantage of it. Speaking of accessible chromatin, we pretty much have mapped every accessible position on the human genome. We have now cataloged over 3.5 million accessible elements on the human genome, and we have decomposed them into components, cell type specific components. So now we know which accessible element is predicted to be active in which cell type. So we have also the cell type annotation. We know these are cis regulatory elements. And as I'm going into the epilogue of my talk, I'm just going to show you an example of how these tools are also made to be user-friendly and accessible. So this is also a development by our lab back in Seattle. This is the Epilogos, which is a browser. It is a genome browser. You can scroll on the human genome and look into, sort of like in a base per resolution, what chromatin states are predicted, and across multiple various cell types. So you can find promoters, you can find enhancers, you can find anything. The information is there. Information is massive. It's not always easily accessible, just as I show you with the Epilogos example, but this is where bioinformatics comes into place. We can do the mining. We can do, first of all, the collection. We can do the mining, we can do the massaging, and of course extract biological information. We can do target prioritization for genome editing, for epigenome editing. We can do even the functional predictions. We don't need to go back to the lab to do the experiments. We have all the information to do the imputations. And of course, we can take all these and kickstart the translation and the development of precision gene therapies. Well, of course, with that, I hope I have convinced you that you can really take advantage of developments in the gene therapy field. And if you're interested in finding out more how you can benefit from that, let's talk during the coffee break. Thank you.
Thank you so much for the very nice talk. Is there any question from the audience? Yeah, of course.
Just a quick question. Congrats for the presentation. Is there a limit in the size of the transgene that can be carried from the vector, in size of kilobases, I mean?
Can you please repeat? I didn't hear that.
Is there a limit in the size of the transgene that is carried by the vector?
Well, for lentiviral vectors, although I'm not an expert on that, like I said, I'm only tangentially on this field. There is a cap on the size of the vectors, and the size one is that you cannot carry a big load as far as I know, and of course it has a detrimental effect on the production of the vector. So for lentiviral vectors, there is a limit. I'm not sure there are people that they are more they're experts on this and they can.
Okay.
believe the cap is around 10 kb or something.
10 kb.
Yeah.
10 kb, okay.
10 kb, yeah.
And the second question, what's your opinion on the imputation or inference for the HLA haplotypes?
Okay, that's a painful story. I'm actually working on that right now with a client. Well, you need a lot of information. It's very hard. First of all, it's hard to do the HLA typing. The alleles are thousands. Imputation based on what data? Like what you be using your input to impute the HLA types?
From NGS data from
NGS data. Well, you don't need to impute if you have the data.
But maybe maybe you have just few loci and not the whole haplotype.
Okay. Okay. It is very tricky. I mean, I know you can do these from RNA-seq data with not a very high precision, though. But I'm afraid that needs the exact sequence. You need to sequence the locus, as far as I know.
So you don't trust you don't really trust the imputation data on this?
I don't have a good sense. I know it is very tricky, even if you have the exact sequence, because they are so similar together. You cannot achieve very high precision. I'm sensing with imputation you can get only to up to a degree of the precision for the allele calls, but depends on the application you want to do. Mm-hmm. We can discuss later on this. Thank you so much.
Okay, great presentation. Thank you for this.
Thank you.
Making a link between gene therapy and chromatin states or epigenetic modifications, that's great and desperately needed. And actually, 1 or 2 weeks ago, the Mark Kay lab from Stanford published something really sensational. They published that the capsid of an AAV vector can define the epigenetic marks on the vector genome.
On the vector genome?
Yes. So they found that capsids of different single amino acid show different marks, histone marks on the actual vector genome. This is actually something that they cannot explain, and I don't know. Of course, it has an impact on the transgene expression out of this vector. Any idea how to explain something like this?
No, I'm really astonished. I mean, I have no clue, and I need to look up this paper. But this is really interesting. The only thing I can comment on that, and again, I cannot answer this, but is the selection of these elements that you can add to amplify the gene can also have an impact on the yield also of the vector. So this is one we can take into account. But of course, one thing is the expression of the vector in the genome, and another thing is also the production of the vector on your production cell line, and there are a lot of factors that can affect that. And obviously, as I was just informed, it is also the capsid of the vector. This is I don't know, maybe it somehow facilitates how it is wrapped around the structural proteins. It's fascinating. I need to look it up. Thank you. Thank you.
Hi, Grigoris. Fantastic talk.
Thank you.
And brilliant results. I wanted to ask you, from your experience on the first part of the talk, where you identify cis elements. If you want to translate this to different tissues, so let's say for us, we are interested about ophthalmic disorders in the eye. How long do you think the process will take through a machine learning approach and plus testing in order to end up, let's say, with suitable regulatory elements and suitable promoter enhancers?
Yeah, wonderful. Well, yeah, I can give you an example. So I'm not entirely sure how well the eye tissues are annotated, but let's say you want to start from scratch. A typical ataxic experiment will only take a day to complete to generate the libraries. Then say you have quick access to sequencing that might take another day or two. Then bioinformatics, it's a matter of of a few days to annotate, to clean your data set and come up with a final annotation. And then, of course, you can do some extrapolation. You can integrate other data to further annotate your elements, because now you have the accessible. You need to also get a sense of whether these are enhanced or what are their function. And then, let's say in under a month, you would have, in a matter of a few weeks, you will have a set of starting elements to use in your application. So it's fairly quick, and of course depends on your previous experience and how good you are with annotating. I think one of the bottlenecks here is like when the bioinformatician and the biologist come together, and the biologist has all the information, so it's the matter of annotation. But it also takes a lot of manual curation, and then to do some predictive machine learning, this is only a matter again of a day. That's it.
Well, congrats, great talk. What I noticed is that there was not a single gel, correct?
A single?
Gel, electrophoresis.
Oh, no gels, no gels. No, we're done with gels. No bends, no pipettes, no.
So do you ever go to the lab or not?
I did.
You did?
I did. I did a lot of bench work, and that's why I had it with bench work and gels, and I converted to bioinformatics. It's so cheaper to make mistakes on the computer. You only waste your time. You don't waste any reagents.
Yeah.
So.
No, I mean, to me it sounds like, it's the end of biology. You map the genome, you go, you find your perfect sites, you find the best enhancers, and then you just have to go to the lab and do the work.
Why it is the end? It's a new chapter for biology.
Well, I guess in the end, in the sense that, I mean, the data is there, right? You just have to mine it, and then test it.
Yeah, but then you do.
That's the new beginning. It's a new era.
It's a new era.
It's a new era. Great.
Yeah. Thank you so much.
Go ahead.
Okay, so thank you everyone for joining us in this session. A big thank to the speakers, and we will take a break of about 20 minutes, and we will be back.
We have to start. Dear colleagues, we have to thank all the organizing committee who gave us the opportunity to present the umbilical cord blood and bone marrow as sources for regenerative and immune cellular therapies. The first speaker is Dr. Stathis Michalopoulos, who is a PhD in tissue engineering, and he is in Biomedical Research Foundation Academy of Athens. His position is equal to associate professor, and he is my best collaborator in our cord blood bank. So, Dr. Michalopoulos, take the podium.
Okay, thank you, Professor Stavropoulos. Good morning to everyone. Thank you very much. Thank you, the organizing committee, for inviting me and be with you this morning. Mainly today, I'll be discussing the importance of the cord blood and the derivatives of the cord blood. So, on the very first few slides, I would like to emphasize also on the importance of a biobank in order to understand what is the infrastructure and what can we gain out of a biobank. So mainly, biobanks, it's one of the best ideas according to Times a few years ago. And just to give you a few information regarding the biobank, these are centers where we collect different kind of materials, and they can contribute to the scientific committee by providing all these different stuff of materials and to scientific research in order to help for the diseases and different research on the medicine, on medical stuff. As you may see on this slide, there's a big profit from the biobanks, and generally the types of biobanks you may find all over the world are biobanks for the clinical cases, biobanks for general population biobanks, and biobanks for storage of genetic material. Just to define a few things, there's two terms, the biorepository and the biobanks, but generally the main term that we're using all these years is the term biobank, which is a type of biorepository that collects, processes, and stores human samples. So you may see, I'm not going to stick on this slide, but mainly you may see that if you want to operate a biobank, you have to follow specific procedures, and of course you have to follow a specific kind of accreditation and also certifications in order the biobank to be functionally. So let's come to our case, to the Hellenic Cord Blood Bank, which is within the BRFA premises, the Biomedical Research Foundation of the Academy of Athens. Professor Stavropoulos, back in 1997, she tried to make this biobank within hospital Genimatas, but obviously that time people they were not ready for such an innovative step as a biobank, so this idea collapsed. So a few years later, she got in touch with Professor Skalkeas, and she managed to get a space within the Biomedical Research Foundation of the Academy of Athens. So in 2003, that was the official establishment of the Hellenic Cord Blood Bank. And then eventually, after 10 years, more or less, we managed to get the FACT accreditation, which is like a universal accreditation. We are the only ones in Greece that we have this accreditation, and one of the 50 biobanks all over the world, and also the EFI accreditation. So with this accreditation, we have to follow specific procedures. So this is mainly our departments. We have seminar rooms in order to educate the families that they're providing the cord blood. We have the reception, the processing lab, the GMP rooms, and the cryostorage facility. So in order a biobank to be functioning properly, you have to follow specific standards, specific rules and regulations. So in our case, we have to have a specific organizational chart, and we have to follow specific SOPs, validations, qualifications, etc., etc. So mainly, just to give you a brief overview of how the whole thing works out, so mainly families, future mothers, they're coming over to our premises. We providing them a seminar regarding the use of the cord blood. So once they are ready, they're bringing us back after the delivery of the baby. They're bringing us back the unit. This is the unit, the cord blood unit. And after they have completed the consents and everything is necessary according to the national rules and regulations and the international rules and regulations, we have to process our unit through GMP facilities. So we do follow specific procedures in order to isolate from a unit which is like over 100 ml. And once we process the unit, we manage to get with a final sample of 20 to 25 ml with CD34, which are hematopoietic progenitor cells. These cells, later, they're going to be cryopreserved and they're going to be stored in liquid nitrogen for at least 20 years. So the families, they donate the cord blood unit in order to be used from somebody that it might be of need for a transplantation, for instance, for hematological malignancies. So this is our premises regarding the cryostorage facility, and these are kind of criteria that we have to follow according to the FACT NetCord rules and regulations. Whatsoever, the thing is that the last few years, the cord blood units, they're not flourishing anymore as they used to do in the old days. So lots of units, they're coming over to our place and they do not fulfill this criteria. So like 5 to 6 years ago, we used to throw them all away. Then, all this time, different researchers all over the world, they were saying that through these units, through these cord blood units, we could isolate different types of cells that they could be used for something else. So for instance, we can isolate NK cells, we can isolate Tregs, and we can make iPS out of these cells. So the future perspectives regarding the cord blood units is eventually this biological stuff, which is of an extreme importance to humanity. It could be used, apart from transplantation for hematopoietic malignancies, for isolating Tregs, MSCs, NK cells, and also creating CAR T cells. And this is another slide showing what would be the future of all these cord blood banks. In our case, we decided back in 2017 to have this material, this cord blood unit, and use it this way in order to be of an advantage to people that they're suffering. So we decided to use it on on patients with a diabetic foot. So according to the wound healing, you may see on the slide, unit healing, a complex cascade of cellular events in order to achieve resurfacing. So our main idea was to help those patients in order to assist them with their wounds. Back in 2016, another scientist, Professor Rebula from Milan, Italy, he got in touch with us and he told us about a project that he was working on cord blood units, and especially with people having diabetes and diabetic foot. So Professor Rebula told us then that we can use these units that they do not fulfill the criteria for being transplanted to patients, and instead of throwing them away, we can isolate the plasma. The plasma, as you may see, has lots with the platelets, of course, the platelet plasma. So the platelets, as you may see, they contain the alpha granules. Within these alpha granules, there are these growth factors, for instance, TGF, PDGF, etc. So if we manage from these units to isolate a significant amount of platelets, and then if those platelets, we can burst them in order the granules to come out of the platelets, we could do some good to patients. So that was the article that Professor Eboula, he did publish years ago, and that was the protocol that he advised us also to follow. So mainly out of a unit, of a cord blood unit, we could separate the platelet-rich plasma, or the PRP, you might have heard it, and use it to diabetic patients. But before we use it, we have to make it as a gel in order to be able to apply it on the wound. So before we move on to applying for a grant, we decided to do some work in the lab and see if that works. So we did take our units. This is the starting unit. And what we did was to separate the plasma. This is the plasma bag. And this is the RBCs that we don't need it. So once we did get the plasma, then we had to add the calcium gluconate or calcium chloride, whichever you got. And what we do create is a plasma gel, something that can be applied, okay, like a cream. So once we were doing this process, also we set our criteria. So we had the criteria that the unit eventually that we will be using, the final platelet unit, it should be less of 0.5 10 to the third per microliter WBCs, and at least to have 800 to 1200 of platelets per microliter. And of course to have microbiology and virology test negative. Also, we did perform some histological analysis to make sure that these things that we do isolate, they do consist of platelets. So we did perform some histological analysis, and then we did move on mass spectrometry in order to analyze the final product and see what kind of different growth factors we do isolate. So this is the panel with the different isolated factors that are within the PRP. So we did publish a paper then, and then with all these factors that we managed to isolate, and you may see some of the results. I don't want to make you bored with all these. And then we decided to apply for a grant. That was back in 2017. So we applied in 2017, and our main collaborator was a Greek hospital here in Greece, Attiko Hospital, where they have a diabetic department, and specifically a diabetic foot department. So according to the doctors, 25% of the patients with diabetes will develop a diabetic ulcer, and 50% of them will become infected and require hospitalization. Of course, in most of these cases, the patients will get amputated eventually. Just to move on something financial data. The cost of managing the diabetic foot in England, for instance, you may see it's around 800 to 950 millions per year. This is the same idea also in all over Europe. Regarding Greece, the number of the amputations is about 3,000 to 3,500, and the total cost without amputations is 230 million. So we've set up the protocol, and we have our admission and exclusion criteria. Of course, somebody had to be over 18 years old with a diabetic ulcer, and they were excluded venous ulcers or bone exposure or presence of fistula. So mainly we collected the cord blood and units that they were not satisfying, they were not fulfilling the criteria of being transplanted. We had to process them through the procedure of isolating the PRP. And once we make it a gel, we can provide it to Attiko Hospital in order to apply it to patients. So if you may see here, this is This is the point where we check the unit, if it does have any clots, and we do weigh the unit. And then the next thing is to connect it with another bag in order to move on with the first centrifugation. So as you may see on the last video over here, we make like a sandwich and we put the unit within the bucket, and we do move it on to, we put it in within the centrifuge. So eventually, after we take the unit out of the bucket, we do see the different levels of the plasma and the Buffy coat and the RBCs. And what we do is we just extract the plasma. So you may see the unit up here. You see the different layers. So with these extractors, we manage to get the layer that we do need for the patients. And then eventually, once we do this, we end up with the final product, which is this one. It is like 6 to 10 ml of PRP. In that bag, to make sure that it does work, we have to add the calcium gluconate. So you may see that the final product is like in a jelly form. So we can apply it to the patient. Just to give you an example, so these are the units that the hospital is receiving. That was more or less our first diabetic foot. By no any circumstances, it doesn't fulfill the criteria, but because they were doing it like a pilot study, they had to find out a patient, and you see how do they apply it on the diabetic foot. So mainly we had two groups. We had the study group and we had the control group. In the case of the control group, they had to clean the ulcer every day, just with normal saline. And in case of our study group, every 3 to 4 days, they had to change the PRP gel. So they had to apply a new gel to the patients. So the end points, of course, was to reduce the ulcer size and to evaluate mainly the ulcer after a few weeks' time. The doctors, what they had to do was to every 2 weeks to take a picture of the ulcer. They were using this application on their mobile phone. So they had to measure the diameter and the size of the ulcer and see if there's any improvement. So eventually, these are cases where the CB gel on day 0, after 2 weeks, 4 weeks, 6, 8 weeks, and 4 months. So you see these kind of ulcers, which are really hard to manage to heal them. Within 4 months, that was completely healed. Another example, again here, 0 day, and then after 4 months, completely healed. Then again, that took a little bit more, 6 months. The main idea was if we did see that the patient was replying to the procedure, to move on, to follow on with the PRP gel. Because at the beginning, the protocol was for 4 weeks. But if we did see after 4 weeks that the patients did well, so we did move on with the treatment. That was the control group, just cleaning the ulcer on a daily basis and changing the gauzes. You see after 6 months, the same case, here again. So that was, if I may allow to say, that was a brilliant kind of a clinical study, which led us to 2 posters on an international conferences and also 4 published articles. And just to sum up, the next steps, the next few steps, according also to the guidance of Professor Abula, is this. So, as you may see, cord blood unit, we managed to take out the platelet-rich plasma, which we did use for the diabetic patients. Apart from the platelet-rich plasma, we can have also the so-called PPP. It's the platelet-poor plasma. The platelet-poor plasma can be used for eye drops. So we can use the PPP, the platelet-poor plasma, for eye drops. Also, the RBCs, which now we just ditch them, we just throw them away. They can be used for the transfusion in neonates. Unfortunately, we did got in touch with some of doctors here in Greece, and what we did get back as a response is, let's see how the rest of the people will do in the whole world, and then we can see if we can do it ourselves. So it's something that we cannot do it here in Greece for the time being. So we're going to continue on the PRP, and our next kind of target will be the PPP, where we might do some clinical case studies on people suffering from eye diseases. And that's it. Thank you very much.
Thank you very much, Dr. Michalopoulos. Some questions from the audience, please? Yeah.
Thank you very much. It was a really nice talk. Actually, I have a couple of questions. The first one, can you freeze the PRP and then thaw it in order to administer it?
Yeah, this is something that I didn't mention, mainly in order to release the factors from the alpha granules. You have to freeze thaw the PRP at minus 80 Celsius degrees. So you have to do like 3 cycles.
I saw that you had it written in the slide. So
you freeze thaw it, and then you manage to release the TGF factors. And in that case, you don't need the calcium in order to activate the platelets, firstly, and secondly, you don't have to wait for 9 days in order the platelets to go on the endothelial and then transform from normal platelets, to activated platelets in order to secrete all these granules. So yes, the idea is, like, let's say that we do have an orange, we do provide you the vitamin C straight away. You don't have to drink it. Just, there you go, you have it. So you freeze throw the PRP, and then we did some ELISA testing, and we did see the factors, the TGF factors. Saying that, I have to mention that, of course, the peripheral blood has more growth factors.
That was my second question.
Yeah.
Can you, because what you proposed is an allogenic and off-the-shelf product coming from cord blood to a patient. So did you compare whether the autologous peripheral blood, PRP...
The autologous is by far better.
By far better.
But when you come to patients that are like over 65, 70 years old, you cannot take them to blood every once in a while.
Okay.
So you can have this cord blood PRP, which you can have it, at the bank and use it because it seems like it's working.
But is it the autologous better, or is it the peripheral blood versus the cord blood better? Because you can, if the peripheral blood is better than cord blood, you just can take from healthy donors, make a PRP bank?
They do have the same growth factors, exactly.
Okay.
Exactly, both peripheral blood and cord blood. It's the proportion that it differentiates. So in the peripheral blood, so the autologous blood, the peripheral blood, you do have more kind of growth factors than in the cord blood. But you still have the same growth factors. But the point is, disadvantage is, if you want to use as many gels as I just showed you right now, you will have to go to the patient every once in a while and just take blood from him, which is not something that...
Thank you.
Okay, you're very welcome.
Thank you, Stathi. Thank you very much for the very informative talk and the nice data. I just want your comment, basically. It's not a question. Would you like to, as far as I know, PRP or PP are not ATMPs, right?
Definitely not.
Okay. So,
if I may say, according to Emma, because platelets, they are nuclear cells. They do not belong, they're not classified as advanced therapeutic medicinal products.
So, would you like to tell us the regulatory framework of using these products and the minimum, let's say, requirements of infrastructure that are needed in order to prepare such samples? Because I think that it's quite important for everybody to know that. I'm sure that the bank fulfills all the requirement criteria, but it's very important to know where these samples can be prepared in order to be used in the clinic.
Right. The thing is that you can hear all over the place, especially in Greece, that they're using PRP.
Yes,
PRP.
I know.
And also they're confusing the PRP with stem cells.
Exactly.
Which is completely
so this is a
misleading.
huge issue.
Completely misleading. And especially you may see on advertisements, some people coming out and they say for the baldness, we can use PRP along with stem cells. That's nonsense. So regarding the PRP, they're not like complete rules and regulations. What you have to do is to have a doctor to take the blood and then to have an approved centrifuge in order to do the, and of course, a laboratory place to perform the isolation. You may see also in the market that there are lots of plastic kits that they are using in order to isolate the PRP. To be honest, I was intrigued of testing some of them, and what I see is that they charging lots of money to the patients, and what do they provide eventually is not platelets, it's just platelet-poor plasma, just the yellowish stuff. So regarding the regulation and the framework, there's none. The only place, the only, let's say, organization that they've set up some standards is the Organization for the IVF, because they're using PRP also for the ovaries, for the premenopausal women, which does work. But if you want to use the PRP, at least you have to have the consent from the patient, at least you have to have the approval from the scientific board of the hospital, and of course to have a decent lab to perform this procedure.
Okay, thank you very much.
You're very welcome. Hello.
Hi. I would like to congratulate you on the nice work, and mostly on keeping in compliance with regulatory data that actually regulatory requirements that actually they are not established as they would. As you said, we all know that there are dozens of hospitals in Greece that they make use of PRP, which is produced under unspecified conditions.
And they do not, sorry to interrupt you, and they do not even bother to take the sample and go to the hematology analyzer and see if they are giving platelets or not.
This is terrible. The surgeons do not know what they deliver to the patient. But and also this costs to the hospital lots of money, paying, I mean, the companies that producing these services. So, okay, congratulate you again for doing all this work under specific conditions, actually GMP, although this is not...
You don't need GMP facilities.
Yeah,
you just need a hood and a good hand to isolate the specific layers, and then measure what have you isolated.
Okay, it's still, I mean, you are producing under specific conditions that are regulated, validated, or and you delivers a product that has been specified. My question is, what is the release criterion for PRP as regards the platelet count, and how did?
I just removed it. It's 800 to 1200 times 10 to the power of 3 per microliters. So that's the criteria we've set, and this is the criteria that also Professor Rebula followed with us. So it's 800 to 1200 times 10 to the power of 3 per microliter. So let's say 1 ml should consist of 1 billion of platelets.
Yeah. And how do you separate the?
We have just one question more, which is Professor Vassilopoulos. So you can discuss.
Yes, please.
You can discuss later on. Yeah.
Thank you very much for your interesting talk and your useful work. I would like also to ask you about the volume of the UCBs that you use, because it was written there that they are less than 90, I think, kilos, and you are using
90 grams.
90 grams. So you use UCBs that they are not suitable for processing.
Transplantation.
For transplantation. Thank you very much.
Okay.
Just a comment that I think as a society we have to take the initiative and try to regulate this issue, because they don't do the PRP in the hospitals. They do it in some apartments with some cosmetic reasons. With everybody's doing that. It's not just for the hospital use. In the
of course, they do it. They do perform it in the orthopedicians within their.
It is a product. It's a biological product. I think as a society we should, the next council should do something and try to talk to the OFF and so on to regulate this issue. I think it's going everywhere, so
of course,
it should be regulated to some extent.
Thank you.
So, thank you very much.
Thank you. Thank you.
Dr. Michalopoulos. The second speaker does not need any introduction, because everybody knows Professor Papadakis, who is the star of the hematology unit in University of Crete. Please, Professor Papadakis, she's going to give a talk on induced pluripotent stem cells from homozygous umbilical cord blood units for regenerative medicine purposes.
Thank you very much. Good afternoon, and thank you very much for the introduction. I would like to thank the organizing committee for inviting me, and also I would like to thank Lila Yanaki and the board of the society for accepting the initiative of a working group from the participants of the public umbilical cord blood banks of Greece. So today I'm not going to present data as studies presented, but I'm going to give a new perspective, at least for the European level, and make a journey on the use of the potential preparation of induced pluripotent stem cells from umbilical cord blood. But first I would like to give you the rationale behind this procedure. We all know that the umbilical cord blood is an alternative source for hematopoietic stem cells for transplantation for hematologic metabolic diseases and immune deficiencies. We also know that there are advantages. However, there is one big disadvantage: the low volume of these units, and this is a great disadvantage for the transplantation. And generally, the umbilical cord blood banks are considered as an ideal model for the coverage of population that called ethnic minorities, which means with peculiarities in HLA. And you can see the recent graph here, where we are in WMDA regarding the cord blood. We have more than 800,000 units, and together with more than 40 million bone marrow donors, we try to cover the needs for transplantation worldwide. However, there are some obstacles, big obstacles in the use of umbilical cord blood. First of all, the high disposal rates of the collected blood units, umbilical cord blood units, that it is over, actually it is more than 19%. And this is due to the high quality standards that are required in order to proceed to the processing and storage of the umbilical cord blood, and also the wide implementation of the HLA haplo-identical transplantations in the routine. And these two obstacles have limited the use of umbilical cord blood and have generated great problems, not only psychological problems for us that we have the banks, but also problems in the economic sustainability of the umbilical cord blood banks, and this is a major problem. But on the other hand, this problem has generated an increasing interest in exploring other use of umbilical cord blood in the field of regenerative medicine that will increase the efficient use of the umbilical cord and also will probably contribute to the sustainability of the umbilical cord blood banks worldwide. This is a very nice graph from our friends from Spain that have only public cord blood, and they have very good plans for the blood banks, cord blood banks. And this is an ideal model where, of course, the first aim will always be to increase the cord blood inventory in a cord blood by using, however, cost-effective protocols and high-quality procedures in order to promote the field of transplantation. Whereas, on the other hand, the aim should be to promote the research in the field of regenerative medicine and in the field of specialized transfusion medicine. Studies gave us a very good example of plasma, but also in the field of immunotherapy and regenerative medicine by isolating a number of cellular products. And I'm going to give you some examples. For example, in the field of immunotherapy, we could use T cells from umbilical cord blood, NK cells for generate CAR therapies, T regulatory cells. I was very glad to hear yesterday that we had a new initiative in Greece that by Professor Spiridonidis, who is going to use T regulatory cells in transplantation procedures. Also, we could use the cord blood multi-components for what we call specialized transfusion, as Tati said, for example, to give the blood for preterm babies, and also the plasmas we have already heard, and also other components that could be used in regenerative medicine, such as the cord blood mononuclear cells, the mesenchymal stem cells, the endothelial progenitor cells, and also the IPS. And this will be the focus of my presentation today. I know that you know more, many of you know more about IPS than I do, at least at the technical level. So IPS can be generated from different somatic cell types, and they have the potential to differentiate towards a number of actually all types of cells. And the great advantage is, one of the great advantages is that they are free of ethical concerns compared to embryonic stem cells. We could use IPS derived from healthy donors in order to give them for autologous or allogeneic cell therapies, or we could use patient-derived IPS in order to treat patients by correcting, for example, genetic lesions, errors by novel technologies, or to generate disease-specific IPS just to study specific diseases and develop novel drugs. The cord blood is an ideal source to start the production of IPS. Why? First of all, the cord blood contains large quantity of stem cells. The cord blood cells are young, free of genetic or epigenetic insults, so they are easy to reprogram. We have already a huge number of cord blood units in the cord blood banks worldwide, but also the cord blood banks have the potential to keep all those cord blood units that are not appropriate for the use in the clinic. The processes that are used in a cord blood bank are in compliance with clinically regulated standards because they are going to be used to the patients. The units are accompanied by the family medical history. They are screened for viral infection and sterility, and most important, one of the most important things which has an interest for the IPS generation is that they are already HLA typed in the public cord blood banks. So the therapeutic models to use IPS could be used in other autologous settings. However, sometimes this autologous setting is not convenient, particularly if we have an acute or subacute disease. The preparation of IPS is time-consuming, technically difficult, etc. And we also have the allogeneic model and off-the-shelf approach, which is less time-consuming. However, it has the obstacle that we should use HLA-compatible cells from the donor. And of course, there are approaches to overcome this barrier. First of all, as we do in the bone marrow transplantation, we could use from the beginning HLA-matched IPS. First. Second, you could eliminate the HLA, some of the HLA, some HLA loci by using the appropriate technology. However, this procedure is too complicated and time-consuming, and it's not cost-effective, I assume. And finally, the idea is to generate IPS lines from HLA homozygous donors, the haplolines, and have the respective repositories, the haplobanks. And as we could see in the next part of my presentation, there are already studies in different parts of the world that study the frequency of haplotypes in different populations in order to define how many homozygous donors could be used to generate haplolines and treat the population. And also there are already countries, such as Japan, that they already have their own haplobanks, and they use haplolines in, let's say, routine practice. So the idea is that banked cord blood units are an ideal material for the establishment of haplobanks with HLA homozygous units. And the concept is, as we can see here, that IPS derived from donors who are homozygous for a specific haplotype can be used for any patient that has this one haplotype, as we can see the example here. And there are two approaches. First, to use what we call super donors, which means donors that have common HLAs, common for a specific population. So the IPS that would derive from these donors would be appropriate for a large number of people in the specific population. However, we could have the opposite approach to generate HLA haplotypes from less common HLA donors in order to treat patients that belong to ethnic minorities. And of course, ideally, we could have, of course, haplobanks at the national level. However, ideally, we could finally have a network of haplobanks that could operate similar to WMDA in a mode of global virtual biobank. I'm going to give you some examples from the research in the field. This is the Korean example where the researchers First, they used their cord blood units and they evaluated the HLA allele and haplotype frequencies in their unit. So they generated the actual population, and then they extrapolated the frequencies in the general populations, and then they estimated how many homozygous units they had in their banks, and they evaluated the coverage of the actual population that was represented by the cord blood units and the extrapolated Korean population. And given that Korea is Koreans are a homogeneous population, we can see here that by having approximately 50 homozygous units, they could cover more than 60% of their population. And when they used also the bone marrow donors, together with units, the cord blood units, they calculated, they estimated that with approximately 100 units, they could cover more than 80% of their population. And we have a more impressive result example. I'm sure that all of you know the Japanese example, which is we anticipated that, given that Yamanaka is Japanese. So what they have done, they started with their pool of their platelet donors and the bone marrow donors and the cord blood donors. They isolated the homozygous donors, units or donors. They generated IPS lines, and following the screening and release and multipotentiality testing, they gave these units in a number of centers for clinical trials. And this is their methodology. They use, as I said before, the platelet donors, the cord blood donors, the bone marrow donors. And you can see here the impressive result that by having only 47 homozygous donors that represented 28 HLA types, they could cover 60% of their population. And then they only used 7 donors, and they estimated that they could cover 40% of their population because it is a homogeneous population. And these 7 donors, they were homozygous not only for 3 loci, but for 6 loci. And here are some impressive results because they have already given these units, and they have published some results. The percentage, the majority of the patients that were treated were patients with ocular diseases, Parkinson's disease, cardiomyopathies, ovarian cancer. There was also one patient with thrombocytopenia. There are already published results. However, of course, the researchers keep communication with the clinical centers in order to define probable long-term side effects, but so far there aren't any. This is another example, the Australian example. Each bar represent one homozygous haplotype, and you can see that with one homozygous haplotype they can cover more than 10% of the population, and they need 23 to 25 haplotypes to cover approximately 50% of their population. In all these studies there is always let's say a special consideration, which is the consent. We have to keep in mind that the cord blood units that are stored in a cord blood bank have been stored with consent of the parents to use these units for hematopoietic stem cell transplantation. And usually the donors give consent for the use of the not appropriate units for quality control or for specific research. However, if we want to use for other purposes, such as the IPS, a new consent is required, and this is something that somebody has to have in mind among others. And you can see the complicated reconsent form, and there is still an open question with ethical concerns. What about the commercialization of the products, of these products in the future? How to handle with this special procedure? And let's go more close to us in Europe. We have the Spanish example where, as I said before, Spain has only public cord blood banks, and they have made their own effort to generate these haplobanks. And one prerequisite for their research is that at least 2 cord blood units should exist for a specific haplotype, because they should always leave one, at least one, for potential hematopoietic stem cell transplantation. So they have recognized the homozygous umbilical cord blood units that are already in the banks. They have taken the donor reconsent, and then they have isolated the CD34 positive cells, and they have started to generate iPS. Of course, we all understand that these plans are very challenging, and we could, of course, understand that we need a wide network to go towards this direction. And we need a network for various reasons. First of all, to homogenize the procedures. So the forms, the consent forms, the regulatory issues, because a line that is generated in one bag could be accepted in other banks as well, and in other populations. Of course, we need to share experience. Most of us that we have the bank we are not researchers, so we need the contribution of basic researchers. We have to define protocols to drive down the cost. This is of course, this is the challenge here, and so to maximize the patient coverage. And of course, ideally, we use a global list of haplotypes. I'm sure that it is probably the easiest thing to do. So a global list of haplotypes in order to see if there are shared haplotypes between populations, in order not to have redundancy in case that this procedure will go further. At the European level again, and this is very important, we have a new cost action. Actually, it has just started, which is the Haplo-IPS Cost Project, which is concentrated in this field, how to use the cord blood banks, not the bone marrow registries, but the cord blood banks to generate haplolines. It is coordinated by Spain and also from EATRIS. And you can see the main aims to develop the framework for generation of haplolines, compatible with a significant percentage of the European population, to elucidate the strategies, to have access in donor pools, to establish the ethical documents, to exchange knowledge for the optimization of the methodologies, and also to proceed to the training, education, and mobility of researchers. There are already 24 countries, including our country and Cyprus, with the 3 cord blood banks, and Cyprus as well with Karaiskakyon. And you can see that the participants are from different areas. We have clinicians, we have people that own the bank, we have basic researchers, regulators, ethics experts, immunologists, etc. And the working group, I'm not going to go into detail. So we have just started with very basic, of course we have not produced IPS, at least here in Greece from the umbilical cord blood. However, we have just started to evaluate our homozygous cord blood units. And this is actually, Lena Lachoudi presented this initial data very early in the morning. Actually, this is a side project of HFRI project that aims to evaluate the HLA haplotype frequency in Greece. In collaboration also with with bone marrow donor centers, the Karaiskakion, etc. So the first data that are based in the cord blood units and have been evaluated by NGS either in 3 loci. We have 2,500 units that have been NGS typed in 3 loci, and we have approximately 1,000 units that have been typed with 4 loci. And we have found just 5 homozygous units, and 3 of them are identical. So if we want to make them, to keep the criterion of at least 2 cord blood units should be available to start such an initiative, we have made our calculation to use this one haplotype, and we have found that with this one haplotype we could cover approximately 5 to 6% of our population. And this is another result of our study showing that, unfortunately, given the heterogeneity of our population, and this is well known, with 100 of homozygous units, we can cover approximately 50% of the population, and we need more than 500 units to cover 100% of our population. So, in conclusion, today more than 800,000 units are stored in public cord blood banks worldwide. HLA homozygous cord blood units would be an ideal source to generate haplolines, and there are already data, at least from Asian populations. And the use of haplolines based on common HLA in different populations could cover the medical needs of broad percentage of the population, but also the opposite. The rare haplotypes could cover the minorities. Of course, this is an interesting area of research. In Europe, we have just started, and of course, we need collaboration. And before I finish, this is my final slide, just to show you, if we use this ideal model where we are, the 3 cord blood banks in Greece, how we collaborate. First of all, of course, we have to work towards the stem cell transplantation always. However, we have changed our policy. We want to identify the regions of Greece with the HLA peculiarities in order to promote policies and recruit donors and units from this population, and also the cord blood banks to be cord blood banks mostly for these minorities. And also in the transfusion medicine, studies gave a very nice example, and together with Professor Rebula and the 3 cord blood banks, there is already an international exercise at the European level, and here is the publication. And regarding the regenerative medicine, we have just started with the haplolines. And with this, I would like to thank you for your attention.
Thank you very much, Professor Papadakis. Excuse me. In order to create an haplobank, except of cord blood, could we use also a product from platelet and blood donors?
Yes, yes, of course, we could identify the donors, the homozygous donors, because we have the donor centers have their donors HLA typed. So one could identify the homozygous donors and go back to the donors and ask their consent. But this is another project under other regulations.
That's true.
Yeah.
Mm.
Thank you very much, and thank you for giving such a very good overview of the problems of the field. I know in ATRIS we talk about all the time about all these regulatory components, and now in the Netherlands we are trying to create GMP-grade iPSC lines, and this issue of the informed consent and who should be the donor and how we are going to deal with the commercialization, especially because the cost of making one GMP-grade iPSC line is very high, so who is going to cover this cost? And if the public sector cannot cover this cost, if we need so many iPSC lines for Europe because of the heterogeneity, then maybe a private sector has to go there. Then the commercialization issue is very important.
Yes.
So I don't know then if the non-efficient or non-cost-effective, as you said, approach of genetic engineering the lines, maybe, I don't know, what is your opinion? In the end, it is more cost-effective. You understand what I mean?
Yes,
in the bottom line. Yeah.
I assume that this is just a comment. All these concerns and all these ideas, of course, are on the table, and these are very important issues. And this is the reason that we should go all together, and not just one bank, but we should go all together in order to solve these type of issues. And if we go all together, we will.
Yeah, because with engineering, if we say that, okay, we will invest the same money, but in engineering, then you don't need.
Yes, but again, there are, again, if the final product is to be commercialized, again, we have the ethical concerns, etc. So.
Yeah, but for one.
Yes, one, yeah.
Thank you.
Thank you very much for this inspiring talk. I think you have a great vision, and you have put in front of us a very, very challenging project. I don't know, it's more than a project. I heard in the International Society of Stem Cell Research, Yamanaka himself talking about this initiative he had in Japan. And he's not only a great scientist, but he has great compassion for humans and human health. So you're trying to do something similar here. But don't you think, I think you already said it, that it's a sort of initiative that has to be connected on a European level. Would that reduce or would that complement the number of haplotypes to cover, let's say, the European population? And also, we have very good experience with iPSCs, and we would be glad to connect with you and contribute, but we are not in a GMP facility, and this brings in another level of complexity. For scientific and research purposes, this is fine, but when we're talking about patients, this becomes a totally different thing, right?
Yeah, and thank you for this comment. This is the reason that I consider this cost action very important, because it has incorporated scientists from different fields, clinicians that have no idea, from the procedure, however they know the diseases, immunologists, in order to create this approach to the HLA types across Europe. For example, we have, based on this collaboration, we have identified our similarities with Cyprus, which is anticipated. However, we didn't know until recently. Yes, this cost action will give the opportunity to scientists to work together in order to start from this defined material and go finally to the patient. And this is very important, that we have the chance to be all together and solve these issues. towards this very interesting and intriguing field of research.
Yes, sorry, but we run out of time. We have to proceed with our next speaker. Okay, if it's a short one. Okay. So our next speaker is Professor Bernd Giebel from Essen, Germany, and an expert at the extracellular vesicles from mesenchymal stem cells. And the title of his lecture is Clinical Potential of Mesenchymal Stem Cell Extracellular Vesicles and Translational Challenges. Professor Giebel.
Yeah, thank you very much for the introduction, and I'm grateful to be invited to this excellent meeting. So it's my first time in Athens, and yeah, I learned a lot. So the session was on cord blood, and I have also some relations of the cord blood, maybe not with the EVs. Basically, cord blood is a really excellent source for different cell types, and we studied hematopoietic stem cells, CD34 positive cells, and searched for asymmetrically segregating proteins. It was a long journey, but finally we identified markers, and two of them were tetraspondents, and they were exosomal marker proteins, which brought me from the hematopoietic stem cell field into the extracellular vesicle field, which I would like to introduce today. But that was also cord blood. So I think, yeah, this is a picture from 1546. It demonstrates the dream of humans to find the elixir of life, that we can stay forever young. Maybe going to the Acropolis, there might be some even older visions to find this fountain of youth, and that is still our mission in regenerative medicine. And at the turn of the millennium, we introduced stem cells, and we think that maybe stem cells are the elixir of life, which can help us to cure diseases, stay young, at least for longer periods, and maybe age in a more healthy manner. So, but then we should discuss which would be the right stem cells. And in the previous talk, we have learned about the iPSCs and pluripotent stem cells, and they are quite nice and interesting cells. So specifically the iPSCs, since they lack the ethical issues, and they can differentiate into all cell types. But the progress is really challenged by the fact that if there is one pluripotent stem cell left in the transplant, it has a very high teratogenic potential, and that slows down the progression of iPSC-derived derivatives into the clinics. I think there are strategies to overcome it, but this is a challenge we need to keep in mind. Then at the same time, the pluripotent stem cells, the embryonic stem cells, became popular. There were papers in science like turning blood into brain or brain into blood, and we discussed that there is stem cell plasticity, that maybe also a hematopoietic stem cell can create neural stem cells and vice versa. And to that time, it was also described that there are mesenchymal stem cells which had the tree lineage potential, creating osteogenic, adipogenic, and chondrogenic structures, and eventually more. And these cells are plastic adherent, can be expanded quite easily, and they lack the teratogenic potential. And so thinking that they can be used now for cell replacement strategies because they can transdifferentiate, they were administered in many different disease cohorts. So there are also acute diseases like myocardial infarction or ischemic stroke, where you would need to treat the patients quite quickly after the insult. And so therefore you cannot use the autologous cells, and you have to administer off-the-shelf products. And that is what we also discussed in the previous talk, the issue of HLH compatibility. And you are aware that organs are rejected by the immune system if the HLH typing is not appropriate. So people started to investigate the interaction of the MSCs with the immune system. And it was found out that the MSCs can suppress effector immune responses. and instead promote regulatory or tolerogenic immune responses, which to my understanding are essential to allow regenerative processes. So, because one of the diseases with an HLH mismatch is the hematopoietic stem cell transplantation for treatment of leukemia, where you myoablate the patients to kill the leukemic cells, but you also kill the healthy hematopoietic stem cells. And then you need the hematopoietic stem cell graft, which might be obtained from umbilical cord blood, but conventionally are obtained from bone marrow or GCSF-stimulated blood. And this is also an immune therapy. In addition to the hematopoietic stem cells, you transplant T cells, and they are required to kill residual leukemic cells. So if the T cells would be depleted, the relapse rates would be very high. So that means the allogeneic stem cell transplantation is a stem cell replacement, but also immune therapy. And it occurs that in 50% of the cases of the patients who received allogenic transplants, these transplanted T cells not only attack the leukemic cells, but also attack healthy tissue, resulting in the graft versus host disease. And since MSCs were immunosuppressive, and many, so I should say the first treatment, the first instant treatment is steroids, and half of the patients with a GVHD react on steroids, and the other half was not able to be cured at these days or to suppress the symptoms. So, and it was then in 2004, Katharina Leblanc published a first-in-man trial, and she treated a graft versus host disease boy of 9 years old with MSCs, and were able to suppress the symptoms repetitively. So, because of the severity and the relevance of the disease, there was a run of many hematologists using now MSCs, and there was a company named Osiris which manufactured MSCs in higher doses, and they ran a blinded phase 3 clinical trial before 2010. And then it was a period when they uncovered it, it was shown that the MSC administration has no effect. And this is very symptomatic for the whole MSC field. There are many groups who report in preclinical animals or in early studies how potent MSCs are to suppress a huge variety of different diseases. But if it comes into the clinical trials, this is really, sometimes you lose the activity. So why can that be? Why is it so diverse? And if you raise MSCs, and that is what we did from 20 healthy bone marrow donors, we raised MSCs, we saw that they all had the key features, but they, for example, in terms of morphology, these ones are much smaller, these cells are larger, you see difference in adherence. So if you look for the bona fide criteria of osteogenic, adipogenic, and chondrogenic differentiation, you observe sometimes many cells get calcified if you use in osteogenesis. Other times it's less. So we have a panel of markers, bona fide markers, and you see the mean fluorescence intensity. It's varying from MSC propagation to MSC propagation. And then we can raise them from different cell types, including these perinatal derivatives, umbilical cord blood. And so overall, it's very heterogeneous. So you also find this heterogeneity in the mean fluorescence intensity if you take cells from umbilical cord or placental tissue, for example. So furthermore, the cells age, and the older they get, they slow down their population doubling rate, which is shown here, and from propagation to propagation, this is also not in a synchronized manner. And to that end, we may think, why did the trial fail? I think the Osiris just expanded them too far, that they become almost senescent and had lost the activity. That means the way of how you culture them also affects the cells, and that could be improved. And Osiris sold all their IP to a company named Mesoblast in Australia, and they run another phase 3 clinical trial after improving their product, Remestemcel-L, and they run another phase 3 clinical trial in pediatric GVHD patient, and it was successful. And that was published in 2020, and that was also the time the failure of the first trial, which was run before 2010 was officially published. So apparently you can overcome it if you have the right protocols, if you do not overexpand. And so they asked for the market approval, and FDA rejected the market approval of Remestemcel-L. And the argumentation was that the MSCs from one donor are not sufficient to treat many patients, so you have many different donors, and that the characteristics of these cells can vary from person to person, and that the mesoblast did not address the potency of the individual units appropriately and did not deliver a mechanism of action, and therefore they say, okay, even though your phase 3 clinical trial was showing efficacy, your product is very heterogeneous and not very well controlled, and therefore they did not get the market approval. And I think that's a very, it's a take-home message maybe. If you work with primary material, it may always be related to that. So very important is the mechanism of action. How do they act? So how, potency testing is essential. So how do MSCs act? So it was believed cell replacement, transdifferentiation, and I think this is off. So now we know they have not the pluripotent potential. So, and it was shown that the effects are paracrine, because if you administer MSCs, most of them end in the lung and disappear, but still they have a therapeutic effect. And after thinking it could be a paracrine effect, some researchers asked, is the activity recovered in the conditioned media? And they administered conditioned media in the myocardial infarction model, and the activity was comparable. And as it was a paracrine factor, people thought on cytokines and started to fraction the conditioned media. And there are two pioneering groups, one of Giovanni Camusi and the other of Sai-Kang Lim. And they processed the media either by differential centrifugation or by filtration devices. And this group recovered the activity in a fraction which could be precipitated by 100,000 G by ultracentrifugation, whereas they recovered the activity in a fraction larger than 1,000 kilodalton. So that means both of them had the idea that there is more solid materials and not just soluble growth factors. They did electron microscopy and recovered extracellular vesicles, which were named here microvesicles and here exosomes. And so we cannot discriminate what are microvesicles. Microvesicles are blebs of the plasma membrane, where exosomes are derivatives of the endosomal system, that we call them extracellular vesicles. So that means today we assume that the active components of the MSCs are the EVs, and we stepped into the field. We manufactured MSC EVs and compared them together with a group of Dirk Hermann in an ischemic stroke model, and we observed that the way of how we prepared our MSC EVs, they had the same beneficial effect in the ischemic stroke model than the parental MSCs, which were applied in parallel. So, and therefore we can conclude MSCs act via their secretome, specifically via the extracellular vesicles. And so, of course, now we know EVs from the right cells may provide the elixir of life. So, and what did we do until now? So we had also a GVHD patient, which was treatment refractory, and it was in 2011 when the OSIRIS trial was published. So there was no public or no MSCs available, which the doctor would have been applied. And so we discussed whether we could apply our MSC EVs, and we did, and it was a quite successful study. We also investigate other diseases. I believe that their important mechanism of action is that they turn pro-inflammatory processes turn into tolerogenic, and by that convert the immune system from defense to the regenerative state. And so we also studied in a couple of diseases with collaboration partners, so we have now own animal models, and remarkably we could also suppress symptoms in a Niemann's pig type C mouse model, which is an inherited disease. So, and one important question is now the EVs, do they overcome the limitations of MSCs, or are they as heterogeneous as the parental MSCs? And that was when we prepared the first larger preparations for the treatment of the GVHD patient. We already saw that they differed in the cytokine content, the preparations. We didn't go into detail, but we were thinking, how can we approach that? So since we began with the treatment of the GVHD patient, we focused on a mouse model of GVHD. And if you have animal models for GVHD, they are differ. And this is because if you have transplant bone marrow, there are T cells in that. And that may vary from mouse to mouse. And Carsten Kirsning, our collaboration partner, considered, so let's deplete the bone marrow from the T cells from the bone marrow, use that as a stem cell source, and instead use defined numbers of T cells from the spleen to co-transplant and see can we reproduce the GVHD symptoms. And that is indeed very reproducible. And you can see it after irradiation, and so the onset of GVHD symptoms is at day 5. And if we don't treat them, it gets more severe. And if we administer potent EVs, we can suppress and revert the disease. So that was over the years. We had so many larger preparations, and we had the feeling some of them are potent and others are not potent. And indeed, we administered different samples, and some were able to revert the phenotype. You see here the GVHD phenotype, the positioning, the fur, and this is this mouse received EVs which are considered to be active, whereas that mouse obtained EVs which we consider to be inactive. And we see also the impact of GVHD on the gut morphology, so these scripts are completely destroyed in GVHD, but in mice which received potent EVs, they were restored. So that was very reproducible. There is a clinical score, and when we repeated it, it was also reproducible, and that has just been published in Cytotherapy. So to that end, we learned not all MSC-EV samples provide the activity, but we cannot test always for potency testing mice, and so we put a lot of energy in identifying assays to learn whether there is activity. And of course, the most simple assay is the mitogen stimulation of peripheral blood T cells, which start to proliferate and to suppress them. And we had samples of MSC-EVs which were very potent in suppressing these T cell proliferation, but unfortunately that did not correlate with the clinical symptoms. What correlated is the mixed lymphocyte reaction assay which we established. And so we pooled mononuclear cells of 12 healthy donors, creating a very strong mutual allogenic reaction, which is, can be observed by the colony formation. So if you just culture mononuclear cells from one individual, you have no colonies. If you pool them, you have plenty of colonies, and they can be modulated by the administration of MSC-EVs. This is not a quantifiable readout, and since we do a lot on flow cytometry, we searched for markers. And what we observed is that in the allogenic situation, there is a proportion of CD4 as well as CD8 T cells which become activated, which you can recognize by the expression of the IL-2 receptor CD25 or ICAM1 CD45. So, and what then we learned, so I have to say, we raise our MSCs in 10% human platelet lysate supplemented media, so it's related to the platelet-rich plasma, which we can discuss. And so the EVs of the platelet lysate are also in our product. So for us, it's the control always to take the fresh platelet lysate supplemented media, extract the EVs from there, and see whether they have immune modulatory activities, and they have not. But if we take the conditioned media from the culture, we observe that a proportion of the fraction is able to really suppress the T cell activity, but not of all. And remarkably, this activity, those samples who could reduce the T cell activation in the mixed lymphocyte reaction assay were also those who had the in vivo activity. So that we think this is a good assay, a surrogate assay for the potency. It's not a potency assay because potency assay needs to be qualified, and by the usage of, again, always 12 different donors, it's a challenge to qualify it as a potency assay. Now I have put a square around these, and I should say all of these three preparations were manufactured from the same MSC stock. And two of them are active and one is not active. And in total, 80% of the sample of that MSC stock created active EV samples, whereas we have also manufactured from the stock 16.3, many different samples, and there were 80% showed no activity, but 20 showed. That means there is somehow stochastics with the 41.5 has somehow a prerequisite for giving active, and it's not reproducible. Why is that? So when we raise MSCs from bone marrow samples, you very often have more than one colony, so they are clonal. And we can ask, what is an MSC? What is the founder cell? And so if you seed any material, it takes 7 days without happening anything, and then they start to proliferate. And I think there's epigenetic reprogramming, which has a stochastic component. That means that each individual cell is different. And maybe some have the potency like the green ones, and maybe others are antagonistic or have no activity. And if you expand the cells, so in the early passages, it's a mixture, but if you expand them in a stronger manner, you have clonal selection, which has been shown by the integration of lentiviruses barcoding experiments of Axel Scharnbach in Hanover. So, and furthermore, they age. Yeah, and I think, and that is what we wanted to address, whether there's also this intra-donor heterogeneity, and we raised independent oligoclones of the same donor, and indeed saw that some have the T-cell suppressive activity, the EV preparations, and others have reduced fractions. So, and therefore, the conclusion is MSCs are a heterogeneous cell entity, and not all MSCs may mediate therapeutic effects, and therefore independent MSC EV preparations can differ in their therapeutic activity, and so somehow we are in the same state as the mesoblast, which was rejected. And therefore it's a strong conclusion. I do believe that products from primary MSCs or from other primary materials will really get hardly the market readiness. Market readiness means we may become able to treat 100 patients, but not 100,000 or even more. So, and then the question is, how can we reach market readiness? How can we manage it that we have just one MSC clone and that it remains young and restores its activity? And the good thing is we can immortalize. So we decided to immortalize them and prevent senescence. And after we used a third-based lentiviral strategy, we expanded the cells, and at a certain point, we deposited single cells and raised single cell clones to avoid this heterogeneity. And we can expand these cells over 100 passages. Here it's shown for passage 10 and 50. The cells look very similar. They have the same phenotype, and we believe that we can maintain them as homogeneous as possible. There might be subclones arising due to spontaneous mutations. That is what we need to stress out. But more importantly, with the immortalization, do they maintain the capability to secrete potent EVs? That is what Yanis Moulou tested in the mixed lymphocyte reaction assay. The primary ones and of the clonal immortalized MSC, the EVs, both are capable to suppress the CD4 as well as the CD8 content. More important is, are they potent also in vivo? Because I mentioned we can screen and score in vitro activities, but finally they may score an activity which is not relevant for the in vivo model. So we have four different disease models. One is again with Dirk Herrmann, performed by Chen Wang in ischemic stroke model, where we saw that the CIMSC EVs are still able to suppress ischemic stroke symptoms. Together with Josephine Herz, which is now already published, we showed in a hypoxia-induced encephalopathy model of neonatal mice that they can effectively suppress neuroinflammation. So in this study, they were administered intranasally. With Claudia dos Santos, Toronto, we explored the function in an acute sepsis model, and mice have RIDS as well as neuroinflammation, and both symptoms could be suppressed. So, and therefore we believe EVs from CIMSC have conserved their function, and the conclusion is that the CIMSC may overcome limitations connected to heterogeneity. Then the media requirements are very important. Our primary MSCs grow in 10% platelet lysate and hardly in 5%. You may educate them, but platelet lysate is expensive, and even we have pooled batches, the quality changes from one larger batch to the other, and we would like to reduce this as much as possible. And the CIMSCs grow in 2.5% platelet lysate quite well, even in 1%. And that is what we are optimizing. Then we can genetically engineer them. That is interesting for basic research purposes, but we can also now overexpress certain proteins. We engineered them to express PD-ligand 1 or PD-ligand 2. We, in vitro, it didn't increase the activity, but we haven't tested them in vivo. We can label them genetically to express CD63, EGFP, or M-Sherry, that the EVs are fluorescently labeled. And we hope that by now using the right clones and also addressing the ethicals, so we changed the ethics because we need to say we want to use the donor material for a market product, for commercial purposes. So we need to erase novel clones with a new ethical thing. That is what we discussed. And there are many things to do, and I could spend another 20 minutes for each of these points to discuss. So if you are interested, approach me. So we have to optimize the expansion protocol, the EV purification methods, the characterization methods. We have to qualify potency assays. We want to search for surrogates, and we have to follow a lot of regulations, which we are also discussing in a community. So we discussed about the money for the iPSCs. This is the same. If we want to qualify them for the GMP processing, it's extremely expensive. It's hard to get public money, and so we are currently trying to get seed money for a company named Exosla, which is in Cambridge located yet. And yeah, hopefully next year we have the seed, so it's not that easy to get the seed. And last but not least, I like to mention the people. So I have an excellent team of current workers, former workers. We have a lot of funding by different funding agencies, academic collaboration partners, as well as commercial collaboration partners. So thank you very much, and I'm happy to discuss things.
Thank you very much, Professor Gibbel. Excellent talk about MSCs. We have time for one question. Professor Giannaiti.
Very nice work. I would like to ask you if you measure the dose of the extracellular vesicles you apply to your model, or you just apply the whole output of the culture?
I expected that question. Sorry, it's gone. So what we are doing, this is how to normalize. You can measure particles by NTA. We introduce it as one of the two groups to the field, and I would say, no, it measures particles, and we have far more particles in our preparations than vesicles, so that means I would not consider this is a good idea. Same holds true for protein. Many say I apply that and that amount of microgram EVs. It's also not a good idea because most of the protein is contamination, impurities or byproducts. And you cannot compare. And so we decided to work with cell equivalents. And that was the dosing which we did for the patient, where we said, okay, roughly 1 million MSCs are applied for a kilogram body weight. And we said we allow the media to be conditioned 48 hours, factor 2. So we said, okay, we apply the cell equivalent of 500,000 MSCs, the EV yield per kilogram body weight of the patient. And if we do small animal experiments, we have a factor 100 higher. It's then 5,000 cell equivalents per kilogram animal. Yeah, so that is.
Okay. Thank you.
Thank you very much for such a fascinating talk. Just a question: have you checked the differences in the content, in the cargo of the exosomes of the immortalized MSCs versus the heterogeneous population? And secondly, are there any type of selection of MSCs? If I put it in another way, do you select a specific population of MSCs that you then immortalize in order to isolate EVs?
Yeah, this is an excellent question. So first of all, the question was when we had the potent and non-potent EV preparation, so go and look for microRNAs, which many consider as the active components. So it's also controversially discussed. I don't believe that the microRNAs are the critical components. And we didn't find any difference. So the non-active and the active MSC EV samples had exactly the same. So then we did also mass spec, and in the two component analysis, so the preparations of the active ones clustered closer together than, and the non-active closer together than they were not mixed like with the microRNAs. We haven't found any concrete marker there, but indeed we have IP'd or applied for IP with one marker where we have one of the checkpoint inhibitors, positive EV subpopulation, which is more abundant by imaging flow cytometry in the active MSC EV samples, but only if we raise the MSCs under the protocol we are doing. If you raise them in the first 24 hours differently, the marker is obsolete. So that is, it remains a try and error.
Thank you very much.
So thank you very much. This is the end of the session, and thank you all.
Thank you very much.
Hello. We continue with the next session of this morning, which is on regenerative medicine. And our first speaker, I'm very pleased to introduce him to you, is Stavros Taraviras, professor at the University of Patras, very experienced in the stem cell niche. And today he is going to talk to us about reprogramming to ependymal lineage, a proof of principle gene therapy approach for treating hydrocephalus, and I do believe it has something to do with the stem cell niche. So, Stavros.
Thank you very much. I would like to thank the organizing committee for inviting me to speak to such an interesting meeting. I have slightly modified my initial title as I believe that will be more relevant to this audience. So back in 1956, Wantington tried to describe with this drawing how a stem cell differentiates different cell types. So basically, with this picture says that a stem cell mimics this rolling ball going through these different hills and valley to one or to the other direction, differentiated to one specific cell type or to a different cell type. So basically, each of these paths we know now that comprises, integrates different signals, different transcription factor, signaling pathways, and epigenetic changes leading to different functionally specialized cells. So recent findings, we know that from a differentiated cell, we can go back with reprogramming processes to a stem cell state, or more than that, from one differentiated cell type, we can go to another differentiated cell type. So my lab during the last years is focusing on neural stem cells and how neural stem cell decides between the different options that has to become a neuron oligodendrocyte, astrocyte, or ependymal cell. And actually, I'm going to focus on this, let's say, cell type, the ependymal cell. And by mimicking this drawing, I will try to describe you how a stem cell, a rolling ball of a stem cell, ends up to become an ependymal cell. So just to give you a short introduction about this particular cell type, the ependymal cell. So this is a section from the cortex, and in this magnification from this wall, we have this specific type of cells named E, ependymal cells, which has a distinct functional characteristic, which is this multiple motile cilia. So basically, ependymal cells forming a barrier between the brain parenchyma and the ventricular lumen, where the CSF is localized. So these cilia are important for the circulation of the cerebrospinal fluid, but the ependymal cells are important for other regions. First is that they are in contact with this type B1, the adult neural stem cells, and they provide trophic and structural support to the neurogenic niche. And we know how important it is because there is a disease in humans which is called hydrocephalus, and the dysfunction of ependymal cell is a main pathogenetic mechanism for this disease. So humans actually develop this disease either congenitally, either during adulthood, from various regions. And what we know is that there is an accumulation of the cerebrospinal fluid that pushes basically the parenchyma. That leads to the neurodevelopmental defects and also to neurological defects in older patients. And there is no really treatment for this disease. So in my next slide, I will be focusing in this geminine family and basically do GemC1 or Linkeas and MCEDAS or EDAS, which are two of the members of this family. That was a long team that started quite a few years ago, several years ago actually. Identified these two genes have a very limited restricted expression just in a cell layer just next to the ventricular lumen in the periventricular space. And that was the first experiment that we did at that time to see the functionality of these two factors. And this is in utero electroporation. Basically, we remove the uterus and we inject in the mouse embryos the DNA of choice. We put in an electric field, and the DNA is incorporated, is introduced in cells which are next to the ventricular lumen. We put the babies back, and the development proceeds normally. The babies can also be born. So in this initial experiment that Christina performed at that time when she was in the lab, we electroporate either the control, which is a GFP, and two plasmids expressing GFP and either EDAS, either Linkeas, and the result, you can see it in these sections. Normally, when the cells, which are stem cells at that point, receive the DNA, become GFP, they differentiate and migrate out in the outer layers, becoming most likely neurons. However, when you overexpress EDAS or Linkeas, the cells basically are stuck in this periventricular space. So I feel guilty with this slide because basically it's 1/3 of this thesis in a slide, but this is how it goes. Summarize a big part of Christina's work, trying to identify the process, what's happening if we overexpress with this in utero system, GemC1 or MCEDAS. Basically, at that point, these radial glial cells, the neural stem cells, they will never give rise to ependymal cells, and what we have noticed is that upon GemC1 or EDAS expression, they downregulate their characteristics of neural stem cells like PAC6, BLBP. They exiting the cell cycle as Ki67 become reduced, and they switch on markers which are characteristic for ependymal cells like CMIP, FOXJ1, and S100 beta. So that was a really exciting result. So what we wanted to see also about a non-overexpression system, an in vivo system, and we generate mice lacking GemC1. And this is one of the sections of the mice that you can see that these mice develop hydrocephalus by this enlarged ventricular lumen. And the next thing is, why do they develop hydrocephalus? The obvious thing to look at is whether they have ependymal cells. So this is immunofluorescent experiment from the ventricular lumen. So we isolate all of the wall facing basically the lumen, and we do immunofluorescence with beta-catenin that shows the border of the cells, or acetylated tubulin, and acetylated tubulin is a good marker that indicates this multiple cilia, which is a characteristic of ependymal cells. So you can see in the control that we have this accumulation of acetylated tubulin indicating this multiple cilia. However, in our GemC1 knockout mice, we have a single cilium. So basically, these cells are not a multiciliated ependymal cell, so they are something different. So when this process has been stopped, is something which is a terminal differentiation defect, or is an early defect? So two of the markers that are critical for the differentiation of the ependymal cells is FOXJ1 and P73, two transcription factors that you can see in the control section that are expressed in this periventricular cell layer, which is going to give rise to ependymal cells. So the same sections in our mutant mice shows complete absence of these two markers, so the cells do not even initiate these early steps of differentiation. And a series of experiments lead us to the following conclusions. GEMC1 deletion leads to hydrocephalus in mice. GEMC1 is needed for the differentiation of the multiciliated ependymal cells, and the absence of that is because in the absence of GEMC1, we disrupt the commitment, the early events towards this lineage. That was further confirmed by transcriptomics analysis of the SVZ wall, and that indicates that all the transcriptional program linked with multiciliated ependymal cells is downregulated. Quite nicely, with a collaboration with Christopher Kale, a neurosurgeon from Yale Medical School, he has identified a patient with hydrocephalus that had a point mutation in GEMC1, suggesting that hydrocephalus occurs also in humans, and this gene has a critical role in the generation of ependymal cells, both in mice and humans. So how about the other gene, MC-EDAS? So MC-EDAS, we again generate the knockout. They do have hydrocephalus. Again, in the same experiment, looking by immunofluorescence the lateral walls. In the control, we have this nice multiple cilia in ependymal cells. However, in our mutant, we have again monociliated cells. The difference with GEMC1 is indicated in this slide. When we looked for P73 or FOXJ1 expression, we see that both in control and in MC-EDAS mutant mice, GEMC1, so absence of GEMC1 maintains the expression of P73 in FOXJ1, suggesting that GEMC1 is important for the early commitment of the lineage. However, MC-EDAS is needed for late differentiation effects. So we investigate by several experiments that are summarized in this slide, which is the mechanism, how these two factors, which are not transcription factors, regulate this program that leads to ependymal cell generation. And what we have identified is that both GEMC1 and MC-EDAS, they form heterodimers with E2F4 and 5, and actually they are binding to the promoters and regulatory elements of all these genes, leading to the transcriptional activation of these key genes for the ependymal cell generation. So one of the questions that we had is that with a GEMC1, it was clear that the cells did not even initiate the pathway of ependymogenesis. But what has happened with these cells that could not become ependymal cells? So I'll show you some of the results. So one of the markers that we have looked is GFAP. You can see GFAP in the control mice. So again, this is the ependymal layer of cells. GFAP has a very specific cuboidal expression pattern. However, in our mutant mice, in GEMC1 mutant mice, the GFAP expression has a radial morphology, suggesting that these cells are like neural stem cell or neuroprogenitor cells. Again, moreover, the expression of BLBP, another marker of neural stem cells, pointing to the same direction by quantifying the number of BLBP positive cells in our mutant mice, we saw also a statistically significant increase of BLBP positive cells. So VCAM1 has been suggested by Builla's lab as a marker of adult neural stem cells, and we examine again by immunofluorescence experiments, whole amount immunofluorescence experiments in lateral wall, the expression of VCAM1 in control and GEMC1 knockout mice. And what you can identify, I think, easily, that we have an increased number of VCAM1 positive cells in our mutant mice. So trying to further define that and clarify, we perform single cell knockout experiment, and by electroporating the Cre and having unique cells in which GEMC1 was deleted, and score, in this case, GFAP and CD31. We know that adult neural stem cells, they are GFAP positive, and they project from one side towards the ventricular lumen, and from the other side, they are in contact with the blood vessels. So what we can identify is a statistical significant increase of this type of cells when GEMC1 is absent. So as it seems, and I will show you also in the next slide, so we have indications that the cells that do not become ependymal cells, they are actually becoming adult neural stem cells. We confirmed that by a TAXEC experiment, either looking in specific genes characteristic for ependymal cells or BLBP, or having a more global picture, and that indicates that upon GEMC1 deletion, a large subset of genes which is linked with neural stem cells is activated. So obviously, I didn't have the time to go through all this data that suggesting that upon GEMC1 deletion, the cells that do not become ependymal cells becoming adult neural stem cells. At the same time that our work was published, was another work published that indicates that there is a subpopulation of radial glial cells giving rise to ependymal cells or adult neural stem cells. Actually, together with Spassky's lab, we saw that GEMC1 is critical for ependymal cells and geminin for adult neural stem cells. So we managed to make this drawing basically with work from our lab and other laboratories, indicate that GEMC1 and MC have a pioneering work in making the ependymal cells. So the next idea that we have, is it possible that these critical transcription factors can turn ependymal cells to ependymal lineage any kind of cell? So we see astrocytes. Astrocytes is a population which is interesting because in hydrocephalus, when ependymal cells are deleted, these are replaced by astrocytes. So we infect astrocytes with EDAS or GEMC1, and we score initial markers for initial differentiation, for terminal differentiation having multiple cilia, and for functionality by having motile cilia. So I will go through some of the results. Is that astrocytes obviously never become ependymal cells upon culturing conditions. You can see from these drawings that upon expression of MC-EDAS, they upregulate P73, and also they upregulating FOXJ1, and so indicate pericentric accumulation, which is a marker that these cells are going to become multiciliated ependymal cells. Moreover, we looked for acetylate tubulin, indicating that these cells carrying multiple motile cilia, obviously upon the expression of MC-EDAS. So the next thing that we wanted to see is that, all right, we have cells which are becoming ependymal cells, but do these cells have functional cilia? So are these cells motile? And as you can see that with these fluorescent beads, that these ependymal cells, reprogrammed ependymal cells, can move these fluorescent beads, and this is actually the quantification of this experiment. And indeed, these cells, they do carry multiple cilia, which are motile, as I just showed you. Similar results we receive also with the other gene, the GEMC1. Just because the time is running out, I will go to the in vivo data. So what we thought, since we can reprogram cells to the ependymal lineage in this, let's say, ex vivo system, is it possible to reprogram cells in vivo in models, in mouse models of hydrocephalus? So basically, what we did, we used two models, one genetic and another one which is chemical-induced hydrocephalus. and we overexpress with electroporation the two genes. So just to show you that in the genetic models that I just showed you, we never get multiciliated ependymal cells. However, when we overexpress EDAS or GEMC1, we have upregulation of P73 in the cells that are overexpressing EDAS or GEMC1, and FOXJ1, again, quite significant upregulation. And then the next thing is, do these cells in vivo now, they become multiciliated? You can see that in these cells we have accumulation of pericentrin that indicates from this multiple basal body that we're going to get cilia, and indeed we get multiple cilia in this in vivo model. So, and the relevant quantifications. And I think we should play this to see it. And these are time-lapse experiments that you can see that we get in vivo in this organotypic culture cells that carry multiple motile cilia. In the mutant, we never get multiciliated ependymal cells. And there again, in the other model of hydrocephalus, which is an LPA-induced model, again, we get upregulation of P73 upon the expression of EDAS, and also we have pericentrin accumulation and acetylate tubuli, indicating that these cells, again in the other system mouse model of hydrocephalus, we have cells carrying multiple motile cilia. So just summarizing the data that I saw you, the last bit of reprogramming, both in vitro, ex vivo, and in vivo upon the overexpression of EDAS or linkeas, we can initiate the first step of epidemogenesis, and actually we can get at the end cells which have multiple motile cilia and they are functional ependymal cells. And similar results we got also in vivo in our mouse, in both models of hydrocephalus. So just I would like to thank the people that did this work. That work initiated when Christina Kirousi was in the lab, continued by Marirena Lagliotti, and today's, in our days, is Konstantina Kaplani, who did most of the experiments of the reprogramming, Georgia Loka, Anna Bibli, and Stella Vassalou. And this work was mainly funded by Hydrocephalus Association USA, the part of the reprogramming, and thanks to collaborators, locals, and outside Greece. There are openings for people who are interested. Thank you very much for your attention.
Thank you very much, Stavros, for this very, very nice talk. Questions? Yes, Mimika.
Thank you very much, Stavros. That was a very interesting talk. I would like to ask you, in the last part of your talk, in the in vivo parts, was C-MIDA sufficient to at least partially alleviate the hydrocephalus, the enlarged ventricles observed in the hydrocephalus?
First, what I want to say, in order to emphasize further the role of EDAS and GEMC1, is that we have also tried with P73 and FOXJ1 the programming process that does not work. So it's only these two genes, is not the factors which are downstream. The targeting of cells that we are achieving is not so high in order to believe that we're going to have let's say, improvements of hydrocephalus. That's why I mentioned this is a proof of principle study, and now we should also try to get higher numbers of reprogrammed cells.
Okay, thank you.
Pano?
Yes, Stavros, thank you very much. Great work and fantastic data. Have you tried? Is this both facts? Have you tried them together to increase I don't know, the effect at the same time?
Yeah.
Okay.
So this is something that actually is one of the goals that we're going to do. We want to, to have them both. Even though, because we know, I didn't go so much in details, we know that GEMC1 upregulates MC-IDAS, the endogenous MC-IDAS, and then MC-IDAS has an auto-regulatory loop that maintains the system. So I doubt that would make any difference, but because we have biochemical experiments suggesting that these two factors are working as heterodimers, so it's an experiment that we'll try to do it. However, with viral vectors, it might be a bit difficult with electroporation, at least to get some initial indications.
And are they involved in spinal cord layer, for example? Have you can mark the lineage?
Spinal cord, they do not have multiciliated cells, so they have biciliated cells.
Okay, so
and
different.
We believe that this is a different origin. At least these are the data up to now. We don't know whether there is something in common. It is something that we investigate right
very
now.
Nice, very nice. Thank you.
Thanks.
More questions? I'd like to ask some questions. Actually, the hydrocephalus in humans, is it related to mutations to these two genes, or is it other reasons?
So hydrocephalus has basically two categories. One is congenital, and the other one is acquired. So a portion of the congenital is based to the genetic. Has a genetic basis, so there are genes which are mutated. However, even in the congenital, there are other factors that contribute to hydrocephalus. For example, viral infection or different accidents of the embryo, or the mother that link to the embryos. Hemorrhages basically is a big important because platelets, when they enter into the ventricular lumen, they destroy for some reason that is not 100% clear, the ependymal layer. And this is also the same for the later on during adulthood. There are infections, there are tumors, hemorrhages episodes, hemorrhagic episodes which are linked with hydrocephalus in adults.
Okay, and when the ependymal layer is destroyed, is this the mechanical effect of the pressure of the cerebrospinal fluid that dilates the ventricles, or what is the mechanism?
So the role of ependymal cells in pathophysiology, it has first the circulation in of the cerebrospinal fluid, meaning because of this motile cilia contributes to the circulation of the cerebrospinal fluid. So if they are not present, they accumulate, and that puts mechanical pressure. But the second thing is that because ependymal cells secreting also factors and supporting the knees, so we know from mouse models that when ependymal cells are not there, neurogenesis it does not operate properly. So I think they have also trophic supporting role besides the circulation of the CSF.
Thank you very much.
Thank you.
And now I have the pleasure to introduce Yasami Kutmane, who has stemmed from our institute, the Pasteur Institute in Athens, where she did her PhD, and is flourishing in the Bioacademy Research Institute for quite a few years. And she's going to talk to us today about corticotrophin-releasing hormone in human brain development.
Okay, thank you, Rebecca. First of all, I would like to thank the organizing committee for giving me the opportunity to present you our work here in this very nice meeting and in this very charming venue. And especially I would like to thank Rebecca Machas for the invitation. Before I start presenting our work, I would like to to tell a short story about why our team is interested in the role of CRH in human brain development. Now, the causal underpinnings of neurological and psychiatric disorders are largely unknown. Scientists have discovered a great number of susceptibility genes that, unfortunately, many of them lack replication. And this is mainly because it is very difficult to identify the environmental factors that are implicated and interfere with the genes and interfere with the causality of the diseases. Now, this is also the case not only in the adult brain, but also during development. Now, the purpose of our lab is to gain an understanding in human brain physiology by focusing on the microenvironment of the most dynamic cell population of the brain, which are the neural stem cells that produce the new neurons in the brain. From all of these microenvironmental factors, we are especially interested in stress hormones. We hope that our studies will help design appropriate therapies and achieve regeneration when this is needed by mimicking this niche environment of the neural stem cells. Now, why corticotropin-releasing hormone? First of all, what is corticotropin-releasing hormone, and which is very often referred as a stress hormone? Corticotropin-releasing hormone, or CRH, was first discovered by Vail in 1981, and Vail, unfortunately, is not longer with us. And he has also discovered that CRH is the major mediator of the adaptive response to stressors. That's why we call it a stress hormone. Now, what are the functions of CRH? CRH has many, many functions. Under stress conditions, CRH is secreted by the hypothalamic neurons and goes to the pituitary gland, where it activates the secretion of ACTH. ACTH circulates in the body via the blood stream and goes to the adrenal glands, and there we have the secretion of cortisol. Glucocorticoids, such as cortisol, have a negative feedback in the hypothalamic neurons that secrete CRH, so the axis stops by a negative feedback. So as you can imagine, CRH is a major homeostatic factor. So when the stressors are not very strong, not very severe, the body comes to an equilibrium. But when the stressor is very intensive, or when we have a prolonged stress, then CRH continues to be produced by the hypothalamic neurons, and then this axis never stops. Glucocorticoids are in a very high levels in the blood, and this causes a very toxic effect in all the organs of our body. This situation could result even in diseases such as depression or cognitive deficits, or even more severe situations. Now, what happens under non-stress conditions? Does CRH have a role in the CNS, in the central nervous system? The answer is yes. CRH is secreted in a great number of areas in the adult brain, and it acts via its two receptors, CRHR1 and CRHR2, which, as you see here, are distributed in a very in a very different way in the adult brain. CRH is also expressed in the periphery and, most interestingly, in the placenta during pregnancy. But we will have the time to talk about placental CRH later. Now, our main focus was to study CRH function in the most dynamic process and the most challenging situation in the adult brain, which is hippocampal neurogenesis. Why hippocampal neurogenesis? Because neurogenesis in the hippocampus is evolutionary conserved in humans, and it is linked to very important cognitive functions, such as memory and learning. Now, as I said, our study focuses in the adult brain and in the adult neurogenic process. As you see here, to produce new neurons, neural stem cells, have to pass through a great number of stages, and that include many cell types. What we found is that CRH system, both CRH and CRH receptor, are expressed during all this process, in all stages and in all cell types. This was further confirmed by adult neural stem cells extracted from the hippocampus and cultured, where we identified in their media CRH in a very big proportions. This raises the possibility of an autocrine action of CRH in adult neural stem cells. But what is the biological significance of all this broad expression of the CRH system here? To address this question, we used the global CRH knockout mice, and we found that complete ablation of CRH results in a reduced proliferation and reduced neurogenesis in the hippocampal niche, so it causes severe defects in hippocampal neurogenesis in general. Most interestingly, CRH depletion causes also behavioral deficits that are hippocampal dependent, such as specifically spatial memory. Now, to assess the ability of adult neural stem cells to respond to the environmental factors, we subjected mice, CRH knockout and wild-type mice, in two different conditions. First, involuntary running, which we know from the bibliography that induces proliferation of adult neural stem cells, and enriched environment, which facilitates the survival of adult neural stem cells. What we found is that in both cases, in both stimuli, the adult neural stem cells in the knockout mice had lost their ability to respond to these beneficial factors. So this is very important because neural stem cells not only loses their intrinsic ability to produce neurons, but also they lack this ability to respond to the environment. Now, all this data were taken from the global knockout mice, where we have total absence of CRH in the system. But what happens locally, at the local level, to find out if CRH has a local action in adult neurogenesis? We performed short-term inhibition and overexpression of CRH, as well as long-term exposure to CRH using pharmacological administration of CRH and CRH receptor antagonists, or adeno-associated viruses that express CRH. And we found in both cases that both the short-term and the long-term exposure to CRH had significant effects in the process. Even more, Spatial memory was found to be at the normal levels after administration of CRH in CRH knockout mice locally. So this is also very important because here we have a proof that CRH exerts its function in adult neurogenesis at the local level, at the niche. Now, to see if CRH directly affects this stem cell population, we used as an in vitro model adult neural stem cells extracted and cultured from the hippocampus, and we found that CRH has a direct effect engaging already well-identified signaling pathways. RNA-seq analysis revealed thus that partly this effect was mediated by BMP4 inhibition, and this was further tested by an in vitro experiment. Finally, we identified CRH expression and CRH system expression, both CRH and CRH receptors, in the human adult neurogenic niche. So we have a conserved expression of CRH in humans. This was also further investigated, and we found that neural stem cells that arise from the induction of human embryonic stem cells also express the CRH system and also secrete CRH in the medium. So our hypothesis is that this regulatory role of CRH in adult neurogenesis may be the case also in humans. Now, all this data I present you was from our work in the adult mouse brain. What about the function of CRH in the neurogenic niche during brain development? Now, 10 years ago, we studied the phenotype of the CRH knockout mice during development, and we found that ablation of CRH causes defects both in proliferation and survival of the neural stem cells during embryogenesis. Now, in humans, what we know is that what we have described is that CRH system is expressed in the ventricular and subventricular zone of humans, so it is present also in the proliferative zones in humans during embryogenesis. So, to summarize, corticotropin-releasing hormone, whether it is under stress conditions, where it acts via a systemic action, or under non-stress conditions at the local level, is a major homeostatic factor. So it is its role to provide an environment of homeostasis and to be able to respond to any environmental challenges so that the balance comes back to the organism. Now, what happens during pregnancy? What happens during pregnancy is very interesting because only the human placenta and the placenta of anthropoid primates produce huge quantities of CRH, that most of which is secreted in the maternal bloodstream, but also some of it crosses the blood-brain barrier during development and affects the embryo. It is quite impressive that during pregnancy, CRH concentrations rise over 1,000-fold. As you can imagine, and pregnancy is a very challenging homeostatic situation for the organism. A lot of changes are present, and the organisms have to adapt. Now, there you see a graph depicting the huge elevation of CRH level during pregnancy. And here you can see that CRH is only present in the anthropoid primates placenta. Now, there are some studies indicating that abnormal levels of placental corticotropin-releasing hormone are associated with brain disorders and cortical thinning of the embryos, of the human embryos. Now, our questions were why CRH is expressed only in the human and anthropoid placenta? And what is the biological significance of this unique CRH placenta expression? Finally, how CRH is implicated in human cortical development if this is the case? To address these questions, as you can imagine, we have some very big limitations. Of course, we have limitations arising from bioethical issues, but also from the fact that we have completely absence of placental CRH in the classical mouse models. Moreover, although human cortical development present many similarities with the mouse cortical development, they have also important differences. For all this reason, the mouse model is not an appropriate model for us. What we have used as an experimental model to find out what are the effects of CRH in human brain development is 3D neural spheroids and human cortical organoids. What is the difference between these models? 3D neural spheroids are relatively homogeneous structures and present no cytoarchitecture, whereas cortical organoids develop inside them structures that are similar to early corticogenesis. Now, we first investigated, we tried to study the stem cells, the human stem cell neural spheroid system, deriving from H9 embryonic stem cells. And we found that in these cultures, we had endogenous CRH expression, but also we had expression of the CRH receptors. So this system can react to the endogenous CRH or to the exogenous CRH if administered. That's why we tried to find out the role of CRH by administering CRH or CRH receptor antagonist, NBI, at the neural stem cell level and until the end of the experiment. So this means 50 days later. Now, when we analyzed these structures 50 days later, we found that the NBI, so the blockade of CRH receptors, causes a reduced size of these homogeneous structures. This is happening without reducing the neural stem cell population, as you see here. Moreover, using specific markers for the neuronal lineage, we found that the inhibition of CRH signaling inhibits the maturation of cells to a neuronal phenotype, and this is more dramatic in the glutamatergic phenotype of neurons. In addition, CRH preferentially inhibits cells to acquire astrocytic morphology, whereas NBI has an opposite effect by inducing the astrocytic differentiation in these cultures. We have exactly the opposite effect in oligodendrocytic lineage, where you see here that NBI causes a reduction of oligodendrocytes. Now, to see if CRH, apart from the effects in the neuronal lineage and the differentiation of cells to different cell types, had also an effect in the cytoarchitecture and the cortical development, we used the human cortical organoid model. Now, we also treated these structures with CRH and with pharmacological, and we also induced pharmacological inhibition of the CRH lineage. And we picked up all these cortical structures that you see here, and we tried to investigate the role of both induction of CRH signaling and inhibition of CRH signaling pathway in this morphology. Although the analysis of these structures is still in progress, we have some preliminary data that, as we see here, focuses on the thickness of the cerebral wall. Now, this is the cerebral wall, and using specific markers for the cortical development, we see that CRH causes a thickening, whereas NBI causes a shrinking of the cortical wall. Here you see the statistics of these measurements, and you can also observe here that not only CRH induces a thicker cortical wall, but also results in a higher proliferating potential in this in the proliferating zones. Now, to summarize, we have, using these two human models, we have revealed CRH system expression, and we have shown that both inhibition and exposure to CRH, to high CRH levels, can cause alterations in the growth rate of the spheroids, in the neurogenic, oligodendrogenic, and astrogliogenic potential of neural stem cells, as well in the differentiation of cells towards a glutamatergic phenotype. Moreover, we see that CRH and CRH inhibition have a direct result in the size of the ventricular zone and the thickness of the cortical wall in these cortical structures. So all these results show us that CRH has a potent active role in the process of human brain development, which paves the way for future pharmacological use of CRH and its antagonists against neurodevelopmental disorders. Also, our findings could lead to a better understanding of the evolutionary significance of the role of the placental CRH in primates, and more specifically in humans. Now, finally, I would like to thank George Elinas and Iliana Theodorou, who contributed in the work of the human spheroids, and also I would like to thank Dr. Katia Karalis, Yannis Serafimidis, and Panagiotis Politis for their scientific contribution in the project of adult neurogenesis. all the other previous members of our lab and collaborators in the BRFAA Institute, but also our collaborators in the Hellenic Pasteur Institute, Rebecca Matza, Georgia Kouroupi, Florentia Papastefanaki, Dimitra Thomaidou, and Pari Koutsudaki, our collaborators in the Harvard Stem Cell Institute and in the Medical School of Athens. And thank you for your attention.
Thank you very much, Yasemi, for this very nice talk and the very nice and recent data you have obtained for CRH. Yes, please.
Hi, thank you. Thank you for the nice overview. Just a quick question. Was a little bit intrigued by these ethical barriers to study the human placental. Do you think recent advances in trophoblast modeling with human stem cells would be a better model to study the interactions with these spheroids, maybe co-culture with trophoblast stem cells? Do you think would be a better proxy for the system?
Yes, of course, very nice question. Recently, I think 2 years ago, there was a paper from a group that produced placental trophoblastic, let's say, structures, okay? Not placental, it's not possible, but for the moment. And these trophoblasts showed an endocrine activity similar to humans, similar to that we found during human pregnancy. So it is a very interesting model to combine these two structures, the trophoblast structures and the human brain, human cortical structures. It's very interesting. Thank you.
More questions? Christina.
Hi, Yasemi. Thank you very much for your talk. It was very exciting. I was wondering whether you have checked in your organoid model, whether you see specific changes only in basal red glial progenitors that are supposed to be those that will expand the neural population in humans, or if you haven't, if you checked whether you have expression of CRH or receptors in these type of populations in single cell sequencing data or any other data?
Yes, thanks for the question. So as I mentioned, these structures are under investigation. So we are analyzing them, and we have only used a small number of markers. But radial glial cells are very important to be checked at this level. And also we have checked for CRH receptors expression and CRH, but we have a problem with the affinity of the antibody in this
But you
tissues.
could check from published single-cell sequencing data, at least the RNA level of
yes,
these
it is
genes.
expressed.
Yes.
RNA is we have also shown this. Because we have performed RNA analysis, and we have expression of both CRHR1 and CRHR2, which is interesting because in
basal red glia. In basal red glia?
No. In the bulk, from bulk RNA analysis, no. We have not done this. But as I mentioned, these structures are under analysis, and this is very important to identify in which exactly population CRH system is expressed, and how do we come up with this structural difference.
And if I may
do
another one, do you think then that the CRH could be a mechanism, let's say, evolutionary mechanism to grow bigger brains? So, for example, what would you speculate if you put CRH during embryogenesis in mice?
In mice.
For
Very
example,
good
if you
question.
put CRH in the embryo.
Yes. A very nice question. That's what we are planning to do. We have done this, but the embryos die, unfortunately. We inject adeno-associated viruses that express CRH in the mouse placenta during several stages of embryogenesis, and unfortunately the embryos die. So we are considering, because probably placenta is a very sensitive organ, and if you disrupt this structure, the embryos are very sensitive and they not manage to survive. But we will try to induce CRH expression by injecting directly in the amniotic fluid now.
Yeah.
So let's see.
Okay, thank you. Thank you so much.
More questions? Yes, it might be an idea to inject in the ventricles as well in the developing embryo.
Yes.
And also maybe in vitro as well, because I had the same idea exactly as Christina. What happens to the mouse developing brain if you raise it by 1,000 times the concentration of CRH, for example?
Yes, that's a very nice suggestion. So injecting it directly in the ventricles, yes, that's a possibility also. But the CRH, just to clarify this, CRH expression is 1,000 times more than the physiology in the human placenta, but an 80% goes to the mother.
Okay.
But even so, a 20% is a huge amount of CRH that the embryo faces during pregnancy. Thank you, for the suggestion.
If there are no more questions, thank you very much, Yasumi.
Moving to the next talk, it's a great pleasure to have with us Dr. Apostolos Klinakis. Apostolos Klinakis is an investigator at full professor level at Biomedical Research Foundation of the Academy of Athens, where he has established the Cancer Biology Lab, and he has very interesting results on tumorigenesis. So today, Apostolos is going to talk to us about in vitro cancer models. Apostolos.
Good afternoon. Thank you for the invitation. Thank you to the committee and personally to Maya, and thank you all for being here. Last week, when I looked at the final program and I saw the title of my presentation, I thought I was being trolled or something, because those who know me know that our group uses almost exclusively genetically engineered mouse models to model tumorigenesis, obviously. So that in vitro was kind of puzzling. I'll do my best to support the title towards the end of the presentation. And to be fair, probably Maya mentioned it at some point that this would be the title, and it just didn't register with my forgetful brain. So the use of models is not new. 2,500 years ago, Aristotle expressed in that work of his on the parts of animals the idea that we could exploit similarities between animals to understand human biology or physiology. And he proposed that we start from those that are similar and then extend out to those that are not that similar. And in fact, people have been using models, animal models, to understand human biology going back to the 6th century BC. And here there are some examples of that. So, and this, I didn't talk with Stavros before, so we have a similar slide here. Our group, in the recent years actually, has invested a lot in studying epigenetics in tumorigenesis. We're particularly interested in the role of epigenetics in DNA damage response and DNA repair, and consequently to therapeutic response, the response to DNA damaging agents. And what is epigenetics? The definition nowadays is any stable and heritable change in gene expression that is not associated with changes in DNA sequences. And this was expressed for the first time by Con Waddington something like 70 years ago in his effort to explain something that today is very, very trivial, but back then was very puzzling. How come a clone of cells, which is the human body, all derivatives of a single cell, the zygote, how come and those cells are so different in phenotype and in physiology? So in other words, without changes in the DNA content, how is it possible that those cells look so different? And this, he thought, should be something beyond genetics, over the genetics, epigenetics in Greece. And nowadays we know exactly what epigenetics is about. It is about changes in histones, post-translational modifications in histones, changes in the DNA, and more recently non-coding RNA. So the majority, not the majority, exclusively in this talk we'll talk histone modifications. As you all remember from high school biology, DNA is not naked in the nucleus. It exists in the form of a complex with proteins, histones, 4 pairs of those, forming these nucleosomes. And thank God this is the case, because otherwise our nuclei would be a mess. Imagine that the DNA threads in each of our cells, if you connect them with one another, they form a straight line of 1.5 to 2 meters. So imagine a very thin thread, or 23 threads of 2 meters in length, being together within a 10 micron nucleus in our cells. How many, what a mess this would be. And this is the nucleosome. As you can see, each histone extends the tail out of the nucleosome. And these tails have amino acid residues there that are subjected to post-translational modifications, mainly acetylation, methylation, and phosphorylation. And it's more than one of those. And there are modifications that are associated with an open chromatin, euchromatin, or closed chromatin, heterochromatin, and those are the active markers and the repressive markers. And this is a dynamic process mediated by enzymes, obviously, each of which has a specific function. So a histone 3K4 methyltransferase is responsible for putting methyl groups in lysine 4 of histone 3, and that's what it does. They're not promiscuous in that sense. And for each of those modifications, because this is a reversible and dynamic process, there is an enzyme that does the opposite. So there is a demethylase. The same way we have acetyltransferases and deacetylases, and so on and so forth. The combination of these post-translational modifications, in other words, which modification in which amino acid residue composes what is called the histone code, and now we know the role of histone code in gene expression and other processes. By regulating the structure and the accessibility of chromatin, epigenetics affect or control transcription, DNA damage repair, and DNA replication, because in all these cases, as you all know, all these processes actually are being performed by huge, very large protein complexes which need to directly access the DNA, and the accessibility obviously matters in this case. One of the biggest surprises of the Cancer Genome Atlas project in the last decade was the realization that epigenetic regulators are among the most frequently mutated genes in most solid tumors and also hemopoietic malignancies, and particularly in bladder cancer, which is coined as the cancer of chromatin. You see that proteins such as MLL3, MLL2, which is now called MLL4, ARID1A, and other histone-modifying enzymes are mutated in frequencies that are comparable to the P53. And this was quite a surprise, and it just happened that at the same time that these papers started coming out, our group was already working with MLL3, which is also called KMT2C, because its knockout in mice, and that was a published report, phenocopies the knockout of the Notch signaling pathway in the urothelium. And because we have been working with the Notch signaling pathway, we thought that maybe there is an interconnection, and we started studying KMT2C or MLL3. And later on, all these studies came out showing that indeed these are important in tumorigenesis. Now, the KMT2, K for lysine, methyltransferase 2 family of proteins, KMT2 A, B, C, D, E, and G, F I think is missing, or MLL 1, 2, 3, 4, 5, 1, 2, 3, 4, and set D1 and 2. They are responsible for histone 3K4 methylation. Now, Methylation of lysine 3 and histone 3 along with demethylation of lysine 27 in histone 3 are the hallmarks of open chromatin. So all regulatory, all promoters and enhancers that are active and all genes that are expressed are demarked in our genome by histone 3 K4 methylation and histone 3 K27 acetylation. And because acetylation and methylation are mutually exclusive, they occur on the same lysine, K27. Only one of the two may exist, so demethylases are important for histone 3 K27 acetylation. And this process, this reaction, let's say, this modification is mediated by the compass and the compass-like complexes, which are large, very large conglomerates of proteins containing, among others, MLL family members. Now, if one looks at TCGA data for the 4 first members of this histone 3K4 methyltransferase family, you will see that they are very frequently mutated in lung adenocarcinoma, squamous cell carcinoma, head and neck squamous cell carcinoma, breast cancer, and bladder cancer with very high frequencies, particularly for MLL3 and MLL4, the C and D, which people have been considering for a long time functionally redundant. This is not the case. And the majority of these mutations are these black boxes indicating that these are stop codons. So either by direct nonsense mutations or frame shifts that generate in-frame stop codons, these proteins are being truncated, indicating that they might be tumor suppressors, and functional data in mice indicate that indeed they are tumor suppressors. And not only they are mutated, not only are they mutated in cancer, but particularly for KMT2C, they are also downregulated. So if one compares pairs of cancer tissue and healthy tissue from tissue from the same organ, bladder, breast, colon, and rectum, and so on and so forth, you will see that in red, the expression of MLL3 is always lower than not always, but it's lower, on average lower than the healthy counterpart, which means that besides mutations, these genes are being, let's say, inactivated, or they are being loss of function due to downregulation as well. And our group has already published that the expression of MLL3 is being controlled by 2 CpG islands on the promoter of the locus that are being methylated in cancer, in bladder cancer. Now, I've mentioned already many times bladder cancer. Our group works on bladder cancer. The urothelium is a specialized epithelium lining the urinary tract, starting from the renal pelvis all the way to the urethra. And its main role is to form the urine-blood barrier, which is one of the strongest barriers in our body, because as you know, all the things that we want to dump off are concentrated in very, very high concentrations in the urinary bladder, and there must be a very effective barrier preventing them from re-entering the bloodstream. Cancers, starting at the urothelium, represent over 90% of bladder cancer. So 90% of bladder cancer is urothelial carcinoma, and they can appear as superficial papillary tumors, or in situ, or invasive, and eventually metastatic. And at the metastatic state, it is a very lethal cancer. So our group has been working using genetically engineered mouse models, practically conditional knockouts with KMT2C, KMT2D, ARID1A, and KDM6A. The two are histone 3K4 methyltransferases. ARID1A is a Schweitzer complex, and KDM6A, or UTX, is a demethylase of K27. And we generated conditional knockout models for KMT2C. Actually, these were the first conditional knockout mice we made in the lab with CRISPR. And as you can see here, 6 months in the mouse life, upon knockout of KMT2C, these mice develop mild hyperplasias. This is the normal urothelium. This is an empty bladder of the mouse. And as you can see here, the wall is very thin. In fact, in the mouse, it's 2 or 3 cells thick, whereas in humans, it's like 5 to 7 cells thick. The human bladder is thicker than the mouse bladder. And you can see that there is an expansion. There are more than 2 or 3 layers there, and this is all you get even after 1 year, which means that these genes are not drivers in tumorigenesis. So alone, on their own, they do not suffice for development, for tumor development. If you combine them with a mutagen called BBN, which is a very long name I can't remember, but it is an nitrosamine, which is a component of the tobacco smoke. Bladder cancer, as you probably know, is the result of smoking and alcohol consumption. You can see that if you put mice in nitrosamine, you put that in the drinking water and they drink it daily, wild-type mice develop invasive tumors and die after more or less six months. MLL3 knockout mice, KMT2C knockout mice, are being sensitized in agreement with their role as a tumor suppressor. And if you make organoids from those mice, you will see that the clonogenic capacity of MLL3 knockout organoids is much higher not much higher, it's a bit higher, but statistically significant in comparison to control. And that's the beauty of organoids. This result you get in 6 days, these results you get in 6 months. Now, when we knocked out KMT2C in bladder cancer cells, and we did RNA sequencing, and we looked at the gene, we performed the gene ontology analysis. In other words, we grouped the downregulated, in this case, genes based on what processes they're involved in. We found a lot of DNA damage response and cell cycle control, and a lot of BRCA and hereditary breast cancer, which directly pointed towards homologous recombination DNA repair. As you all know, BRCA1 and BRCA2, among other proteins, are involved in the homologous recombination, and homologous recombination is one of 3 methods for double-strand breaks to be repaired, and it is the most accurate one, it is the preferable one, unfortunately, because it uses the sister chromatid as a template for DNA repair. That means that it can only perform beyond the S phase when DNA has been replicated. There are special assays to monitor double-strand DNA repair, and double-strand breaks are the most dangerous breaks the most dangerous DNA damage in our cells, and there are specialized assays to measure and quantify a homologous recombination. As you can see here in comparison knockdown cells, the number of sister chromatid exchange events, which is the hallmark of homologous recombination, is lower, is much lower. Practically, these cells are HR deficient. And how does this happen? At the time we published this, we found that BRCA1, BRCA2, ATM, ATR, and other proteins involved in the homologous recombination machinery are downregulated in the KMT2C knockout cells. And then by going to TCGA, we found that in breast cancer, in colon cancer, in non-small cell lung cancer, and head and neck squamous cell carcinoma, and something like 10 other cases which are not here, in all those cases, the expression of BRCA1, BRCA2, ATM, and NTR correlates with the expression of KMT2C, and actually chip sequencing experiments, which I'm not showing here, indicate that KMT2C directly controls the expression of those genes by sitting on their promoter and opening the chromatin. And 2 papers that came out, one before ours and the other after ours, propose alternative methods through which KMT2C is involved in homologous recombination DNA repair. As you probably don't know, at least those that are in the oncology field, ovarian cancers, and nowadays breast cancers and prostate cancers and pancreatic adenocarcinoma cases that carry either germline mutations in the BRCA genes or somatic mutations in homologous recombination genes. And also nowadays another indication is HR deficiency, HRD, in ovarian cancer, respond very well to PARP inhibitors. The first PARP inhibitor that got into the clinic was olaparib in 2015. The action of these PARP inhibitors is a process called synthetic lethality that dates back to genetics in Drosophila, where investigators knew that certain mutations individually, each of them individually had no phenotype, but when combined, they had a lethal phenotype. And these were called synthetically lethal mutations, and this is an example of synthetic lethality. On one case, you have HR deficiency due to mutations to a gene that is involved in HR deficiency, for example, BRCA, and by blocking PARP inhibitor, by blocking PARP, a molecule that is involved in DNA repair of double-strand breaks, but also single-strand breaks, which, if left unrepaired during DNA replication, leads to double-strand breaks. Combine the two are synthetically lethal, and this is why these drugs are so effective in HR-deficient tumors. And as you can see here in our case, in cell lines from these are xenografts, cell lines from HTB9, from bladder, colon, lung, and head and neck, squamous cell carcinoma. In all those cases, if you look at the knockout cells, which are the bottom, and follow the green line, and actually the blue line is olaparib alone, you see that they fail to grow, whereas control cells respond more poorly. Combined with the cisplatin that induces more DNA damage, it is detrimental and is failed to go. And this was later confirmed in lung cancer cell lines in which KMT2C was knocked out. In other words, loss of KMT2C leads to HR deficiency, which can be exploited therapeutically by using PARP inhibitors. And the other thing we know from the clinic is that the same tumors that are HR deficient respond very well to cisplatin. Why cisplatin? Because this is, again, like mimicking synthetic lethality. You have cells that have lost 1 out of the 3 DNA repair mechanisms for double-strand breaks. So if you come with cisplatin and induce exogenous damage that is orders of magnitude higher than the endogenous damage, it is too much for the cells to cope with. So they cannot deal with it, they cannot repair it, and they die. That's why cisplatin is so effective in women with ovarian cancer and works for a very long time. The expectation in our case was that because these cells are HR deficient, they would respond well to cisplatin. As it turns out, they don't. So they are resistant. And if you look at mice, the conditional knockout mice in nitrosamine, you will see that wild-type mice benefit from cisplatin. You see the Kaplan-Meier here, the red line. If you go to MLL knockout mice, they don't have any benefit. And if you go to patients with metastatic bladder cancer that took cisplatin-based chemotherapy either in the MVAC with methotrexate, adriamycin, and viblastin scheme, or with gemcitabine, you will see that patients that express low levels of KMT2C have a poorer survival. And we know now, and this is under review as we speak, we know now the mechanism through which cisplatin resistance occurs. It has to do with the fact that cells learn to live with higher damage. There is damage tolerance. So they progress through the cell cycle with a lot of unrepaired damage, and this has to do with the compromisation of the cell cycle checkpoints, particularly the G2M checkpoint. Now, we went into published data, exome sequencing data from the TCGA, from primary bladder cancer patients, and whatever I'll say here is also true for the metastatic cohort of patients in the TCGA, and we looked at the tumor mutation burden. Because HR is the most precise mechanism of DNA repair, we assume that cells that are defective for the precise mechanism of repair resort to imprecise ones, like the canonical end joining and the alternative end joining, and we assume that they will have more mutations. Indeed, KMT2C mutant cells, KMT2D mutant cells, that's MLL3, MLL4, and KMT2CD double mutant cells have a higher tumor mutation burden. So that means they have a higher mutation count. And when we looked specifically at missense mutations, we found out that if you follow in pairs the bottom blue line here, the blue bar here, you will see that the mutations that are enriched in the mutant KMT2C is the C2A the same for KMT2D, the same for the double, and it is also true for the T2G transversion. Now, the C2A and T2D transversion is the signature for oxidative DNA damage. Oxidative DNA damage through oxidized oxoguanine, which pairs, instead of C with the cytosine, it pairs with the A, leads to this C2A and T2G transversion. And actually, these were the only transversions which positively correlated with the tumor mutation burden. And this is true, as I mentioned already, for another metastatic, that one, cohort of bladder cancer patients. We know now that we knew already that OXO-G, the oxidized G, is being repaired by the base excision repair mechanism, and we know now that this is defective, and this is work in progress. I'm not going to go into details about this, but it seems that MLL3 and MLL4 are involved not only in the homologous recombination DNA repair, but also in other forms of DNA repair. And now, actually, last week even, we found yet another mechanism in which these two proteins are involved in terms of DNA repair. This is a metastatic bladder. Yeah, I showed it already. Consequently, mice that are that are wild type or mutant knockout for MLL3 were treated by, we call this nivolumab. It's not nivolumab, it's an anti-mouse PD-1 blocking antibody that blocks the immune checkpoint inhibitor. And as you can see here, wild type mice treated with this anti-PD-1 antibody benefit. It's marginally not statistically significant, whereas mice that are knockout for MLL3 benefit a lot from this treatment, and actually we extended their life as long as 3 months, which is like, if you consider that they die at 6 months because of these invasive cancers, this is quite successful. And we have ongoing experiments with KMT2D and double KMT2C, KMT2D mice, and now we know and we suspect what is the source of this favorable response to immune checkpoint inhibitors. It doesn't have to do with the role of MLL3 in HR repair, but rather the oxidative DNA damage repair and maybe another case. And in the last couple of minutes of the talk, to support the title of the talk, I'll present some ongoing research and very, very preliminary and not very important yet data on the use of organ-on-chip platforms to study some aspects of tumorigenesis. So this is the emulate, the very, very basic microfluidic device they use, where you have inlets and outlets through which you can administer medium, and they have chambers in which you can grow cells or tissue, cells in this case. Now, 3 years ago, I had an important role in setting up a very large European consortium, which was eventually funded, and the project started 2 years ago, to study the tumor and lymph node interaction using human samples. And why do you want to study this in an organ-on-chip and not some other system? One reason is that we wanted to study this with human samples, and particularly primary non-small cell lung cancer samples and lymph nodes from the same patient, but also because this is something that you cannot study in mice. Mice have this peculiarity that all metastasis is hematogenous. It rarely involves the lymph nodes, and if one wants to study this crosstalk, both in terms of cell migration, but also the molecules that mediate the crosstalk, maybe the organ-on-chip would be a good platform. And what we did is we designed a chip. There was a group in the consortium which is made of 11 partners, 6 of which are small, medium enterprises, and the other are academic partners. And what we do is we take from the Soteria Hospital, which is next door, a piece from primary non-small cell lung cancer, a piece of a lymph node of the same patient, and also peripheral blood. The idea is to put them in different compartments of the microfluidic device the two compartments communicate with one another through channels that mimic the diameter of the lymphatic vessels, and also there is a flow controlled by cilia and micropumps that mimic the flow of the lymphatic fluid in our body, which has a tonal kind of movement in our body, and see whether we can build, through this, we can build gradients from one tissue to the other and see whether we can follow live the migration. For this purpose, we have employed imaging devices that focus on the channels between the two compartments, and the idea is that we are going to monitor these live, and also by characterizing molecularly the conditioned media, the lymph, that is the media between the two compartments, we will identify cytokines and chemokines that are involved in these processes both ways. This is not fully integrated. The project has a couple of years more to run out. So I'm going to present a couple of findings. The first thing we had to do is to find the conditions to culture these tumor slices for a few days, right? And these are slices, literally. We take a piece of the tumor, we slice it live using a compressome, which is a special kind of slicer, let's say, vibrotome, but it's not a vibrotome. It vibrates. It's an advanced vibrotome, let's say. These are live. You can slice them from 150 microns up to 300 in thickness, and you can culture them on static or flow chips, and we have managed to culture them for up to 2 days. And if you look at the uncultured tissue, this is an H&E section, and then the day 2 and day 4, and we also have day 5. You will see, and the pathologist at Soteria, Yannis Vamvakaris, confirms this, that this is a live tissue and super happy. And the same is true for the lymph node from the same patient, which is also being sliced live and cultured. And I have to say that from these cases, we also, as a necessity as well, because sometimes the piece of the tumor we get is too tiny to be sectioned in the compressome. Imagine that you make something that looks like ham that you slice, and sometimes it's a very thin slice that we get, because obviously the pathology comes first. So to circumvent that limitation in material, what we do is we graft it in NSG mice, in immunocompromised mice, and they grow, and the second time around, it's a big, nice, round tumor, which we can embed into this sausage-like agarose-based thing, whatever you call it, cylinder, and slice it in the compressome. And from these patients, most of these patients, we generate organoids, we generate PDXs, and we aliquot the lymph nodes and store them, and these are live stored. Like we thaw the organoids, we thaw the tumor slices, we put them back in mice and they grow again. These are not fresh frozen, these are live frozen, and upon thawing they grow. And of course we characterize molecularly these tumors, and I think this is a very good biobank, because it allows you to study various aspects of tumorigenesis and use them in preclinical testing of drugs and schemes and things like that. Anyway, so one of the very first things we noticed is the following. Keep in mind that these are operable non-small cell lung tumors, the definition of which is that it hasn't spread. So at the time of the operation, all these patients are presumed to be non-metastatic. Okay? As we all know, even if you operate and you have a clean operation, you have clean margins, something like 30 to 40% of those patients recur and progress because at the time of the operation they had micrometastases which were not detectable by the imaging modalities we use. So eventually, and although these patients most of the times, particularly the stage 3, get adjuvant therapy, they eventually progress and show metastatic lesions, which, as I said, were not detectable at the time of the operation. Now, in all cases, whenever we see migration from the top of the chamber through the membrane to the bottom of the well through an 8-micron membrane in all the patients that proved to be metastatic. Afterwards, within a month, 2 months, 3 months, 4 months, all those patients migrate, whereas those patients that remain disease-free and didn't progress, didn't recur, all those patients do not show migration. You can see these cells. These are not tumor cells. They don't stay for EPCAM, which is a marker we use. These are hemopoietic cells that penetrate the membrane and also some fibroblasts. So, in other words, in this setup, and by culturing the tumor for like 4 or 5 days, you can already predict whether this tumor is metastatic. It might take a few months for the patient to show, but in this chip, it comes in a few days, and this is kind of encouraging, although it doesn't mean anything at this point. The other thing we notice is that if one plates, we've done these experiments in simple setups, transwells, or in the chip, whenever in the culture you include lymph nodes, so you co-culture either beneath the transwell or in the other well, what you see is that the number of tumor cells that migrate through the membrane and try to reach to whatever the source of any signal there goes up. These are absolute numbers. The only case in which this goes down is this case in which we co-culture the two for 4 days, and as you can see here, the immune system of the naive lymph node, this is a naive lymph node, so the tumor coexists with its immune microenvironment in the primary tumor, but obviously it evades or it suppresses the immune response. But with a lymph node, this is not the case. So whenever there are lymph node cells from the same patient beneath that see for the first time the migrating cells of the tumor, crossing the membrane, they immediately attack. So we are at the stage now that we have isolated migrating cells, both lymph node cells and cancer cells, and we have isolated the conditioned media. And we're in the process of characterizing these initially with bulk RNA sequencing and then with single-cell RNA sequencing. Obviously, the idea being that we're going to identify specific markers that mark these migrating cells and, in the long run, try to exploit that therapeutically. And I'll stop here by thanking past and present members of our lab that have contributed to the work I presented today, our collaborators in the National Technical University of Athens, Ioanna Zeriotti and her group with which we set up the tumor lymph node on chip, various collaborators here and there, surgeons, urologists that provide the samples for the bladder, Konstantinos Potar is the thoracic surgeon at Sotiria who provides the samples with Yannis Manvakaris, and also various clinical collaborators in Greece. And of course, all those that trusted their money with us. Thank you very much.
Thank you, Apostolos, for such a nice talk. Any questions? Karsten?
So, apologies, I might have been at fault for giving you this in vitro title. I didn't suggest a title, but when we discussed the content of the conference, I referred to your exciting data, I think, on lab on a chip, and that's probably what brought it about. Hold on. So, you didn't say how many samples you've already analyzed, I think, for the lab on a chip, but it seems very exciting in terms of power and sensitivity. So maybe you would have false negatives where you get a sample where the metastatic behavior doesn't show, but I guess as soon as you see movement there, it would be indicative of metastatic. So how many samples have you analyzed so far?
So the biobank is approximately 40 samples. The truth of the matter is that, as I mentioned, sometimes the primary material is limiting, so we need to expand that in mice, and that might take from a month to 3 months for a PDX to grow, patient-derived xenograft to grow, so that we can then put it on the slides. We've done it in approximately 10 cases. The truth is that, as you all know, to get a pathology report in Greece takes something like 40 days. So by the time we get the pathology report, we've done the experiment. particularly when the primary material is enough. So we go agnostically with it, right? And sometimes we see migration and we're wondering, oh my God, could it be that this could be metastatic? Because sometimes local metastasis, infiltration of lymph nodes is seen by the pathologist right after the operation, right? Because the surgeon clears the area from lymph nodes. So sometimes afterwards, the report comes back and there is an upstaging of the tumor because they found already infiltrated lymph nodes. So when we do the experiment, we are agnostic. It is absolute, it is 100% now that I think about it. All those that progressed were found positive by the pathologist right after the surgery or progressed within a couple of months. All those migrate in our hands and the others do not. Okay, this is not yes or no. We're talking numbers, so you might see 15 counts in the fax that are EPCAM positive, and there you start scratching your head. Is this real? Is this not real? Because obviously you cannot go just mouth pipette each and every cell of those. At the second stage, we're going to look at those as well. But right now we're focusing on the properties, the molecular properties and the markers, possible markers of those cells that migrate, both ways actually, right? Because, and also it would be nice to see if you leave them for 2 days, there is no attack by the immune system. After 3 or 4 days, the migrating cells are eliminated. So we're going to sequence, to RNA sequence, I mean, the immune cells also to see what kind of immune response is involved there.
So what's the target number, and do you plan to maybe validate this as a diagnostic test?
So long the clinicians give us samples, we will keep doing this. Yeah, at some point, I mean, for the needs of the project and the paper, maybe, I don't know, 5, 10 would probably be enough. Because we want as a lab to build a big biobank of those live frozen cells, because this can initiate projects, funding, and everything. We'll keep doing this for as long as we can.
Any other question? Yes, Pano.
Apostoli, thank you very much. Great work and fantastic presentation. Can these chips, can they be reused, or you done? I mean, I'm thinking about diagnostic, expensive. How expensive is it?
One of the things is that you cannot reuse these because these are at the very end stage, let's say. It's going to be a closed system with micropumps and everything. So to disassemble it, you need to break it apart. Yeah. But so the focus in these projects and in these approaches, the focus is to reduce the cost of the chip rather than try to reuse the chip.
Yes.
So you need to manufacture them through ways that are cheap, efficient, and reproducible.
Yeah. And the second question related to the very nice mechanistic work that you have done in epigenetic modifiers in MLL3. Have you how to say that? Have you correlate I don't know, in patients or functional level? Effects or differential expressions related to the readers, to the corresponding readers. Is there, like a binary code or?
This is something we have published already, and anybody can go to the TCGA datasets in the CBio portal to look at the correlation between the expression levels of these epigenetic regulators. It is baffling that they seem to be regulated en bloc. So the Pearson correlation in the expression between KMT2C, D, UTX, ARID1A is like 0.5, 0.6, 0.7, which is like for human samples, with all the heterogeneity, genetic heterogeneity in humans, this is, you just don't find it. So it seems that because there are such global regulators of chromatin structure and everything, it seems that, you know, they're, I don't know if they are co-regulated, because these are housekeeping genes in essence, right? I mean, histone-modifying enzymes exist in all cells. They are expressed. Okay, they're not involved in metabolism and acting cytoskeleton. Yeah. Still, they are very essential proteins.
But there are some differential expression of some of these players.
Yes, obviously there is some tissue specificity. It's not a coincidence that MLL1, KMT2A, was identified as an oncogene, in fact, in leukemia, and MLL means mixed lymphoid. Yeah, I'm finishing. Okay. Mixed lymphoid, mixed lineage leukemia. So yes, there is tissue specificity.
So thank you very much. There is time for a quick lunch. We are running a bit late, so thank you for this session, and we can continue with questions during the lunch. Okay, 10 minutes break.
Hello? Hello? Expedite, expedited, expedite, expedite. Hello? Do you hear me? Hellenic on enhanced interaction, early dialogue with developers, promising and to optimize development plans and speed up evaluation so these medicines can reach patients early.
Θουκυδίδης, εισαγωγή στην ιστορία του. Διάβασε τι θέλει να πει. Τι?
Φαίνονται αυτά εκεί. Ανάγνω. Ανάγνω, όχι ανάγνω. Ούτε αναγνωστόπουλος, ούτε αναγνώστη. ANAG, όχι, περίμενε. Εγώ άρχισα το συνέδριο.
Η κυρία που είναι.
8. Είναι στις Βρυξέλλες.
Όχι, η κυρία αυτή που είναι. Δεν θα είναι εδώ.
Όχι.
Πασχαλιά, τον Λοκότσιο. Α, γι' αυτό είναι με τέτοιο.
Ναι, ναι, είναι αυτή. Okay, we welcome you. Hello. Good afternoon. Dr. Kostandopoulos and myself welcome you to the last session of our meeting, which deals with some burning issues involving the advanced therapeutic medicinal products, which need to be covered and addressed in the next few years. That's why we invited people who can cover at least 3 areas of these ATMPs. The first speaker will offer you the regulatory aspects of these ATMPs. Dr. Gazouli will cover you the specific aspects of hospital implementation with the hospital exemption issue. And Professor Hughes will cover the economics of ATMP and will provide some novel strategies that can be implemented in the future to meet this burning issue of reimbursement of TMPs. So we have the pleasure to welcome Mrs. Pascalia Kufokociu, who is the Policy Officer and Pharmaceutical Health Policy at the Direction Générale in Santé in European Commission. And she is going to talk about priority medicines, support from EMA for unmet medical needs. Mrs. Kufokociu, thank you very much for joining us, and the floor is yours.
Thank you very much. Good afternoon, everyone. Kalispera, Athena. And I would like first to thank the organizers of this meeting, and especially Mrs. Evangelia Giannaki for inviting me to make this, to deliver this presentation on PRIME scheme. I will try to be short. I understand it's the closing of your meeting, so I don't want to get you more tired. So that is the outline of my presentation. I will briefly talk about what are the exclusive benefits that the PRIME scheme offers to developers of medicines. It's not exclusively directed to ATMPs, but to medicines in general. I will give you some statistics on the PRIME eligibility requests, then what was the main outcome of the 5 years review of this scheme, and finally just a few words for the submission readiness meeting, which is one of the 3 new features of the PRIME scheme. So to get started, the PRIME scheme is a scheme run from the European Medicine Agency, which is the European regulator for the authorization of medicinal product for human use, for veterinary use as well, but now we are talking about medicines for human use. And the purpose was to enhance the regulatory and scientific support to developers of medicines that target an unmet medical need. So it was launched in March 2016, and it's a voluntary scheme. Now, with adoption of the new pharma legislation in April this year, the PRIME scheme will obtain a more solid legal base in the legislation, in the European legislation. But when it was launched in March 2016 and up to now, it remains a voluntary scheme, and it is based on enhanced interaction and early development between EMA staff and people from the different committees organized by EMA and developers. And there are 2 main objectives. First is to optimize the development plans, for example, better designs of clinical trials. And second, to speed up the evaluation of medicines in the context of the marketing authorization. So the ultimate objective, of course, is the early access of these medicines to patients. Now, for ATMPs in particular, we have an interaction of different pathways. And within this regulatory framework, we have also the prime scheme. The general principle in Europe is that in order to put in place, to put on the market, to place on the market medicines, first they have to be granted a marketing authorization, a license. But for ATMPs, the first question that comes is a classification question. So whether the developer, in order to ensure that it's not the ATMP classification, it's not mandatory, again, it's optional, but it gives assurance to the developer that the product under development is an ATMP. And then there are three different types of marketing authorization: the standard, the conditional, and the exceptional circumstances marketing authorization. And the type of the marketing authorization depends on the level of clinical evidence at the stage of the authorization. And there we have, as you can see, 4 points also within this complex of the ATMP regulatory framework. We have the orphan designation, when there are specific orphan designation eligibility criteria, again related to unmet medical needs and rare diseases. Of course, when the medicine is, the purpose of the medicine is, the target of the medicine, the target population are children, there should be a pediatric investigation plan. And within all this framework, we have also the prime scheme that I'm talking now. The prime scheme builds on 2 existing tools. First, that they exist already for years in the European legislation for pharmaceuticals. One is the scientific advice, and the second is the accelerated assessment. Accelerated assessment is something that speeds up the assessment of the application for marketing authorization when there is a promise of a major public health advantage. And the process can be reduced from 210 days, that is the standard, to 150 days. So this is where you can see the prime scheme located in the workflow of the marketing authorization of ATMPs. Now, prime gives exclusive benefits to developers, and I'm not going to describe them all in detail. The idea of this presentation is mostly to help you navigate on different aspects of the prime scheme. There is from EMA a person that is appointed to be the scientific coordinator for the product. There is an early appointment of a rapporteur from the EMA's CHMP committee or CAT. It depends. If it's an ATMP, the rapporteur will be from the CAT, the dedicated committee for advanced therapy. If for any other case, it will be a rapporteur from CHMP. And then there is a kickoff meeting, which is aimed at optimizing clinical trial designs through prospective planned scientific advice at key milestones in order to ensure that data suitable for the marketing authorization application are generated quickly and efficiently. So that is the core element of the kickoff meeting, to advise on the clinical trial design, but also to plan in the future which are the main points, the critical points where scientific advice will be requested and delivered. Then there is a possibility within this continuous scientific advice to involve additional stakeholders, such as representatives from the health technological assessment bodies or FDA. This submission readiness meeting, I will skip it for the moment because it's one of the new features that followed the 5 years review of the scheme. And I only want to mention from this slide that there is a dedicated toolbox to support the quality data package development. All this information, you can find it, there are many links at the website of the European Medicine Agency. Now, especially now for the academic actors and the small and medium enterprise, medium-sized enterprises, there are additional benefits for the PRIME scheme. So the main difference is that there is an early entry PRIME status, and then the transition to full PRIME status. So the early entry PRIME status is based on proof of principle, and then once the developers, the academic developers, have been accepted to this early entry PRIME status on the base only on proof of principles, which means basically non-clinical data that provides some early evidence of promising activity of the mechanism of action. And then within the scientific staff from the European Medicine Agency, they will get targeting scientific support in order to make this transition to full PRIME status, where we have proof of concept, that we have some preliminary clinical evidence this time to demonstrate the activity of the medicinal product and its potential to address an unmet medical need. Of course, very interesting is that the SMEs and applicants from the academic sector, they have a fee exemption for the scientific advice request, which is very important. And as I said, that there is also an introductory meeting in order to raise awareness of all these regulatory requirements. Now, just I'm not 100% whether you are familiar who qualifies as an academic actor. There is full definition, full explanation available from the website of the European Medicine Agency, EMA. But very briefly, I can tell you that are applicants fulfilling the definition of public or private higher education establishment awarding academic degrees, or public or private non-profit research organization. But there is a requirement that this organization, other educational or research organizations, should not be financed by private profit organization in the pharmaceutical sector. In this slide, you can see the life cycle of development of medicines from early research until the authorization, the marketing authorization of medicines. And there are different tools available to academia. PRIME is one of them. It's not the exclusive tool that EMA uses in order to support academic development of medicine. So there is the Innovation Task Force, the Scientific Advice and Protocol Assistance with fee exemptions, both of them, the ATMP certification, the Orphan Drug Designation, Pediatric Investigation Plan, qualification of novel methodologies. There is also a dedicated entry point if you want to contact the European Medicine Agency. There is an email address. This framework for the collaboration with academic sector has been established since 2017. And the most recent announcement was this pilot to offer, that is something in addition to PRIME, to offer more enhanced support to 5 products that they will be selected. This announcement from the European Medicine Agency took place in October this year. Now I will give you some statistics for the PRIME eligibility request. So in total, since 2016, we have 470 applications, and the proportion of ATMPs is 126%, 122. Now, the number of PRIME supported products that have already received a European marketing authorization is 26. For PRIME, we have 54 ATMPs that they were granted PRIME on a total of 116. You can see that it's quite substantial, the percentage of ATMPs that receive PRIME status, and that is obvious because these are innovative medicines with high potential. We always like at the Commission to say that they are life-changing medicines, so it makes sense that they take so high percentage of the PRIME scheme. You can see I just put in these 2 slides the ATMPs that have been authorized, and they have received PRIME scheme. There are these 6 drugs, the 6 ATMP medicines for cancer, and also within this list there is the ATMP Zolgensma, who gives a very good results for the spinal muscular atrophy, and Hellenic, which is one of the recent authorized ATMPs that is for hemophilia B. These are the authorized ATMPs, and you can see, of course, the days of the PRIME status of the acceptance in the PRIME scheme. Also, you can see in this slide the proportion of ATMPs compared to non-ATMPs biological chemical and others. And also what is the percentage of the requests that have been accepted and the requests that have been rejected. And here I don't know if it's visible. You can see the main therapeutic indications of the products received PRIME. And you can see, of course, that oncology, neurology, endocrinology are the main, followed by infectious disease or cardiovascular disease and immunology. But there are also other therapeutic indications. Now, as I said, that with the initiative of the European Medicine Agency, there was a report on the 5-year implementation of the scheme, and the report covers data collected between March 2016 and June 2021. And this report is available. There you can see, you can download this report from the website of EMA, and you can find a detailed analysis and review of the experience with the scheme in these first 5 years of application. And specifically, it gives information, more detailed information, how the scheme compares and with the standard procedure, how it helped developers to prepare for the marketing authorization assessment, and the comparison between PRIME and non-PRIME products. And of course, the general conclusion is that PRIME indeed scheme has helped to speed up the patient access to new medicines that target unmet medical needs. And also the developers that received PRIME status were much more efficient to answer to questions from the committees, from the assessors during the marketing authorization process. And in order to further improve the scheme, the agency has decided to add features in this scheme, and the 3 main features are a roadmap for its PRIME development, and also a product development tracker, expedite scientific advice, and submission readiness meetings. For the submission readiness meeting, the idea is that this will take place around between 9 months to 1 year before the marketing authorization application. And the scope of these meetings will be to discuss the status of the development, including the implementation of previous scientific advice, and the resulting data package intended to support the marketing authorization application. That is for, I will stop here, and I will be happy to respond to any questions. And also I send my thanks to my colleagues from the European Medicine Agency that provide me the data of this presentation. Thank you.
Thank you very much. Thank you, Mrs. Fokočiou, for this nice lecture. Are there any questions from the audience? Dr. Yanaki.
Thank you so much for the presentation. I'm Evangelia Yanaki. Thank you for joining us in this last session. I have a couple of questions. Actually, the first one is a short. I mean, will be a short answer. If this prime call is open all year, and I continue then.
Yes.
Okay.
Sorry.
Okay.
My laptop sometimes is freezing. Yes, the answer is yes. There are
Thank you. Thank you so much.
No specific deadlines. The communication about Prime is open all year.
Okay, great. And the other question is, you have shown us the approved through the Prime scheme ATMPs, and you actually said you have approximately 50% of approved applications. Unfortunately, I mean, to my disappointment, all these approvals were coming from Big Pharma. So I don't know how many academically produced ATMPs have received authorization through the Prime scheme. Are there any? And can you give us an example?
Thank you for the question. Yes, that's true. Although 54 ATMPs have granted Prime, in the end, the ones that have been granted marketing authorization, they come from the Big Pharma. I'm not sure if Big Pharma, because I think some of them, they don't qualify, they are smaller companies, but not any academic developer. But I can tell you that we have the example of I thought it was also in the schedule, in the program of this meeting, the Clinic of Barcelona, that they had received this year, last year Prime, and also they have been selected under this pilot program, which aims to support in total 5 academic developers of ATMPs. And in the future, they aim to apply for a marketing authorization application. But of course, at that point, we don't know whether, how it will evolve in the future.
Yeah. Thank you.
That's why within this presentation, I tried to give you that it's not only Prime, that since 2017, EMA has a dedicated engagement with academic developers. I don't think if there is so much awareness of this, and there is this the email address that you can communicate with the EMA and get this support for the development of the ATMPs. And of course, we have also the hospital exemption, which is a different subject. I think Maria will cover this topic afterwards.
Thank you. Thank you so much.
Yeah, I just want to extend the statement of Dr. Yanaki regarding the participation of the academic sector in the development of ATMPs. Unfortunately, this is an issue and stems from the fact that the infrastructure of the academic institutions cannot provide the whole chain of production. They can go from the basic up to the translation stage and up to the IND stage. Then you need the collaboration with the big pharma, whatever other industrial sectors are required to produce this. But again, the benefits, we should say that the benefit that will result from this application of the TMP commercialization should go back, and at least part of this benefit should go back to the academic and the research institutions, if you want to have it in a fair statement. I just want to, I was looking at your scheme, the Prime schemes. your assessment so far, I mean, are you satisfied with the procedures so far employed regarding the speed of regarding the handling of the application, the speed of the authorization, and the available pathways like the standard, the adaptive, and the fast-track pathways? Do you think that in the future some peculiarities of the ATMPs will dictate some additional alternative pathways for decisions and commercializations?
Well, first I would like to say that PRIME scheme is a tool in order to improve the clinical development of ATMPs, but it's not a guarantee for a successful marketing authorization application. We had recently, but during the last year, you know that DG Santé, we were very much involved into the revision of the pharma legislation. And one of the very, let's say, controversial topics is hospital exemption, where we organized dedicated meetings with representatives from the academia and the industry to discuss about these topics. And there, the people who represented the academia, they have emphasized that applying for a marketing authorization application, it's very challenging because of the lack of regulatory knowledge, but also they claim there are financial issues. PRIME, on the other hand, is something that it makes sense to be used by a developer that aims to go up to the marketing authorization application. Currently, we have within the pharma proposal that has been adopted in April, and we will see when it will be implemented. The hospital exemption remains as an exemption, so there is no a different pathway. There is, as it stands now, we have marketing authorization, the centralized marketing authorization, and then the hospital exemption, which is not really a different pathway, but it's an exemption that enables the access to patients of ATMPs developed by hospitals, university hospitals. So the answer is that there is no thought at the moment for a different tailored pathway for academic developers.
Okay, thank you very much. Costa? Thank you very much.
We'll continue with Professor Gazzouli, who is Professor of Biology in Athens Medical School and also an active member of the Greek Medicines Agency, which is the European equivalent for regulating for drug regulation in Greece. Dr. Gazzouli will somehow answer to the complaint of Dr. Yanaki, who claims that no academia institutions are getting any marketing authorization, are applying for getting a marketing authorization for ATMPs in EMA. I would say that it is not their proper job. Academia cannot get involved directly with the market. It can provide the know-how, it can collaborate, but the market is something completely different. But there is in between the poor academic job and the market, a place that will be covered by the topic that will now Maria Gazzouli will refer to. Maria.
Thank you. I'd like to thank the organizing committee for this invitation, and I will speak for the hospital exemptions that, unfortunately, there is a cloudy landscape still. I declare I have no conflict of interest, even if Board of Directors of EOF and member of CAT Committee of EMA. And to continue from Lina's presentation, ATMPs are in clinical practice. We have a number of gene therapy that have been developed, cell therapy also, and tissue engineering product. And we want this legislation framework that can permit us to introduce this and to unmet for serious clinical problems and to give the opportunity to patients to have a therapeutic approach in the cases where no other alternative therapeutic approach exists. We face a number of scientific a priori challenges in order to develop all these ATMPs products that we should have a good manufacturer practice. It's a specific requirement in order to have the production of these ATMP products. The starting material and to have a continuously material supply is also an important issue. We faced a number of immature production technologies, and in some cases we do not have comparable therapeutic approach to compare these new developed products, and there are a variability and process validation. Also, we need better characterization, potency testing, etc. We face no clinical challenges. We do not have all the time the relevant animal models in order to test all these new developed products. Also, the proof of concept, the safety aspect, the possibility for bleeding studies, the possibility to do biodistribution studies, etc. And also we have the product-related challenges like the safe, the dose, the tumorogenicity, the biodistribution, as we said, the integration, and the efficacy, especially when we have inter-individual variability. We have risk and limitation of the ATMPs, as we discussed, infections, tumorogenicity, the differentiations, the ectopic engraftment, the shedding, the small sample sizes, etc. And all these challenges should be addressed. Also, apart from these scientific challenges, we have the developer-related challenges, because a lot of new products are in the pipeline, but most still are in phase 2, and mainly developed by academia, hospital, and SMEs. There are limited knowledge and experience on regulation requirements. There are no other products to be sold when the first marketing authorization approval is under preparation, so we do not have resources to compare, etc. In some member states, a lot of products have been under national authorization. Actually, the most of these products were under a national authorization, and there are difficulties to accept the new standards and requirements, difficulties in gathering all data needed for a centralized license. In hospital and research centers, strong wish to maintain cell-based products and transplantation transfusion products, and always we face the conflict with ATMP industry. Also, there are socioeconomic challenges because there are different ethical views. The development still is in hands of the small research entities. We produce small batches, and the most of them are autologous products. We have high production and testing cost per batch. The ATMPs are more expensive from the traditional drugs, and there are difficulties to get novel ATMPs reimbursed, the laborious negotiation with HTA bodies, etc. And the main point is the legal, regulatory, and political challenges, because there are different national requirements from clinical studies, especially when we speak for multi-center studies. There are interpretation of hospital exemptions in different member states. There are conflicts with industry. We face ATMP tourism because the patients have to travel to another country where this ATMP therapy can be found, and also the classification of ATMPs at national levels, and we don't know where are the borders between transplantation, transfusion, and ATMPs. Should be clear definition for classification into national legislation. Thus, the European Commission developed different kinds of frameworks and state that the framework is important to enable patients to receive an ATMP, but this should be done under control conditions in cases where no authorized medicinal products are available for an indication and with a high unmet medical need. And EMA introduced the hospital exemptions as hospital exemptions rule is considered a valuable European legislative initiative that guarantee patients access to novel therapies based on NTMPs in the member states, but should be under control conditions in cases where no authorized medicinal products is available. And the hospital exemption should be used appropriately to ensure that patients are protected for unnecessary risk, and research in the field is encouraged, and should ensure that the hospital exemption is not misused to circumvent the marketing authorization and clinical trials procedures in Europe. However, we need robust, transparent, evidence-based and harmonized procedures for ATMPs and increased transparency and harmonization in a hospital exemptions procedure. And hospital exceptions should be limited to the situations that we need them. In 2020, EMA issued a very strong warning because they have a side effect from unapproved cell therapies. And for this reason, we can see that there are serious differences between the member states and how the member states refer to these ATMPs and use the hospital exemptions. As you can see, here is the data from 7 European countries that have legislation regarding the hospital exemptions. Unfortunately, Greece has not yet a legal framework that describes the hospital exemptions used. And you can see that there is not a harmonization on these countries. There are a number of ATMPs that are licensed and they have the, let's say, the requirements for market approval authorization, and there are a high number of ATMP products that have with no license. And the problem is that there is not a transparency of the use of these ATMPs, since most of the countries, they do not describe exactly how the products are and which are those products. So the Europe Working Group on Gene and Cell Therapies highlight that we need the appropriate use. We have to limit the use of hospital exemptions to the situations when there are no centrally authorized ATMPs. We need to have a long-term follow-up. We need to have transparency. We need for publicity available information about the product, if we have side effects, etc., the efficacy, the safety. And also we need the harmonization to be further harmonized of the hospital exemptions license and eligibility criteria across all the member states. The hospital exemptions should be subject to regulatory oversight requiring the evaluation of minimum quality, preclinical, possibly clinical data before an authorization could be granted. We need the pharmacovigilance requirement to report of adverse effect, etc. A mandatory is a registry that should be established with publicity accessible information regarding the ATMPs that produced, the manufacturer, the source of funding, the responsible physician, etc. Also, hospital exception products should be manufactured according to the good manufacturer procedure for ATMPs. We need the ethical committee in order to examine all this application. Financial or other incentives should be made transparent. Academics and hospitals should be better educated about the regulatory requirements for ATMPs, and the collaboration with industry should also be considered. As we hear from Linda, in April 2023, a new directive regarding the pharmacological application in Europe came out. But here also there are describe very well the market authorization procedure. They describe very well when the hospital exemption is required. But there are also some gaps because the no-routine definition is missing. What we mean non-routine? To have something specific and to use in the same clinic, in the same country, but the BATs cannot describe the non-routine. And this is an open question. And also, this directive does not cover the publicity available registries that record the implementation of hospital exemption legislation and collection data regarding safety. This still remains open question. Academia now is the list of organizations eligible of developing orphan medicine and translate their discoveries into authorized patients-focused medicine. And EMA, under this scheme, provide consultation to academia in order to develop new products and ATMP. And thank you for this presentation. The hospital exemption is a very, very good opportunity to bridge the new ATMP's development to the patients, but should be under a scientific-based manner. Thank you.
Thank you for this lecture, Maria. Before any question, I have one to make. What about any type of reimbursement for this hospital exception made products all over the European Union? I ask.
We cannot
is there any country
we
that...
cannot say from all the European Union because each member state has to do the reimbursement requirements, etc. And each member state has different guidelines for hospital exemption. The majority of the states, they said that the ATMPs regarding the hospital exceptions should be developed in the country and used in the country. But the French law can export these ATMPs to other countries, but under the clinical trial use. We cannot harmonize these requirements since each member state has its own legislation regarding the hospital exemption and the use of ATMPs.
So the help, this EMA helpline, say, or help office, has to be adapted to the local legal regulations?
No, this framework gave the opportunity to consult the scientists that want to develop a product during the development.
Only?
Only.
Okay. Any other question, please?
Can I?
Yes.
Very, very nice talk. Very informative to us. I would like to ask what do you think that the situation will be here in Greece as regards hospital exemption? We are now more than 3 years that EOF has stopped doing anything about. And why don't we foresee a common, from EMA, a harmonized hospital exemption where EMA decides on the requirements and regulations, and all European memberships and competent authorities comply. As it is the situation actually for the clinical trial authorization in its country, I mean for the European state members, is a fully harmonized procedure as far as I know.
Yes, regarding the first part of the questions, as far as I know, there is a framework that was in the Ministry of Health, but there is still there. We do not have an authorization to proceed with hospital exemption. And from EOF, this is the answer that I can give you. And regarding the second part of questions, yes, a lot of countries, they force EMA to take an harmonization, but there is still a problem with member states' specific regulation in its country.
Because they already have their own hospital exception.
Exactly.
Yeah.
If I may, I think that the general European how to say, thought is that even if a certain member state does not proceed further to adapting the local legislature to this scheme, there is a minimum of rights from the side of the hospitals and the centers to apply a type of hospital exemption. I have now in mind what happens in the Greek case with the universities. According to the local, the national legal system, no universities private are permitted. But according to the European applying program for the higher education, any higher education within the European community can establish a certain section or office or department in Greece. Although the local laws do not permit it or prohibit it. So you have, I think, to look it further.
Actually, the EMA should push to have a transparency registry and a European registry. This should be in order to safety of all these products.
Yeah, thanks.
Yeah.
I have also two questions.
Yeah,
Anastasia Papadopoulou from George Papanikolaou Hospital from Thessaloniki. The one is quite similar to what Dr. Yanakis said, and I can understand that since there is a local legislation, it's really difficult to harmonize all this legislation. But since in Greece there is no legislation at all for the hospital exemption, I think that at least for the countries which do not have legislation, maybe EMA could provide a frame that we can also have this option, since it is really important to apply some therapies into our patients.
EMA, it's not a decision-making organization. It's a consultative organization. Cannot provide such legislation. Can provide only the framework that each member states should follow. And this framework is already provided because they said that all these products should be harmonized under this condition of development, etc., or to go to UNID in order to feed the market authorization approval, etc. I think that in Greece soon we will have a hospital exemption legislation.
Okay.
It is in
we hope so.
Yes.
We truly hope so. And the last question is, since we are not, as a hospital, we are not foreseeing commercializing a cellular product, for example, and we do know that we can manufacture under the framework that we have to manufacture cell products, and under the what can happen if something can happen if we have the ability to generate a cell product which we know that it works, which we know that the companies can also manufacture it and sell it, but we can manufacture it homemade and with minimal cost. Is there an option to have an approval, not for marketing it, for selling it, but for applying it into our patients? Thank you.
This is under the hospital exemption procedure. If we do not have this framework, we cannot do it.
Exemption, it's only for a specific number of patients.
Yes,
I don't know if I'm wrong.
And there is also the framework under the small clinical trials and the requirements that they needed for small clinical trials. Unfortunately, there are not other framework that you can use this.
Thank you.
It is my pleasure to introduce Diefrick Hughes. He's a professor of pharmacoeconomics and co-director of the Center of Health Economics and the Medicine and Education at Bangor University in Cardiff in Wales, and director of Impact and Engagement. He is also an academic head of pharmacy at the Becce University Health Board, and is an honorary professor of the Department of Molecular and Clinical Pharmacology, University of Liverpool. He graduated from the Pharmacy of Cardiff University Department before completing his PhD in cardiovascular pharmacology at the University of Liverpool. He leads the pharmaceutical economics, pricing, and prescribing research group on research activities covering personalized medicine and methodological research. Lastly, among the other honorary awards and positions, Diefrick is also an expert and advisor at the committees of orphan medicinal products of the European Medicine Agency. Based on his research publication, Diefrick is ranked second in the world and first in the UK for expertise in pharmaceutical economics, according to the Expertscape. And he has led and contributed to research totaling about £56 million. We're very happy to have you here, and thank you very much for accepting our invitation to participate in our meeting, and we're looking forward for your topic on the economics of ATMPs as an expert and your optimistic view for the future.
Well, thank you very much for the very warm welcome. Thank you for that introduction, and thanks for the invitation as well to present this afternoon. So I think the big elephant in the room, as they would say in the UK, would be the price of ATMPs. And this slide is from 2 or 3 years ago now, so it might be a little bit dated, but the picture is much the same, that the price per unit of dose or per patient per year is extremely high, much higher than conventional medicines, even biological treatments. So the list here compares different countries, because obviously prices do vary by country, different markets, different availabilities of products. When there are blank spaces, it indicates that these treatments aren't available for general use. But it indicates the hundreds of thousands of euros that these products cost. So there are a number of agents that are now marketed, as you're aware, and each of them do carry a very high price, particularly when compared with other conventional medicines. So that leads to a great problem in terms of market access. So once a ATMP has marketing authorization, access to treatment depends on the reimbursement of the product by national public healthcare organisations. And as mentioned in the previous presentation, this differs from one country to the other. They have different systems, they have different procedures, different policies, and different evidence needed to make decisions as to whether to reimburse a particular treatment. But fundamentally, patients cannot benefit from treatments that are not available. So even though that ATMPs can be life-changing in terms of their benefits, potentially, if they are priced too high, such that a country would be unable to reimburse, then no patient will benefit from it within that country. And that has led to great geographical variability in the availability of ATMPs, even across Europe, where arguably the market would be comparable, same regulatory authority, etc. But internationally, that variation is even higher, and it will depend very much on the ability of certain countries to pay for healthcare. Across Europe, there are 3 general recommendations. A product can be recommended fully within its marketing authorization. A product could be recommended for use with some restrictions, perhaps a subgroup of patients in which treatment is deemed to be more cost effective, or the treatment can be not recommended at all. So it would have marketing authorization, but it would not be reimbursed by a healthcare system. These decisions are made by health technology assessment organisations, and there are many of these. Every country now has at least one. Some has regional HCA organisations in addition to their national bodies. And this review, again from a couple of years ago, assessed the availability of a range of ATMPs across a number of European countries. Those in green is where they are fully recommended within their marketing authorisation. The amber, it's meant to be amber at least, coloured cells are those that are recommended with particular restrictions, so not the full licence indication, but a subgroup of patients. And those in red indicate those whereby the treatment has not been recommended for use. So you can see that there is a pattern here, certainly across treatments, but also across countries. Some countries are able and are willing to pay for ATMPs more so than other countries. The cells in white is where there hasn't yet, at the time of this analysis at least, been any recommendation made. So this variability then indicates that for certain treatments there is widespread adoption across Europe, but for others it depends where you live, which is quite unfortunate because a patient with a particular disease should not be restricted by where they live. It should be something that ought to be available to them if they are deemed to be good value for money. So that's the picture across the landscape across European countries. But we do have to acknowledge that no healthcare system can afford the prices of medicines at any cost. There has to be some balance between the availability of treatments and the cost that they impose on a healthcare system. And that is driven by 4 main determinants. First and foremost, the demand for healthcare is ever increasing. You might argue, in fact, that the demand for health is infinite in the sense that everybody wishes to live as long as they possibly can and as healthy as they possibly can. So because of this high demand, the intrinsic demand of wanting to be healthy, there is also an ageing population. Demographic change across most countries now is for ageing population, and as you age, of course, you accumulate more chronic diseases. A requirement for healthcare increases in parallel, and technological change across all dimensions, not least obviously the topic of this conference and the development of ATMPs. So while the demand increases, the supply is limited and is probably contracting in many healthcare systems. There is scarcity in finances, in the money available to pay for healthcare. There is scarcity in healthcare professionals. There is scarcity in the capital, the infrastructure, the hospitals, the healthcare service facilities. And because of this, it places great demands, doesn't it, in terms of how should healthcare payers afford and be able to reimburse all the demands that are placed upon it by the patients, by drug manufacturers, by pharmaceutical industry and others. So ultimately some difficult choices need to be made. So for instance, should a healthcare organisation invest in a new ATMP, which for 10 million euros may treat 10 or 20 patients? Or should they invest that same money for perhaps a community mental health programme that could arguably treat thousands of people and derive much greater benefits for the population? So the choices that these, the committees responsible, the health technology agencies are responsible, are challenging, and they really do make for very difficult decisions in public life. So ultimately, some form of prioritisation is necessary. There needs to be some metric for the fair distribution of resources for the delivery of healthcare. And it boils down to needing to assess value for money. So any and every treatment, the intention would be to improve health. And so the question is, by how much does a particular treatment improve health, and how much does it cost to improve that unit of health? So it's challenging to judge what is and what is not good value for money. But fundamentally, in a limited healthcare system of limited funding, it requires that the health benefits gained through a new investment into an ATMP exceeds the health benefits foregone by not using that money for an alternative purpose. If it's the case that the health benefits gained do exceed the health benefits foregone, then obviously that intervention would be deemed to be cost effective and represent good value for money. And this is where we use economic evaluations. These consider not just the cost, the acquisition cost of the treatment, but also the wider medical and also non-medical costs associated with treatment, such as, for instance, time spent in hospital, the time required and the costs associated with monitoring, with administration, etc. So we quantify costs related to medical and non-medical costs of care. We also quantify benefits, and the metric we used most often is the quality adjusted life year, the QALY. And this is a metric which combines two elements of health, survival and health-related quality of life. And arguably everything we do in medicine is about either improving the well-being of the patient, how well they feel, but also how long they live. So it condenses the effects of treatment down to those two variables. And an economic evaluation then compares the additional cost with the additional benefits that can be achieved. And it compares those against a threshold for cost effectiveness. And most countries nowadays operate a cost effectiveness ratio, or a threshold, below which things are cost effective and above which they are not. This is a schematic representation of a QALY, a quality adjusted life year, and you can see the figure here plots quality of life against time, and for standard of care, perhaps the shaded colour illustrates a disease that impacts on quality of life, but there is a period of remission, and then there is a relapse period, and then another remission, etc., such that the patient ultimately deteriorates and dies. With new treatments, there'd be an improvement in quality of life and an extension to survival. And so the difference between these two curves in terms of the area represents the number of QALYs that can be gained with the new treatment. And this is the metric by which many HTA organisations make their decisions for value for money. We now know the costs of treatment, the outcomes associated with them, and we can compare that with standard care. That gives us the incremental cost effectiveness ratio. This is the direct valuation of value for money. And this is the threshold that operates in the UK. Above about above £30,000 per QALY, then treatments are not deemed to be cost effective and would not be generally reimbursed in the UK. Below £20,000 per QALY, treatments would be cost effective and are generally reimbursed in the UK. And there is some flexibility between those 2 points, A and B. So a year or two ago, we did a systematic review of economic evaluations of ATMPs. We went through over 4,000 articles, we retrieved them, screened them, and removed those that clearly weren't eligible for inclusion in the review. And we ended up with 23 economic evaluations of ATMPs. This is a busy slide, but it illustrates the evidence that was available, the published evidence. It indicates the ATMP on the left-hand side column. We have then the cost of the intervention, which would be the ATMP, the cost of the comparator, which is typically the standard of care, and then the third column there in terms of the results would be the gain in QALYs, how many additional QALYs are projected for each of these treatments. And the final column is the incremental cost effectiveness ratio. So there are a few things just to note here, and I'll just highlight these. The interventional costs are invariably much higher than the comparator costs, and that reflects the costs of the ATMPs primarily. The QALY gain are very large. There's a big range here between these different drugs and different evaluations, but a QALY gain of 1 or more is a significant clinical benefit to a given patient. And then on the right-hand side is the cost-effectiveness ratio. We need to be mindful that most of these were industry-sponsored evaluations, and so there may be some cause for bias in what data are presented, but I've highlighted at the bottom there a couple of instances where the cost-effectiveness ratio is far in excess of 2 or 3, in fact, they're about 4 or 5 or 600,000 euros. So just to summarise, of the 23 studies that we'd identified, there were 4 ATMPs whereby the treatment was more effective, yet overall also less costly than the comparator. So you might question, well, why would they be less costly? And the reason would be that they would prevent ongoing treatments for decades. And so these would be treatments that would effectively save money from elsewhere in the healthcare system through patients not requiring healthcare for many years to come. 2 economic evaluations indicated that the ATMP was the reverse. In other words, the comparator was more effective and cheaper. And the remaining studies had incremental cost effectiveness ratios that ranged from about £14,000 to over 600,000 euros per QALY. And be mindful that the threshold for cost effectiveness is about £20,000 to £30,000. So ICERs vary by country, by the methods that they used, by the healthcare service context, and of course by the ATMP that's been evaluated. We identified from this review key methodological challenges, that the estimates of cost effectiveness were very much associated with significant uncertainty and high likelihood of bias. And this bias resulted from 3 main sources. Firstly, largely unknown long-term outcomes. Clinical trials tend to be short-term in duration. We don't know as to whether the findings of those trials are generalizable, whether they are sustained in the long run, and neither did the analysts of these studies. So they made assumptions and projections, and the reliability of those were quite poor. Secondly, there is a paucity of evidence on health state utilities, and these are the quality of life indices used to calculate QALYs, the quality adjusted life years. So if you don't have the data on the quality of life that patients experience, then there's no way of understanding the value that those medicines provide each patient. Thirdly, the modelling approaches that they used in the economic evaluations are based on a number of assumptions, and the plausibility of these assumptions can be questioned at times. I think they, making the link between a surrogate outcome to a final outcome to make the projection over time, they required some fundamental assumptions that could not be verified. So we focused on a particular methodological challenge, which is the projecting of long-term outcomes, and explored whether we could come up with better methods for doing this. There is often very little mature survival data for ATMPs at the time of marketing. When using those data in an economic model, we are faced with what's called a time horizon bias. in that analysts must make extrapolations of trial data to capture long-term differences between treatment and comparators in costs and consequences. For a clinical decision, it may well be fine to have short-term data, but for an economic consideration, where costs or cost savings would accrue over many, many years, as would benefits of survival and quality of life experience, it's really important to minimize time horizon bias. And we identified a number of modeling approaches that could be suitable for capturing what we call a complex hazard function. I'll come on to that in a moment. And two families of models were the mixture models and the restricted cubic spline models. These are statistical techniques to try and project a variable, such as survival, over time. And it's important that we use the correct information for such projections. We use the clinical evidence that's available, which is product specific. We use expert opinion. We use data on natural disease progression. And we combine these in a Bayesian type analysis to try and get a best estimate, or the best projection based on existing evidence at least, of longer term outcomes. If new evidence were to come along, then we can revise the model, update it, and give a better estimate of long-term outcomes. So this is an example of where we have a standard constant hazard function. So in other words, the rate of progression, rate of deterioration is constant over time. And this would be a typical results of a clinical trial of some treatments, typically for cancer, where you have 3 health states. People can be in a progression-free survival state. They could have progressed disease, and they could be in the death state. And so the clinical trial in this illustrative example would provide 12 months of data, but that would introduce time horizon bias. And so we need to project into the future, and we assume a constant hazard function, and the curves would continue at the same rate. And I guess that seems reasonable. There's nothing that would tell you that there should be an abrupt change in the direction of those slopes over time. The problem when you have the potential curative treatments is that a proportion of people could have near normal life expectancy, and another proportion would have a variable time to survival. And this is what we call a complex hazard function. So on the left-hand side, if we were to have a constant hazard function, it would be just a horizontal line on that figure on the left-hand side. Because it's a complex hazard function, then there's a change in the slope of the hazard function over time. And when we use those hazard functions in the survival model, as we had in the previous slide, then we have a figure such as the one on the right-hand side. The blue line indicates the survival in the general population. And by contrast, the black line at the bottom, or it's dark purple, I think, is the uncured population, those without any benefit from treatment. And then in between, we have different scenarios of a 25% cure rate, 50% and 75%. And you see how the slope changes over time. And that's a consequence of the hazard function being complex. So without going into too much of the technical detail, these are a set of modelling techniques that can be informed by opinion, evidence, and already published epidemiological studies, for instance, that can be synthesized together to get a more accurate view of survival over time. So I'll finish with a short section about posing a question as to whether some QALYs are more valuable than others. So should it be the case that a year in good health experienced by a child should be valued higher than a year in good health experienced by an adult? Or should they be the same? How about at the end of life? So someone with a few months to live if they were afforded an extra 6 months, should that period be valued higher than any other 6 months in that person's time in their life? How about a severe disease compared with a mild disease? Should we value a health gain in severe disease higher or the same? Similarly with rare disease, cancer versus non-cancer diseases, or maybe because the product is innovative. Should we value a health gain just because a product is innovative versus a product which isn't? And these are really challenging questions because, and these are key to making decisions for reimbursement. NICE is the National Institute for Health and Care Excellence, and it's the organisation, the HTA organisation in the UK, that makes these decisions. It has applied what it calls decision modifiers to assess ATMPs. So in other words, it deals with these treatments in a different way. It has a preferential view of treatments because of the way that they benefit patients. We identified 14 modifiers, and these permit greater leniency in the consideration of cost effectiveness. 12 of these modifiers have been applied in at least one HTA, and we found 13 appraisals that had at least one decision modifier applied. Unfortunately, in the reporting of the assessments, there wasn't much transparency in the documentation from NICE, so we weren't particularly clear about which modifiers were specifically used to inform the decision problem. We've also conducted separate research to see whether the general public are aligned with the views of NICE in thinking that these modifiers are fair. In other words, that a year in the life of a child is worth more than a year in the life of an adult, or that a year in the life of someone with cancer is worth more than a year in the life of someone with heart failure. Okay, that's the premise, that's what the policy dictates, but that's not what the current population preference studies indicate. So NICE is applying these modifiers unsupported by evidence from the general public. These are some examples. I'm not going to read, I know these slides are quite busy, but age as one example. So for instance, considering the value of a given health benefit to a younger person as a greater than the same benefit in someone who's older. The curative potential, so the fact that a treatment might cure a patient, that might be a modifier for more lenient consideration of cost effectiveness. Life extending, end of life treatment. This would be the preference to pay more for additional life at the end of life than in the middle of life. The identifiability of this additional life is also relevant. And a few more here. Innovation. So for instance, HTA bodies may consider innovation itself as a benefit or to support research and development of health technologies. And rarity, at the bottom here. Treatments for rare conditions may not be as attractive for pharmaceutical companies due to limited market size. Therefore, society may need to pay more per quality to make these treatments commercially viable in developing. So I'll finish with the conclusions that from our research over the last few years, ATMPs pose a challenge to healthcare providers and payers. It's an enormous challenge. Currently, we have a limited number of ATMPs. They tend to be for the rarer diseases, but when there are several more and they become valuable for prevalent diseases, if these prices remain, then it'll be a huge challenge to come. Most of the ones evaluated to date haven't been cost effective based on standard criteria. And so reimbursement depends increasingly on different mechanisms for payment. So for instance, the incorporation of the modifiers, so it places ATMPs in a special category. And secondly, the introduction of novel managed entry agreements, such as payment based on outcomes. These are new novel methods to reimburse drug companies for the cost of treatment. But ultimately, these, to me at least, they raise questions about opportunity cost. What is displaced by investing in this area? equity, the geographical variability, for instance, and the equity from one patient group to another. And finally, affordability. That will become an increasing challenge for healthcare payers in time to come. Thank you.
Oh, thank you very much. Thank you, Dr. for this excellent, comprehensive presentation involving holistic approaches and having an opportunity to have a more promising future with the ATMPs. I have a quick question regarding the extrapolation of the clinical data, of the trial data, clinical trial data. In view of the heterogeneity of the trial data, I mean, from even using the same ATMP, how can you reliably normalize those data?
The variability between patients within a trial?
Yes.
Yes. Okay, when we design an economic evaluation and produce the survival curves, as an example, the ones I've presented there, they would be the mean data. We would sample for the heterogeneity in response, such that some patients would have no response and others would be complete responders. And we would incorporate that in a probabilistic way. So if 20% had this cure potential, however defined, then 20% of them would have that survival curve representative of the normal age matched population. Yeah. Whereas the remaining 80% would have a variable degree of life extension. So the result at the end of it then would be multiple cost-effectiveness ratios. For those who have been cured, the treatment could potentially be very cost-effective. For those in which they have not experienced any clinically meaningful response, the treatment would be not cost-effective at all. If there was a way a priori to identify which people are most likely to respond, then obviously we can restrict the recommendation to those populations where the treatments would be most cost effective. And that's one way of subgroup assessments, but having a biomarker prior to treatments that would predict their response in itself is a challenge as well.
I see. Thank you very much. Dr. Yanaki?
Thank you so much. That's what was great. I agree, I definitely agree with you that most ATMPs are not cost effective. And I don't have a good idea of how we will convince the companies to lower down their claims, their financial claims. However, I would like to stay on your very first comment that choices are needed, and should we go for a new ATMP or a community mental health program? So I think we need to discriminate the value of a new therapy versus the cost. So who should decide to for a child with congenital blindness to not receive Luxturna, a gene therapy drug that will make the child see her mother face, the sun, whatever. And this is a great value, I think we all agree, because the cost of the drug is 500,000. 700,000. So is this what you propose? Let's go for a community mental health program rather than taking care of patients that need and that they should have access to a life-saving or life-changing gene therapy or ATMPs products.
I mean, this is the crux of the problem. This is the real challenge that healthcare providers are faced with. In your example, for instance, there, you might have, or if you take a hypothetical example, that we invest, the healthcare system has a fixed amount of money. If you introduce new technologies, there's no reaction with an investment to fund those specific technologies. So anything spent on new technologies needs to be taken from elsewhere. The money has to come from somewhere. The example in the UK is very much, we have to wait longer for treatment. If I needed some hip surgery, I need to wait 18 months, 2 years. The more that's spent on new technologies, that wait would become 2, 3, 4 years, etc. So there is always an opportunity cost. The question then is, for individuals who would benefit, there's no denying, obviously, that to restore someone's sight, etc., their vision, they would individually experience maybe 20 QALYs of health gain if we measure it in that unit. The same money, if used for other purposes, could buy 200 QALYs from having the money extended across more populations. Now, individually, those people would benefit less, but collectively, there's a far greater health benefit. So the question is whether they are equivalent, okay? Whether we should, because we can buy more QALYs by going for prevalent diseases, low-cost interventions, or we go for fewer QALYs, higher cost, but greater health gains for individuals. And that really is what decision makers are faced with. I don't have an answer to that. We have judgments, we have some flexibility to make those considerations. We have the techniques and tools, but they're all imperfect. Everything's imperfect in this world. So the way that it's done typically is deliberative. You'd have a committee with a patient representation. You would have a patient representation from an older age group, the ones who would lose out on this money being spent on children. You would have clinicians, statisticians, health economists, you'd have a full range of, and they would between them work within the policy guideline to make what they consider to be the best decision with the available evidence. And they would make these trade-offs. They are really challenging, but within a public healthcare system, I think that's a fairer way forward than to fund the latest technology just because a big pharmaceutical industry is trying to get it on the market.
Yeah, I can see your side, the economist side. On the other hand, I'm a clinician, a scientist, and I cannot, I say that no scientific discovery is value if the patient do not have access to it.
Exactly. And that was one of my opening lines was exactly that. And there is some onus on the commercial developers to give to sell it at a fair price as well, because obviously any reduction in price would make it cost effective.
Thank you.
Thank you.
Thank you very much. Thank you very much again, David. Now we arrive at the end of the session and at the end of our 6th meeting. And I would like to thank all the speakers and the audience for their participation. And I would like to ask Dr. Yanaki, the president of the society, to make the concluding remarks, to come to the podium.
I would like to thank you all for attending this year also the annual meeting of our society. And I hope to see you again next year, and I hope we be expanded and be more attendance, more participants. My belief is that the meeting was successful. I hope your belief is also the same. We hear all a very broad spectrum of topics that cover gene therapy, cell therapy, and also regenerative medicine, and this final session of focusing on the regulatory aspects for the advanced medicinal products, and also the last presentation covering the financial aspects of the ATMPs. I would like to thank you all just for closing this long meeting. To let you know that we had 186 participants. We would like to thank our sponsors, give them a big applause because without their sponsorships, this could not be materialized. Also, I would like to thank from here the secretary of the Secretariat of the Congress and the girls who helped us so much, Nina and Aphrodite. And also our society's secretary, Marina Stogiani, she helped us a lot. And I'm closing here. I invite you all to give a big hug to our next president of the society, Rebecca Matza. So I just... I encourage you to participate in the elections that we'll have in June, in a few days, either as candidates or as voters. So we need to be to massively participate in the elections. Thank you.